we are and where we were in earlier, right? Am I audible to all?
Yes, you are
Yes, sir.
audible.
Yes, sir. You're audible, sir.
Yeah. Yeah. So even good morning, so Agya sir.
Good morning, Kashyap. How
Yes.
are you?
Yes. All well, sir.
Good morning to one and all. Good morning to the sir.
Good morning, sir.
Yes. So here we are talking about digital transformation, pharmaceutical R&D, and its implication and efficacy on the routine process. So, as you know, in the earlier days, we were in not much engaged with the digital technology, multiple platforms. As you have very aware about this meetings, now we are connecting through virtual mode and various meeting modes like Google Meet, Zoom Meet, so so on. And now we can deliver and communicate through different modes and can share the updated information to the direct students or some professionals what we are doing in a routine days. So that can be easily transformed from one person to another person. There is no need to travel from one place to another place. Similar way in R&D, why we are focusing on R&D? Because I am since 15 year I am engaged in sterile pharmaceutical formulation, where we also have seen the multiple challenges and in previous days. While in if we see the journey of last 3 years after the COVID-19, so we have seen a lot of things which has improved, and the IT plays where a very important role. We have various data in cloud. We have particular data science library, artificial intelligence, machine learning. These are the current topics where we are seeing the digital transformation application, not only in pharmaceutical R&D. In every sector, we are seeing the updates where this application has been given the real ground of the functionality. So there are two different approaches. First is incremental innovation, and second is disruptive innovation. So incremental innovation just focusing on a single smaller number of business where like which helps to improve the marketing technology, and definitely marketing technology gives the major potential on the basic R&D. If we have our own in-house R&D center, that definitely will help to in-house R&D scientists, and it a booster for the in-house R&D center where we have to grow and to implement and to introduce so many numbers of new products from our new R&D in-house center. Only the things is where how we can apply the different technologies. So like in three or four ways I can say there are the, if I say in digital strategy, how we can focus. So major four fundamental pillars are there. First is connectivity. We have to connect properly with the basic information to the end users. How we can implement this. Second is innovation. So day to day we have to improve and apply the new ideas. How we can improve not innovation, not only to introduce a new product, new technology. Innovation can be something like if we have existing product, or existing process in our lab. How we can improve this? By applying certain digital application. Third is automation. As everyone knows, automation plays a very, very important role in the industries. So if you have heard about the blow fill seal technology, it's a very common technology and majorly engaged for the basic IV infusion container manufacturing. So it's a totally robotic and automatic operation, right? And fourth, I can say decision making based on data. If we have certain data that we can introduce based on the data evaluation, the landscape should be there. That plays a very important role. So as a formulation scientist, I just would like to give a message to all the young scientists and all the senior dignitaries presented here, how we can improve, how we can introduce, how we can connect it well with the new innovative technologies, how we can apply the automation system in the existing practice, and how fast we can take the decision based on the current data which we have available in our library or database. So that's all from my side. And I just give you two words: always be approach to the focus on innovation, and it should be constructive approach, not the destructive. If you have constructive approach in mind, so definitely you will grow in a far way manner. So it's a message from my side. Thank you.
Thank you, Dr. Nagaria, for your motivational words. We are waiting for motivational speech of our another eminent speaker, who is none other than the Vice Chancellor of Delhi Pharmaceutical Science and Research University, and who is also the chief guest of the today virtual inaugural ceremony of day second of the APP 11th Annual International Convention. Welcome, Professor Goel.
Thank you very much, Dr. Dr. Rajiv, Dr. Rajiv Daya, other personalities.
Well,
I can see
how are
Dr.
you?
B.N. Swagia.
Well, sir.
And hello, how are you? So
Yes.
it was a great to see all the dignitaries, Dr. Dr. Daya, then all the distinguished personalities there. I'm honored to be in this conference. I thought it is in Hyderabad. Yesterday I was seeing what I have blocked the date for Hyderabad. So how come it is I have blocked for Hyderabad and not having? I forgot that it is a virtual one. And anyway, now thank you very much once again for inviting me. I would like to share the screen. I could see the importance. Can you have my PPT? Can you allow me to share the PPT, please?
Yes, sir.
Yeah. Okay. No, you have disabled me, so it will be good if you can now.
Professor Kiranmai, please make sir the host.
Yes, sir. Yes, sir. 1 minute.
Okay.
So
Yes, sir.
Yes, sir.
Two
yeah.
parallel sessions are there.
Yeah, for oration competition, there will be two parallel sessions.
So I am not in this session, but I am in the second session. So I want that link.
No, no. This session will continue the scientific lecture session where you are the judge.
Okay.
This session will, this link will continue with the lecture session.
मैं चेक करता हूँ अगर अदर लिंक है तो ज्वाइन वहाँ करता हूँ.
So how are you, sir? Dr. Swagia, how are you?
Well, sir, I am very fine.
Okay, nice. My screen is not
So we
getting
made you
shared.
the co-host. You can
Yeah.
present the presentation.
Okay,
sir.
now, yes. Okay, I'm sharing it now. Is it okay?
Yes, sir.
It is shared?
Yes, it is visible.
It is there, right?
Yeah.
Okay, and I would like to see first, because many times it doesn't move. So, uh, okay, can you see now?
Yeah, it is
Full
visible.
screen, and it is
Yeah,
moving
it
also?
is changing. It is changing.
Okay. Okay, so.
Well,
Yes,
well,
sir.
good morning.
Good morning, Dr. Diwan. How are you?
Hey, I'm fine. How are you? Tell me.
You can see me. Not as young
No,
as you are.
I do agree.
Only
Repeatedly
thing-
seeing will definitely improve our good health.
Well, the only thing
Okay,
I'm
go
not
ahead.
as
All
young
the
as
best.
you are. That's all.
I'm here. I'm here.
I'm growing
Okay.
older, so I forget the things also. Unless I...
Okay.
Okay.
Right, go ahead.
Okay. So now coming to my present, as I can understand, we are talking about the academy interaction, academy industry interaction for pharma innovation. And in this, I will like to focus on what can be humanizing it to the fifth industrial revolution into a reality. That is what is there. Well, I would like to tell that now, before 2020, I used to show it very often that we are in the era of industrial revolution, of fourth industrial revolution, wherein it is a combination or integration of the industry revolution 1, 2, and 3, that is the mechanic, electrical, electronic, computer, IT, and reaching to the artificial intelligence. That is what has been there, fusion of all technology. So this I used to tell for last 3, 4 years. But what it has done, it has disrupted the future. There is a digital disruption now. So we have growth hackers now. That is something interesting. And so what is the result ultimately of fourth industrial revolution is artificial intelligence, robotics, 3D printing, virtual imaging, use of mobile, so many things. You can go on listing. Even the sector wise, in the health wise, in the pharmacy wise, we can go on listing like that. And this fourth industrial revolution, again, as I'm telling again and again that this was the thought process before 2020, the COVID arrived. 45% of the impact of fourth industrial revolution has been on healthcare sector, followed by, of course, others, but definitely it has made it. I will not go into details of that. But another thing which I could realize and I used to see is, oh, I'm sorry, again I could see was that there had been change in the practice of medicine also, wherein we are affected. Like the source of herbals, herbal used to be the source of drugs and simple surgeries like that. But today we are seeing gene therapy, which was a sort of a myth sometime, but it became a reality. Telesurgery, telepharmacy, telemedicine. It is all realities now. I'm not saying that fourth industrial revolution is not continuing. It is continuing. I'm going to come to the fifth one now. So what we want to tell is that even the symptomatic diagnostic, previous Vaidyas used to simply touch the pulse and used to tell many things. Today, I mean, after that, slowly and slowly, the dependence became on the biological test. Slowly and slowly, there was a dependence the on the biological MRI, and without that, nothing can be done. It was something like that. Even the genome now, time has not come, but still, the genome-based analysis has already come. Genome-based diagnostics have come, and today we are thinking health for all. Every world Now it is a worldwide concern to have total health. And this COVID has even given much more than what we expected like this. So there has been the change in traditions. Now here comes that because of this, the interdisciplinary things started coming in. See, when we talk of the industry means engineering and management. And everything is linked to whether you call it as a medicine or pharmacy. You cannot do anything in gene therapy unless you are thorough in the computer. Or if we talk of the stem cell therapy, again, without engineering, you cannot do that. It is possible that in future we are likely to have genome-based health ATMs. Not the bank ATMs, but health ATMs. So like that, things have been changing. But ultimately, all these things have to be managed. See, administrative process or data managing, that is ultimately is going to tell about the artificial intelligence. So there have been many breakthroughs in this era. We can say, I mean, I'm not talking, I'm going right from the very first era to fourth industrial era. So many breakthroughs came in. Today, if we talk of pharmacy, yes, nanotechnology, precision medicine. We never thought that precision medicine will be a reality. I still remember when the pharmacokinetics I used to teach to the students of B Farm. And then in 1983, I distinctly remember we had one symposium with the Cadila Labs. Indravadan Modi ji was present at that time. And we were talking in the FICCI, actually, I still remember Ashram Road of Ahmedabad, that when we were talking that and there was a question that how can you be precisely doing this pharmacokinetics and accordingly treating, keeping that in mind. And then how can you have a precise medicine or precision medicine, but it is a reality now. Nanotechnology, I need not tell the whole, everybody knows. Gene editing, I never thought that CRISPR technology, when I heard in 2014 about the CRISPR technology, that you can inject something, it will automatically go and treat the genes. It will modify the gene by itself and do that. At least in agriculture, it has already been there. And I am sure that in medicine also, it will be a reality. So like that, regenerative medicine, I remember when I was working with cardiologists in around 1998, 99, and they used to take, can we regenerate heart tissues? It is a reality now. Not only that, now organ on chip, it has started coming in. Well, so if it starts, we can go on talking wherever technologies or artificial organs or all that, it will be there. Of course, the credit, if I have to give concisely to one, it will be Human Genome Project of USA. But it was again a myth that can we use this Human Genome Project for the development of drugs? Answer was yes, but at the same time, many challenges. And when we saw that in 2008, the cost of genome started declining like anything. Sorry, this became the beginning of the real precision medicine, I guess. So it has come up like that. And today, RT-PCR, for example, after COVID, nobody was knowing this PCR itself. Now RT-PCR is the talk of town. I mean, even the small person will know what is PCR, RT-PCR. So diagnostics on one side and development of drugs on the other side. So this is what is the reality is slowly coming in. But certainly, Corona created havoc. I don't want to spoil the mind of anybody that how dreaded it was. I know it is, many must have lost their dear and nears also, maybe friends also. But yes, it was a reality. It was a big challenge in that emergency use of medicine. We can simply say, take the good part that, oh, okay, our pharmacy became the best and important role. At this time that everybody realized that how pharmacy, not just medicines, but diagnostics to medical devices to all. It is a reality. But adversities on one side bring opportunities. I can simply say like that. See, what has happened, there is a sense of like I am calling, previously I used to call about 6 months back, or that it is a post-COVID era now. So after 2020, it is the post, or 2021 or 2, it is a post-COVID era. Many things have emerged. Data freely available, scientific journals freely available, and many things have come. And innovation has reached to the peak in the world. I'm not talking India, but in the world, COVID, post-COVID, the main thing is that science of science is such that there is internationally the innovations are going on. Yes, 20 to 22 has been a period of transition now. Like today, we are in a position to do online because of this COVID, I can say like that. Otherwise, people would have refrained to do that. It was not that it is not available. This technology was available, but nobody knew to think. But now it is, without saying, we go in. Biopharma is something which every pharmacy professional must know this. How this biopharma has emerged. So many things now. We are talking biomolecules, biologicals. We used to talk, but now it is going to be a different shape by itself, other than this. I will simply say that because of this COVID, because of the 2 years of this COVID period, we have the taste of technology now, trust in technology now. But this temperament in technology, it is something mind-boggling. I don't know where it will lead to. But certainly we are having a temperament in the technology. So so what I want to say is that after fourth industrial revolution, which was talking about the robotics and artificial intelligence, there is another era awaiting. Now, what is that era? That we have to see now. Certainly, there is a wave of innovation. Wave of innovation utilizing artificial intelligence also, I can say. Wave of innovation utilizing digital technology. And more entrepreneurs are coming up. And that is what is going on. So, this graph was, again, it is a figure of little bit earlier, I mean 2020 only. But certainly healthcare has the most important application. The question comes now that can artificial learning, intelligence, or machine learning do everything? I think everybody will say yes. Today there can be driverless car. Today there can be, like I'm talking, just like the ATM for money, there can be ATM sitting for your diagnosis. You may just have your health card, type on your symptoms, and you will have a prescription in front of you. I don't know, it may happen. So that means you are replacing doctors also at times. You are getting the telesurgery done. Surgeon is sitting somewhere else and you are doing it here. But can they be better than humans? We say, yes, it is better. Yes, it is better. With AI, it is better. But can it be better than humans? And which human we are talking about? Rural people, younger people, older people, or mid-age? If it is a robot, then as compared to what? Can you have anything which is more robust than the human being itself? Okay, forget all this thing, but can artificial intelligence use common sense or understanding? To me, that answer is no, because it will depend purely on what data has been given to them, what program it has been for the machine learning. It cannot use its common sense. So under these circumstances, if there is a misdiagnosis, something happening, oh, that data was not there. That is not the case. Humanization can do that. On the spot decision, and that is what is important in medicine or in pharmacy. See, there may be a pharmacy supervisor. If instead of that somebody else is some mechanical thing is kept, which is based on the artificial intelligence, it makes sense. But a human person will sense some emergency which is coming up. This is only by human being only. So what we want to tell, yes, it is good to use computers and all that, but human approach has to be more important now. And that's what is going to be the fifth industrial revolution, rehumanizing. Humanity has already been there, rehumanizing the interaction. That is what is going to be. So, I mean, ultimately, machine learning and all that is all right, but humanizing that. You may earn more money, but not the humanity. That is what is going to happen. And I could find that Albert Einstein wrote 80 years back that concern for the man, I mean, our technology may exceed human performance, but concern is that it will be human concern is going to be the interest. And after 80 years, again, that Albert Einstein's statement is coming correct. That is what we have to do. So, humanizing technology is more important. Technology, yes, you got the taste of technology, trust in technology, but the temperament of the technology should be how to see that it is humanized. That is what is the requirement. Because technology, there are dangers. It may be totally opposing or some becomes addictive also. Like we have got everything like this, website, laptop, everything is important. Use of mobile, if you see, drastically increased in the China and India only, which has the highest human populations. So this, like Gandhiji used to say, don't speak, don't listen, don't see. Here we are seeing that mobile does everything at a time. When somebody is sitting with mobile, he will neither see nor listen nor speak. This is what is happening. If I talk specifically in pharmacology, everybody, like if pharmacology student comes, he wants simulation experiments on. He is not interested in anything else. If somebody wants in pharmaceutical, oh, I want to do some TOE only and QBD. What? The experiments are important. The experiment of the preparations, formulations. If we talk chemistry, everybody is interested in molecular modeling, not going in the lab and doing some experiment. This is how slowly and slowly, wherever these skills are there, they are getting lost. This is what I want to tell. People have become a laptop rather than laptop workers. That is what is the problem, which may come in the race. So pharmacy should be considered not just one single entity, but also with the nutraceuticals to Ayurveda to all that. It is important. Vaccine, it is again because of pharmacy. I am a pharmacologist. I used to talk about the 4 eras in the beginning: chemotherapeutic, biological, molecular, genomic revolution, genetic revolution. Now it became omics revolution, and now 7th revolution, which we are thinking of. So we have to think beyond that. The whole discovery process has changed. We ourselves use that in the COVID era, and we got the drug. I will not go into detail. Startup, again, one has to understand in a human way rather than just going in the calculations or like that. Certainly, in the COVID era, we have come up worldwide. This is the figures which are there, that so many startups and getting into it. And it has been something mind-boggling, that the way in which it is flourishing. Don't want to go into detail, but what I'm trying to tell you, differentiate strategies versus tactics. That's what is the message I want to give. So education, irrespective of what type of education, has to be a weapon to fight poverty, social stigma, and superstition. I would like to mention that this quote is of 12 years old. Our Prime Minister Modi used to tell. So I always feel that we have to go in a humanized way. The progress of science without human concern will not be possible. So accordingly, in such conferences, when all distinguished speakers are there and all that, we have to see that we should think rather than just follow some path, either it is machinery or like that. So I wish you all the best. I think I might have taken a little longer time. So I stop here, and I hope I have tried to convey the message expected from me. Thank you.
Thank you, Professor Goel, for your motivational words as well as
You are not audible, sir.
Am I audible?
No.
Yes, you are audible to me.
Not audible.
You audible
I
to
cannot
us.
hear the voice. Oh, sorry.
So,
yeah, okay.
Okay. So thank you, Professor Goel, for your
Okay, it is my
motivational
fault.
words and research, this
No.
guidance to us to execute.
I'm unable to hear. Okay.
So I'm audible.
Okay,
to
sorry.
other-
Now it is my fault. I think there
Okay.
was something wrong here. Okay.
So
now,
I will
Sorry.
repeat. I would like to thank you, Professor Goel, for your motivational words as well as the message you conveyed through this presentation. And we hope for your support on this type of platform in future also. So we value your time, and thank you for giving us time and opportunity.
Thank you so much for inviting.
So, colleagues, let me introduce to the next session, which is the scientific session, which will include 3 presentations. Our judges for the session have joined. They are Dr. Palnisambi Selvamani from Anna University, Trichy, Tamil Nadu, Professor Piyush from Tirthankar Mahavir University, Moradabad, Uttar Pradesh, Professor B.N. Suhagia from Dharam Singh Desai University, Nadiad, Gujarat, and Dr. Sunita Dahiya from University of Puerto Rico, Sama, USA. Our first speaker of the session is Dr. Sachin Dubey, who is the Director of Analytical and Formulation Sciences at KBI Biopharma, Switzerland, and who is President of our APP Switzerland International Branch. So without taking not much time, I would request Dr. Dubey to start his session. Thank you very much. Dr. Dubey, floor is yours. You are not audible, Dr. Dubey.
Am I audible now?
Yeah, you are audible.
Okay, great. So thank you, Professor Dahiya. Let me quickly share my screen. You need to allow in order for me to share the screen, please.
Yes, sir. I made you the co-host. You can please share your presentation, sir.
Unfortunately, not yet. It says host disabled, participant screen share. Okay, now it's okay. Do you see my screen?
Yes, Dr. Dubey.
Great. Okay. Good. Well, thanks a lot, Professor Dahiya. Thanks also to Professor Goel, with whom we reverberate the same message. Essentially, we are moving towards new medicines. I think we have came a long way. And I will put up industrial perspective where we stand when it comes to one class of molecule, which is very important one, monoclonal antibodies. I'm not 100% sure, but this is one of the area which is currently not completely covered in our pharmacy syllabus. Nonetheless, I think this is one of the area which is highly in demand from biopharmaceutical organizations as well. So I will go through step by step, going from what are the different requirements, starting from discovery and going to the clinical setting. Okay. I am not able to move for some reason. Okay. So we will look what biopharmaceuticals are, mainly focusing on monoclonal antibodies. Then, as I mentioned, we'll start with discovery. Once again, the focus here today will be monoclonal antibodies. There are, with a limited time, I think the best is to really focus on one class, which is one of the very, very important one. And then I think once again, Professor Goel mentioned about QPD approach. I think it's very important from industrial applications, certainly. And I will try to walk you through on from what are the regulatory requirements, what are the different steps in terms of from QPD approach development of a product. And once again, this will be with an example of monoclonal antibody. And lastly, we'll touch base a little bit about future outlook. So starting with biopharmaceuticals, I think we all know about small molecules like aspirin or paracetamol. These are of course very, very traditional. I'm not going one step beyond. Professor Goel mentioned about herbal medicine. Certainly, they are there. But when we study modern medicine, aspirin, paracetamols, lidocaines, etc., all those small molecules are categorized here. Then we start moving into peptides. For instance, insulin, glucagon-like peptides, GLPs, etc. Those are then coming into the peptide category. They are much bigger than a small molecule. And then finally, we start moving towards proteins or monoclonal antibodies as well. They are large structures. They have various different level of structure as well. But for the moment, I would just suggest keep in mind that typically they are almost 1,000-fold bigger in their structural size as compared to a small molecule. So, in other words, what I mean to convey as a message here is whatever challenge we are facing here, that we have to stabilize one of those bonds, we have to make sure that they are, proper racemic configuration, etc. Many of those we have to make sure in these big structures as well, but at a 100,000-fold more. Yeah, structurally, what they are. So proteins or monoclonal antibodies are structured into 4 different categories, starting with primary structure, which is nothing but amino acid. Starting with a peptide bond from carboxylic acid, and one amino acid get attached to the second, etc. That forms the sequence in one word, or the primary structure in other. When we get this primary structure to a sufficient length, they try to form a well-defined structure depending on the amino acid sequence. The 2 of the most common known secondary structures are alpha helix or the beta sheet. As the name suggests, these are either in the helical form or the sheet form. The determination of which of these will be formed will be dependent on which amino acid is present, typically on those loop forming regions of alpha helix and the beta sheet. Once again, once the secondary structure evolves into a significant size, or if there is a combination of multiple alpha helix and beta sheet, they start forming what is called the tertiary structure. So this is nothing but the increased number of amino acid getting sequence and peptide bond formed, eventually leading to a tertiary structure generation. Then last part is quaternary structure. Here, when multiple of the tertiary structure, so multiple of those sequence, when they start coming together, that is called a quaternary structure. And for a biopharmaceutical to be active, and I'm not talking about the stability of that one amino acid, that is of course important, but also the structural aspect that a quaternary structure, this green has to be here and it cannot go on the backside of this molecule, is equally important. Now, as I promised, monoclonal antibody is the focus of this presentation. I would like to point out few structural features of monoclonal antibody. So on a high level, monoclonal antibody is composed of two main parts. The very first part is the heavy chain, the two blue in this cartoon here. These are heavy because of their molecular weight, typically double than the light chain. The light chain are depicted here. Then the most important part of monoclonal antibody, or IgGs for that sake, is the antigen binding region. So antigen binding region is the top part of the Y-shaped structure, which actually interacts with the antigen. Antigens are the targets for these antibodies. A few other things. One important part is the effector function. These are not really necessary for target binding. However, from the functionality point of view, this is very important for certain therapies, like for instance, for oncology, where monoclonal antibody finds a lot of application, they become very important. Then you will see a lot of sulfhydryl bond, disulfide bond, and these are very important to keep the structure intact. Now, why we have chosen monoclonal antibody? Why they are so important? The reason is sheerly by numbers, right, by facts. So here there are two graphs, and we can focus on one of those. Any one of those really. This is the number of overall biotech product which got approved from in last 4 decades or so. And on the right-hand side, you see the maps versus non-maps. The maps being the blue one, and non-maps being the orange one. If you see, especially towards the right side of the graph, you will start seeing more and more blue there, meaning that more and more monoclonal antibodies and monoclonal antibody-based products are coming to the market and making patient at comfort from their diseases. Now, getting into the next part, discovery. The very first part of discovery, like for a small molecule or any other indication, the first thing is to identify the target, right? And here I have taken a summary of some of the very successful products where several biosimilars are also under development. There are some key targets: TNF-alpha, tumor necrosis factor, HER2, VEGF, etc., etc. Those are the targets which are doing something in your body. And I will take an example of VEGF in a short while. And with the antibody, in this case, either that pathway, that cascade is blocked, and then the therapeutic indication is, therapeutic target is achieved. They are either used in this particular case for monoclonal antibody. They find major application in autoimmune, so immune modulation, or in oncology. There are a few others as well, but these are the two of the most common indication for monoclonal antibody. As I mentioned, here is an example. VEGF is a pathway, VEGF based pathway, which is where we have onto the cell, there are receptors, and then there are VEGF. When the VEGF reaches or attached to its receptor, there is a full cascade of signalization, and a few proteins are produced. These pathway, in summary, leads to neovascularization, meaning new blood vessels are formed when this interaction, when this pathway is activated, right? And what will happen in a normal human being when a new blood pathway is formed? This is for sure helpful for this related to growth. On the other side, if this is happening at an uncontrolled level, this is where the condition starts getting into oncology or cancerous situation, overgrowth. The new blood vessels are being formed, and new blood vessels are formed serving food or supplying food for the oncolytic cells. So here's another depiction. You see in this particular cartoon that you have an oncological area, oncological cell where new blood vessels are being formed. They are through this VEGF, and the antibody against it, which was developed, is called Avastin. So that's the brand name. Bevacizumab as a antibody name. What does Avastin do? So this blue antibody, it blocks the interaction such that VEGF or new blood vessels are not formed, such that this blood vessel starts getting slower, leading ultimately to the shrinkage of the cancer. So that's one example of how a target is identified. VEGF in this particular case is a target. There are several targets which are being where the monoclonal antibody exists already, and there are many hundreds more where monoclonal antibodies are being developed either in discovery phase or in clinical settings. So once we identify the target, what do we do next? The next thing is to generate the actual antibody against that target, right? And here I'm depicting it in, let's say, 20-year back model, but this is the most simplified one. So you take that antigen. In the previous case, for instance, that was VEGF. That is injected into a mouse. What does mouse body do? Mouse consider that VEGF to be foreign particle, and mouse generate antibodies against it. So those antibodies are the antibody which will ultimately kill or destroy that VEGF in the mouse body. So now you take out those plasma cells, you take tumor cells. So these are tumor cells for production, not tumor cell from patient. So these are tumor cells from production. Tumor cell have a very important property. The property is that they grow uncontrolled. They really duplicate and grow uncontrolled. And that property is fused with the plasma cell, leading it to what is called hybridoma, right? So these hybridoma cells are the fusion of what antibody the plasma cell can produce and with an uncontrolled growth from the tumor cells. These hybridoma cells then ultimately start producing the antibody of your interest. Okay. So that's how we get to the monoclonal antibody as a first step. However, as I mentioned, this was what happening around 20-25 years back. We were getting murine mouse antibodies, but the challenge is that they were highly immunogenic. Why they are highly immunogenic? Because they ultimately come from an organism which is not which is outside our body system, right? So mouse antibodies are not the same as human. Thus, human consider those as foreign, right? So there were challenges of having immunogenicity, which leads to the further development in a stepwise manner. The first part was to develop chimeric antibodies, which means that the first part, or the antigen binding part, was taken from the murine from the mouse, and it was grafted onto the human part of the constant region. In the next step, humanization of antibodies were done, meaning that only few amino acids, which are really responsible to the target or antigen binding, they were taken and they were adapted into monoclonal antibodies. And last is human. Of course, in this particular case, this is a control situation when by default a human get a disease, his or her plasma is taken, and then the antibody sequence is generated out of it to be considered a full human. The advantage as we move from left to right is that the immunogenicity, as you see more blue color appearing, the immunogenicity goes down. Now, moving to the next part, now we know our target. Now we have developed also antibody against it. The next step is to develop the product. And the very first thing is QTPP. QTPP is the first principle of QBD, quality by design. QTPP stands for Quality Target Product Profile. Typically, we also have TPP, Target Product Profile, which is a little bit more from business perspective. I will just show you in a short while as well. But from quality perspective, from pharmaceutical and manufacturing development perspective, QTPP is always with a Q, which is quality, of course. So here we define what the indication is, what the formulation is, what the osmolality is, what mechanism is. So all what is required, all what you would like to have in your molecule, this is what is captured in the document called QTPP. Then this QTPP leads to quality attributes. So, okay, the QTPP can be achieved by a molecule if it has so and so structure, so and so function, so and so composition, etc. So that's what comes into the quality attribute. How do you generate quality attribute? Then you get into the studies, the characterization, the release aspects of it, and finally leading to the definition of CQA, which is called critical quality attribute. Some of these we can call it, we can remove it to say from risk assessment, meaning that, for instance, osmolality, if we have sufficient information from literature saying that, okay, if we use sucrose at this concentration with histidine, for instance, it will give you a osmolality of that can be calculated. So that's a risk assessment you can perform, and then you don't have to monitor it as a critical quality attribute. But otherwise, critical quality attributes are all the attributes which are required to be followed, ultimately responsible to meeting your quality attribute targets. Yeah, as I mentioned, the precursor to QTPP is TPP, and TPP can have certain business targets as well. By business target, I mean that what type of presentation you may want, right? So for instance, I'm talking once again for biopharmaceuticals here, which is largely administered parenterally. You can have liquid presentation, or you can have a lyophilized presentation, or you can have presentation in vial, or a presentation in a prefilled syringe. All those are the requirements of or composition of TPP, which is fed in from strategic perspective or business perspective. The QTPP is largely driven by what quality we require. Once we fix that we are going with a prefilled syringe, then the QTPP will be driven. Okay, so what gliding force I should have, how much force I require to administer that molecule, those will be part of QTPP. And then CQA will be the parameters on how to test those and how to ensure that those are reached, not just from the performance perspective, but also from, or more importantly, from safety and efficacy perspective. Here is an example of a QTPP. So you may have, route of administration, what dose range we are looking at, how frequently we are thinking to administer it, what should be the pH range, whether we need tonicity to be controlled. If the administration is not direct, so for instance, if you consider a product which is supposed to be diluted and then administered, then you don't need tonicity to be controlled, because of course once you dilute it, your tonicity may come from saline or any other diluent media, etc., etc. So these are all what is required, and then you define certain targets, right? Uh, and this is happening much before you start to develop. These are the targets you want, right? So this is what we want. But ideally, because we are administering our product by IV, we cannot go below 3.5 pH and above pH 9.5, because that will disturb the physiological system. Similarly, isotonicity limits. It's a must to have in that context. So that's what we define, that's what we write, and there are several versions. But this is like the first version, which we start with before actually going into the lab. So now going into the lab part, which is essentially the development. The very first part of development for biopharmaceutical, including monoclonal antibody, is to establish a cell line, a reliable cell line which can produce it. Unlike small molecule is produced in bioreactor, where you bring in a couple of or a few of chemical reagent, enzymes, etc., and you get to the final product. And because the system is so controlled, you can control the quality of your raw material and you're good to go. But in case of biopharmaceutical, which are produced by cells, cells are the most important one, and cells inherently are very variable. So the very first target we have in development is to establish a robust cell line. How does this happen? There are certain cells which are known to be robust. They are very well structured. They are very well characterized by the biopharmaceutical industry. For instance, CHO, Chinese hamster ovary cell lines, is one of those with their defined medium. So what do? We transfect with the sequence of your antibody, put it into the CHO cell, start screening. Why the screening is important? Because ultimately, from commercial perspective, you want the cells which produce the highest, right? And when you take 100 cells, when you transfect 100 cells, not all the 100 cell will produce antibody with the same package. Some will produce 5 gram per liter, some will produce 10 gram per liter, but some will produce maybe 1. And you don't want the 1 gram per liter. You will select something which is at a higher level and with a higher productivity, right? As we move further towards the right side, another important thing is to assure monoclonality. What does that mean? This means that your production is actually coming from one cell or one type of cell, right? As I mentioned, if we transfer 100 cells, you have selected the high producer, but that high producer is a single cell, and then single cell, of course, can be multiplied into. But attribute-wise, the cell has to be monoclonal. Otherwise, what will happen is that during one fermentation, we will produce 10 gram per liter, while in another one we'll produce 1 or 2, which is going to be not supported from regulatory perspective. So once the cell line or cells have been established, then we have to do the process development. There are 2 parts of process development in biopharmaceutical. First is USP, which is upstream processing, and the second is DSP, which is essentially downstream processing or purification. What do we do in the upstream? We take those cells which we have characterized, which we have defined, they are monoclonal. We take those cells, we put it in a smaller vessels, we grow them. We grow them at a very small scale, at the shake flask level, very small volume, get it into expansion, stage 1, 2, 3, 4, can be, depends on your production bioreactor, can be multiple steps, leading to the final production bioreactor and grow the cells further to, a couple of weeks or so. So in theory, upstream processing, if I were to define in one word, you are taking a few cells and growing it to several billions of cells, which ultimately lead to production of your antibody. Now, once the cells are grown, maybe just one more thing before we go to the DSP. The complex part of growing cells is that cells are very sensitive. Cells are sensitive to several things, right? They're composed of sensitive, right? They're sensitive to pH, dissolved oxygen, temperature condition, and all of those have to be monitored, shear stress, sparsonic agitation, etc., etc. And all of those conditions have to be monitored within the well-defined boundaries in order for our CQAs, critical quality attributes, to be met time and time again. Now, we have produced the cells. The next step is to purify, right? The cell produced hundreds or thousands of protein. We are not interested in those. We are interested in the antibody which it has produced, right? We have to remove all the other proteins. We have to remove all the DNA composition, all the lipid, all the other things which are not required for us have to be removed. And that exactly is the process called downstream processing. Okay. What is happening here is the very first step is affinity chromatography, and for monoclonal antibody, it is typically protein A. Protein A is a target, is a protein which interact with antibody very, very specific. Okay. So that's where we get all the other protein removed to a very high efficiency. Then comes acid treatment. Why is it important? Because the cells which we use to produce may have contamination from virus, or the raw material we might have used may have contamination, right? So that is where what we do is we bring the pH down at a condition where virus cannot survive, but our monoclonal antibody can. So that is the acid treatment. Then comes AEX or CEX steps. I will just explain in a short while. Finally, viral filtering, once again, based on the size of the virus, we make sure that all the viruses are removed. The removal of virus, once again, is a regulatory obligation. We need to have at least 2 orthogonal methods to remove or inactivate virus completely during the purification process. Plus, there's a calculation of log fold removal, which is required to be demonstrated as well. How does the process development work? How does the protein A work? So protein A, consider those round beads as protein A, and those multicolor things as your supernatant after USP process. So it has protein of interest, but also other protein. You make everything flow through. What will bind is the protein of interest, because that has affinity with your protein. All the other things, all the other proteins, DNAs, etc., they will wash through. Then you change the condition to elute the protein of interest. In this particular case, either you change the pH or you change the concentration. So that's how you purify the first affinity chromatography. Here is the second and very important process as well. Once again, this is cation exchange, and as the name suggests, all the proteins are charged, and depending on their pH condition, you make your protein of interest interact with the stationary phase, ultimately leading to the elution of protein and the impurities in different. The last step of process is fill and finish. So once you have purified protein, you formulate it, you essentially add all the stabilizers, you mix it very well. You make sure you have a sterile filter. Why is it important? All of those are parental. So sterilization is important. We can't do it by autoclaving or dry heat. We have to do it by sterile filtration. Proteins are temperature sensitive, so that's out of possibility. Once you sterile filter it, you start filling it into the vial, and your drug product is pretty much ready. Now, last, going into the critical quality attributes and why they are important. Critical quality attributes essentially are the attributes which are important to ensure that your product comes out consistently with the right quality and meets your quality target, right? And these are composed of these are influenced by critical process parameters, so CPP is critical process parameter, and critical material attributes, right? If you start with a material which is not of right standard, it will be impossible to control it in terms of quality. The same goes with the process parameter. It is important to have, for instance, when we are growing the cells, it is important that the pH is maintained within a certain range. And that is, for instance, one of the critical process parameters. If we go beyond that pH range, we don't know how the cells are going to behave. They may start producing proteins which are of different quality attributes. So all of this together leads to the critical quality attribute as an outcome. Now, once again, from an antibody perspective, it is important to monitor critical quality attribute, not just at the time of release, which is important, but also during stability to ensure that it is of suitability during all its shelf life. Some of the standard critical quality attribute for biopharmaceuticals are here. Purity is very, very important. So purity is typically done by 2 or 3 techniques orthogonally. Aggregation, purity, or that it's structurally the same. Potency is obvious. We want to administer anything which is potent, which is functional. So of course it's a given. Identity is important as well. At least 1, or preferably 2 assays are required where the identity is confirmed that the product is really what we intend to have. And then there are some general ones. So general ones are typically, appearance, pH, osmolality, etc., etc. So these are general properties. And last but not least is the safety, or safety are sterility if it's a sterile product, bioburden, endotoxin, etc. We don't want to administer that, subparticle, and so on and so forth. So those are the ones which are required to be monitored from safety. Many of those are also monitored during stability as listed as well. Now, moving towards the last section, which is manufacturing and quality control, on a couple of few slides. So manufacturing in theory is expansion of what we have just learned during development. I here only want to introduce a notion of single use versus stainless steel. In biopharmaceutical, single use is a very, very important concept. And it's a very, very important one from the perspective that contamination or cross-contamination is an important risk which has to be controlled. Stainless steel is is a vessel where we produce it, we clean it, we sterile it, we produce it again. So there is certain risk if your cleaning is not proper, you can have contamination, right? So everything is established here, but it has certain risks. In single use, what you do is you of course have stainless steel as the body, as the structure, but what you do is you put a bag like a polythene bag in a very simplified version. You put a bag, of course, strong bag, which is then used only a single time. And this bag is produced and supplied in a sterile form. You use it, you produce it, you do your one batch, discard it, and you don't worry about sterility or contamination from that perspective. There are several advantages and disadvantages for both of these systems from manufacturing perspective. Single use is high on consumable. You need to have these bags again and again, so it's important. While for stainless steel, you need a higher investment to put up, plus a higher investment in terms of cleaning and sterilization, right? Scale wise, you cannot have a very, very big bag. Nonetheless, I think boundaries are being pushed. A few years back, 2000 liter was the limit. Now we are getting into 5000 limit. And in the coming days, I'm sure this will go beyond as well. Stainless steel, it's a sheer, it's design, right? So you can have 20,000 liter, you can have even bigger, right? So there are many of those where we have comparison in that context. Now, we'll touch base on this. Nonetheless, here is pretty much a complete list of quality control, which essentially come from either the release testing or testing of during stability, and or both, I would say. Safety, this is for quality control for a drug substance, bioburden, and endotoxin. For drug substance is not sterile. Drug product is, so that's why sterility is not here. We already discussed about general. A few words about purity. The techniques, standard techniques used are size exclusion chromatography, CESTS page. These are like workhorse. Charge profiling is typically the identity testing method. So if I had to summarize 4 techniques to focus on from studying perspective, the 4 of those will be size exclusion, capillary electrophoresis, ELISAs, and then charge profiling using CIEF. These are pretty much the workhorse for majority of platforms. Now, a couple of slides on future outlook. We talked a lot about monoclonal antibody. The first monoclonal antibody was administered almost 30 years back. The field has advanced, they have improved. There are new modalities which are coming. One of the very important one is ADCs. These are antibody drug conjugates. What do you do here? You take your antibody, which can have target antigen or can be simple IgG, but it has a payload. And this payload is essentially a molecule which either is insoluble or it has one challenge from a pharmaceutical perspective. It may have instability, etc. So what do? You conjugate, you link your payload, cytotoxic payload in this case, and administer this as ADC. Since antibodies are specific, they go to the specific cells, interact with them, and then the payload or the cytotoxic molecule here does its job with that particular cells. Similar is the strategy with bispecific as well. So bispecific are the antibody which can bind to 2 different type of targets, right? So just imagine here, you have a tumor cell, the yellow one, and then you have T cells. So what does T cell do? T cell are the cells which kill the unwanted cells in our body. So that's their normal function. And if we have a bispecific, one arm binding to a T cell, one arm binding to a tumor cell, thereby bringing the two in close proximity, and T cell getting a more proximal opportunity to kill the tumor cells, right? mRNA vaccine, I think we all now know very, very well after COVID. mRNA vaccine has really picked up. In principle, what does it do? mRNA is into the cell. The cells take mRNA, process it, it produces the protein, and that protein is essentially the viral protein, and the viral protein, the antibody produced against it as an immune response, then in future protect us against a future infections. Cell therapy is yet another very interesting part. What do we do here? So instead of going with an antibody, you take out the cells, the same T cells. Essentially, T cells are, in a healthy human being, T cells are responsible to maintain a good dynamic. They should remove the cancerous cells. But if the cancer cells are overwhelming, then what do? You take out the T cells, you engineer those such that they express something, a target, which attracts T cells, right? So which binds to the cancer cells, right? So here, then you grow those cells, and then you inject the modified T cells now, which have their target. So they know which cancer cells to attack. They already know. Earlier, they need to develop their memory, but here they already know, because we have engineered them that, listen, this is the target, you have to reach that target in the cancer cell. And so that's what you do in this particular cell therapy, which is called CAR-T therapy. And with this, I would like to thank you. And if there are questions, more than welcome.
Thank you very much, Dr. Sachin Dubey, for such a nice presentation. A very, very interesting topic on the monoclonal antibodies, and it is still more interesting because you joined it with the QBD and its components. So congratulations for presenting such a nice topic in a simplistic manner so that everybody can understand. I have a question about the two things, that is QTPP and risk assessment. Do you use any software for the QTPP and risk assessment, or just based on the prior knowledge we can use it?
So yes, there are multiple software which are available to do the QTPP. However, this is not a must to have a software, because essentially QTPP is a list of parameters which we can, in a simplistic manner, can generate it on an Excel table, for instance. The drawback of using an Excel table is that you will have to link with a different parameter. The whole process of manufacturing and development takes like 8-10 years. And in order to have them well controlled, it is best to have a software which can control, which can version it automatically. But it's not a requirement.
Okay. And in the beginning of the QBD, you implement the QTPP is the first thing that must be done before starting with the QBD, right? Yes. So I just want to know if there are a lot of CQAs, and you mentioned that we can reduce the CQAs by doing the risk assessment, right? But even though I have observed that there are several, if you look at both the formulation parameters and the process parameter, there are a lot of variables, and they really affect the product quality. In that sense, if you determine, for example, 5 to 7 CPPs to achieve 5 CQS, which are really important, so the number of experiments to be run is very high.
Absolutely. And that's
Is
why
there any way to reduce that?
absolutely. Yes. And that's where the risk assessment comes in, right? I mean, as you mentioned, if there are 7 parameters, out of those 7 parameters, you may have some of those which are much more pronounced effect to have on your molecule, on your output, while some other may have a much higher occurrence, right? And some on the other side may not have such a big impact or may not have that high occurrence, right? So if an occurrence is happening, once in 1,000 time, or if the impact is gonna be very, very low, based on the risk assessment, RPN, risk priority number, we can always put that low, and you can discount it. You can remove that parameter from your studies, and regulatory agencies are completely open to that. I think it's a pragmatic approach. So such that out of those 7 parameters or so, you focus on 3 or 4, but you characterize them in a very nice manner, instead of having all of those 7 considered, but then just doing limited exercise for that sake. So that's how we use risk assessment.
Yes, right. Because yeah, that's correct. Because otherwise there are usually like more than 50 batches usually, and then if you use the DOE, then it usually creates like around more than 50 batches that you must do to check. So I think preliminary trials can help us to identify what are the pronounced factors or what are the
So risk assessment,
light factors.
yes, risk assessment, a lot of things goes in. What I just explained is theoretical risk assessment. What you mentioned, of course, is pre-screening. That does help. There is a third parameter, which is prior knowledge on similar formats. There are more than 50 monoclonal antibodies which are in market, many hundreds more in clinical trial. So there is wealth of information which is available. We now know that oxidation of one amino acid at amino acid sequence 296, for instance, is a critical one. But all the other tryptophans or methionine are not important from that perspective. So we can discount those things based on that prior knowledge. And last is the platform knowledge as well. Because if you have a platform, if you have done certain studies in your past, you can take into consideration that part in your risk assessment as well. That your platform use this pH condition, you have produced 3 molecules with the same thing. So the 4th one, we need not to do that study. So all of those things combinedly go into the risk assessment to reduce the whole experimental set.
Thank you so much. Thank you so much for answering all these questions. I ask for other chairpersons for their comments.
Yes, Madam. Dr. Sachin, it was a very nice presentation.
Thank you. Nice to meet you,
Excellent
Professor
narration. I
Silverman.
have a question to you. Whether CAR T cells can be made universal?
CAR T cells are getting to, they are not designed to be universal, right? There are 2 types of cells. There are 2 types of CAR T possibility. One is where we can make it, target specific, but they are, once again, I will not call them universal. I will call them that they can be used for multiple patients. But still, what will be the first step? The first step will be to really identify that pool of patients have that target expressed into their cancers, right? So that's a pre-screening which will be required. That's one type of CAR T. The second type of CAR T is really personalized, where you take out the T cells from that particular patient, you modify for that particular patient and put it back into the system.
Thank you. Thank you so much.
Okay, thank you, Sachin Dubey sir. Give very nice talk on biopharmaceuticals. It is the developing branch and it is the demand of the day. So thank you for your very precise and simple language to explain all the things. Even though it is risky, but still it is a developing branch, and we will hope it will come in future. Thank you.
Thank you. Thank you, sir.
Thank you, Dr. Sachin. I am Dr. Piyush. You have given a very comprehensive lecture, I can say, from the inception till the future of the biomolecules. I have a question regarding, like, when we are having a vigilance for anti-cancer drugs, or similarly we are treating the patient with these monoclonal or polyclonal antibodies. Do we, in future, maybe we have a pool where we can have some software which can see the interaction of all these things. So when you screen it out, do you know when you are launching the drug in a market, so this drug may interact with this kind of other drugs or the other kind of formulation, because the patient, sometimes when they take these drugs, they are on the other kind of medications.
Yeah. So that's a very, very interesting question, Dr. Piyush. I think this is one area. I'm not saying from pharmacovigilance perspective. Yes, pharmacovigilance, I think we should follow, or there is a follow-up in the same way as for a small molecule. That's for sure the case. However, I think at just the beginning of pharmacovigilance part, the PK/PD part is one area where, for proteins in general, not just monoclonal antibody, but for biopharmaceutical, this is in its inception phase. I think there is a lot of opportunities there, which can be done, which can be explored. I mean, right now we don't know what will make the distribution of antibody more focused into one organ over other.
Yeah.
Or what will make it to permeate through blood-brain barrier, for example. It's a large molecule. And things like this. So the PK/PD principles are, let's say, less explored when it comes to biopharmaceuticals. A lot of biopharmaceutical developers traditionally, things are changing, but traditionally have been coming from life science or biotech background, not from pharma background, meaning that PK/PD is often not paid sufficient attention. So an area of future. I think there are a couple of very good professors. They are now looking into it very, very systematically. But a lot of things are still to be done there.
Thanks. Thank you.
Thank you, Dr. Sachin. So, thank you for the wonderful and informative, knowledgeable lecture, and the output, the things you have shared. Obviously, it will be useful for the research project of students, and we will share the feedback on that with you. So just I would like to thank you for your time, and I would like to inform you that the memorial award what is sponsored, we are highly thankful for you, and it will be announced as the APP Outstanding Orison Award in the memory, as memorial award in the valedictory function. If you are able to attend, it will be at 3:35 p.m. in India time. Otherwise, I will share the certificate of the winner with you.
Sure.
Okay.
Thank you.
Thank you, sir.
Thank you. Thank you.
So, as per schedule, our next speaker is Dr. Vijay Kumar Shankar. who is the DMPK research scientist at C4 Therapeutic Institute, MA, USA. And without taking not much time, we are skipping the introduction of speakers because our session is running a little late. So sorry about that. So I welcome Dr. Vijay Shankar to start his talk.
Thank you for giving me this opportunity to give a talk. Can you just enable me to share the screen? I guess, or should
Yes, sir.
enable
One
me?
second. 1 second.
Thank you.
Yes, sir. You're the co-host now.
Okay. Can you see the screen?
Yes, we can.
Yes, sir. We can see this.
Okay. Just getting into before getting into the presentation, I would like to thank us, Dr. Sachin, for giving a wonderful presentation. Even I am new to the large molecules. It was great comprehensive presentation. Today, I will be giving a talk on drug discovery, academia, industry collaborations, how it works. And just getting into the introduction. In drug discovery, we know that different stages are there, like target identification, where what target needs to be targeted. That is the important steps, like the receptors or some cell lines, which cells which needs to be targeted. And the second steps, there is a huge opportunity to for collaboration at the target identification stage as well. Something like the example we can give for target identification is by University of Pennsylvania, who identified the RNA vaccine, which was useful for the Pfizer and Moderna to use it as a basic research, which helped them in developing a COVID vaccine. And next, coming to the it identification, it's something like identifying a molecule which can be targeted to the cells or receptors. It is something like usually the lot of universities are working on those, like their molecules, the university where the chemistry synthesizing the molecules have been used by various industries. Example is even my industry where I am working, C4 Therapeutics. It is using the target which has been means molecule which has been synthesized by Boston University. And coming to it to lead is something like optimizing the molecule to get a better property, drug-like properties. And from there, it's a lead optimization stage to get optimum levels into the required tissues and everything, optimizing the chemistry, and simultaneously evaluating its PD effects and pharmacokinetic effects. And further, it is IND enabling studies and clinical trials, which is regulatory requirements. To give an example, so it's something like there are various industries which have been getting into this collaborations. Like I can say that university spin-off, the example is Genentech, which came out of University of California. Now Genentech is a group, Roche group, where over 17,000 people have been working. And other example is Crucial, which develops vaccines. And this is taken over by Johnson & Johnson. And giving another example is like Locus Biosciences, which works on CRISPR, as Dr. Ramesh Professor Ramesh was talking about this CRISPR technology for treatment of disease. And Locus Bioscience has been a university spin-off, and they are developing a CRISPR technology. And again, Professor Ramesh has spoke about the human organ and chips. Even this is a university innovation which has been used by industries, and there are also various companies which are working on the human organs on chips, which have been right now, which has been accepted by FDA as well, which is getting into the IND enabling stages. And these are the top university spin-offs, and each company is something like worth of more than $500 million, which has been opened in 2018 to 19. Just giving an example how then university and industry collaboration can be made out to welfare of humans. Something like the main thing is something the focus from the university standpoint should be to be towards the society and how to reach the market. Hopefully, in future we can see many of this when universities are open to target the market and get more industrial collaborations. With this, I'll be going into the couple of topics where I work with industrial collaboration when I was working with the University of Mississippi. And it is effect of sulfobutyl ether beta cyclodextrin on oral and dermal pharmacokinetics of drugs. This was been the studies were been funded by Ligand Pharmaceuticals, and Ligand Pharmaceuticals is something that they are the inventors of Captisol, a sulfobutyl ether beta cyclodextrin, and this project was funded to study the effect of sulfobutyl ether beta cyclodextrin on oral and dermal pharmacokinetics of drugs. And just getting into the introduction, the most often and probable, whatever the popular expectations from any drug discovery scientists target the receptors or any of these cells. For that, the molecule should have an sufficient lipophilicity which increases the drug target binding and also increases the tissue distribution. And so in the market, we can, whatever the drug synthesized by a chemist in a drug discovery setups, or over 90% of drugs are lipophilic in discovery pipeline, and over 40% lipophilic drugs are approved. So because of lipophilicity of the drug, there is complex drug discovery and development process. And each stage of the drug discovery, the molecule should be solubilized in aqueous media, and it becomes very difficult for carrying out different discovery experiments. So, and also further getting into the animals, when the discovered molecule is getting into the animals for being tested, there is variable absorption and bioavailability of variation in the bioavailability of the drugs. And the main approach what they use is drug solubility enhancement approaches, and there are different approaches like co-solvent approach and converting a drug molecule into a salt and using surfactants to solubilize the molecule, and cyclodextrins complexation, and preparing the amorphous solid dispersions and changing the polymorph of drug, and using a lipid-based systems, and co-crystals, preparing a co-crystal. As my projects were mainly focused on cyclodextrin, I'll be getting more into the cyclodextrin. There are three major cyclodextrins which increases the solubility of lipophilic drugs. Those are alpha cyclodextrin, beta, and gamma cyclodextrins. And based on the number of glucopyranose units, it's been classified as alpha, beta, and gamma. What does cyclodextrin do? Cyclodextrin as an external hydrophilic group, and as a cavity of hydrophobic cavity, which can accommodate the lipophilic drug. By this, it increases the solubility, and also there are reports that it stabilizes the drug from external media, and it masks the taste, and also it decreases the irritations. And what are the major cyclodextrin which are used in pharmaceutically or beta cyclodextrin? And there are various beta cyclodextrins, like the generic beta cyclodextrin, And other derivatives are like hydroxypropyl beta cyclodextrin, sulfur beta cyclodextrin, and methyl beta cyclodextrin. And they vary with the molecular weight, and also the solubility in the water varies. And we can see that the generic for beta cyclodextrin has only 18.5 mg per ml solubility. With hydroxypropyl, it is 600, and sulfur beta ethyl cyclodextrin is 500, and methyl 500. And with FDA, these three cyclodextrins have been approved, except methyl has not been approved yet because of its renal toxicity issues. Beta cyclodextrins are been like oral and topical products are in the market, and hydroxypropyl beta cyclodextrin oral and IV is in the market. And for sulfur beta ether cyclodextrin, only the parenteral route has been available, but not the oral route. So the objective was to see the utility of the sulfur beta cyclodextrin in dermal and oral pharmacokinetics. And what does cyclodextrin do? Whether it increases the drug absorption? Something like the cyclodextrin carries the lipophilic drug to the cavity, and it increases the solubility of the drug at the biological barrier, and which assists and facilitates for drug absorption across the biological barriers. Getting on to the first project, what I will be talking today. It is optimization of sulfur beta ether cyclodextrin levels in oral formulation to enhance the progesterone bioavailability. And progesterone, as we know, that it regulates the menstrual cycle and pregnancy and embryogenesis. And it is prescribed for luteal phase deficiency and estrogen dominance treatment. And it is a BCS class 2 drug, steroidal drug with having a variable oral bioavailability. Marketed oral progesterone products are available, which is something like available as an micronized and suspended in peanut oil base. And because of this, there is a varied oral absorption. And there are cases where it has been under-absorbed in some cases, and it is over-absorbed and leading to the side effects and also subtherapeutic effects have been seen with the existing marketed oral progesterone products. And the objective was to enhance the solubility of progesterone using the cyclodextrin and evaluate the effect of cyclodextrin on oral pharmacokinetics of progesterone in ST rats. Here, this is the phase solubility curve of progesterone. We can see here, with increase in the concentration of cyclodextrin, you can see the solubility of progesterone was increased. And the fold was something like, we can see around 2000 fold increase in the solubility of progesterone in presence of cyclodextrin. With this, we just wanted to characterize the sulfur beta, this complex. In collaboration with the molecular modeling group in the university, we just wanted to see at what orientation the progesterone forms and complex with the cyclodextrin. And with this, we got to know that the progesterone can form a complex. Either the A ring can get into the hydrophobic cavity of the cyclodextrin, or a D ring can get into the cyclodextrin cavity. And this was been confirmed further by like FTR and proton and carbon NMR were conducted to confirm the orientation of the progesterone forming a complex with cyclodextrin. And we performed a pharmacokinetic studies of progesterone in rats. And here in this case, we just wanted to see whether the we are thinking of just filling the complex in the capsule and administrating to the rats to check the pharmacokinetics. And intravenous administration, just to see the bioavailability of the progesterone, we administered through the tailwind. And blood sampling time points, these are the blood sampling time points, and the blood was collected through the jugular vein cannulated animals. and 4 animals was used in the group. And we can see the bioavailability of the compounds here with the oral progesterone capsule. The bioavailability was something like 2.36. Just the progesterone was filled in the capsule. And with the cyclodextrin complex filled, we saw around 6.3% bioavailability. Even with the increase in the fold was 2000. And we didn't see the prominent improvement in the bioavailability, even with the cyclodextrin complex. Just we wanted to evaluate this. Why is the reason? What is the reason it's happening? And just to give a preface, and why was this? I can give an example. Prior joining to University of Mississippi, I was working with Jubilant Biases back in India in DMPK department. Even in that cases, we were seeing similar kind of trend, saying that even with the solubility enhancement in drug discovery stage, we were not able to get an sufficient bioavailability. So just with that background, I thought what could be the fate of the cyclodextrin complex in the intestine. So I just evaluated what could the solubility of progesterone complex in different simulated gastrointestinal fluids. It's a fasted state gastric fluid and fed state gastric fluid, and fasted and fed state intestinal fluid. The solubilities were examined. And here we can see there is increase in the solubility of progesterone to some extent in the passive, that is fed state intestinal fluid. And for complex, we can see that all the three, it's something like in water and fasted and fed state gastrointestinal fluid, we can see that there was no difference in the solubility. However, when we saw that in case of fasted and fed state intestinal fluids, we saw there is a decrease in the solubility. The decline was somewhere around 33% decline in the solubility in the fastest state simulated intestinal fluid. And when we saw with the fed state, it was further decreased. It is something like 80% was declined. Here in this case is something like we can see what could be the reason for the decrease in the bioavailability, even with the solubility increase, because there is precipitation of the drug in intestine. So we wanted to check what could be the reason for decline in the concentration. As we know what were the contents of the stimulated intestinal fluid, we just titrated, saying whether the different components of intestinal fluid can make the progesterone to precipitate. So we performed an assay where these different components were, different components were titrated against the solubility. And we saw that sodium taurocholate, increasing the sodium taurocholate concentration was decreasing the solubility of progesterone. Then what we assumed is that progesterone complex, when in presence of sodium taurocholate has a higher affinity to the cyclodextrin, it replaces the progesterone. So at these cases, we felt that, okay, we need to optimize the concentration of cyclodextrin so that the solubility of progesterone can be maintained at the intestine when drug is administered through oral route. For that, we just wanted to see what would be the optimum concentration of cyclodextrin required to maintain the solubility of progesterone when given on oral route. So the experiment were done, and we can see that this is something like 15 millimolar of sodium taurocholate in water was used. The initial experiments to see whether the excess of captisol, that is cyclodextrin, can improve the solubility of progesterone or not. And we felt it was improving. And further, we just evaluated what could be the optimum concentration of cyclodextrin required in the fed state in animals and fasted state in animals. With estimating this concentration of excess cyclodextrin was added in the formulation, and those formulation was administered to animals. And before that, we just wanted to see whether the excess of cyclodextrin will impact the permeability of the drug across the intestine. We performed an assay. Ex vivo rat intestinal permeability study was done. with complex. And with this, we found that there was something like with increase in the concentration of the cyclodextrin, there was increase in the permeability as well. So we thought, okay, it would be better to get with this. And with this, we just again performed another group of animals where we gave an progesterone complex with an excess cyclodextrin, which was been calculated using the in vitro experiment. And we found that something like close to twofold increase in this bioavailability compared to the progesterone. And this is something like we felt this is quite significant improvement from the twofold to 2% to 11% increase in the bioavailability. And further, we just wanted to check whether in humans do we need to add the excess of progesterone. So we performed an dissolution studies, and we saw that from the cyclodextrin, the release of the drug is something like instant within 15 minutes. So we felt that instantly it will be once it is administered to humans, it will be instantly dissolved in the stomach, and there is no impact of the fed state intestinal fluid effect of intestinal fluids, fed state intestinal fluids on the solubility of progesterone. So we just concluded the addition of excess cyclodextrin not needed, which would decrease the bulk of the formulation in clinical cases. And summary and conclusion of the project is something like the progesterone solubility was increased by the cyclodextrin, and in vitro experiments were developed and employed to determine the amount of cyclodextrin required to prevent the progesterone displacement from the cyclodextrin cavities. And this experiment can be used in industries in drug discovery stages, where cyclodextrin is the major solubilizer used for increasing the solubility of various lipophilic drugs. It's something like default solubility enhancing molecule is cyclodextrin. If we find something like in that where using cyclodextrin is also not improving the bioavailability, we can just perform an in vitro assay to identify what is the root cause for not increasing the solubility. And overall, it's something like there was 5-fold increase in the bioavailability compared to the progesterone API suspension. And this work has been published in the International Journal of Pharmaceutics. And the next project, what I will be talking is on development of silymarin topical formulation, and it's in vitro and ex vivo dermal kinetics of silymarin. And this product has been the project, as I earlier mentioned, this was been funded by Ligand Pharmaceuticals, and this product has been patented, and it's they are looking for partners for scaling up the formulation and marketing it. Introduction is something like silymarin is something like it's an extract from the Silybum marianum plant. It is it's something like it's a silymarin extract contains 7 constituents. I'll be getting into the 7 constituents with next slide. And silymarin has been traditionally used from over 2000 years for liver poisoning, hepatic poisoning, and everything. There is the main, they call it as mushroom poisoning, that is Amanita phalloides poisoning. And also with this silymarin's antioxidant potential, it has been seen that they are clinically been evaluated for hepatitis B and C, liver cirrhosis, alcohol intoxication, and paracetamol poisoning, and fatty liver disease. And also there is some studies are going on to see its effect on Parkinson's, Alzheimer's, multiple sclerosis, and amyotrophic lateral sclerosis. And also the clinical studies, people have been getting recruited for seeing its effect as an adjunct drug for myocardial infarction, asthma, lung carcinoma, and diabetic nephropathy. And this is because of its antioxidant potential, And various in vitro studies from past 20 years have been proven that this can be used for UV-induced cancer. And it's something like various cell line studies have proven, and even some animal studies have proven with Silamarin is useful for helping to treat cancer. With this background, we wanted to check whether Silamarin topical formulation will be useful for treat the UV-induced erythema, and also something like the studies have shown that the melanoma and non-melanoma skin cancer can be prevented by application of Silamarin, and which protects the skin from the UV light. And also there is also the utilities more for the Silamarin because of its antioxidant potential. There are rosacea and melasma and vitiligo. Wound healing also has been evaluated, and also the psoriasis. The objective was the first objective of the project was to determine the non-specific binding of Silamarin to the human skin and intrinsic skin clearance of Silamarin in human skin model. And the second aim was to develop the topical formulation to potentiate this Silamarin skin penetration and permeation. And I said that Silamarin constitutes an extract, and the major constituents are these seven molecules: the taxifolin, silcrystin, silidyanin, silibin A, silibin B, and isosilibin A and B. And we can see that these are isostereomers. These 6 molecules have the same molecular weight, and except this is something like all these 6 are derivatives of taxifolin. And just because we wanted to develop formulation with entire extract, the main objective, the first challenge was to develop an HPLC method for the HPLC method and validate it for our routine purpose. This was the HPLC method developed, and this is the chromatogram for that. And further, to check what is the Silamarin constituents physicochemical properties, it's something like as we know that the main objective was to see what was the constituents present in the extract what we have been procured, because we know that the extract from the different sources have different constituents in that. So we just wanted to check what is the constituents, and we saw that there's something like Silamarin constituents in the extract was something like 11 mg per gram of taxifolin, and highest being the silcrystin and the lowest being the isosilibin B. And we wanted to check the solubility of these molecules in the water, as we were planning to prepare oil in water based formulation. And we saw that solubility for taxifolin and silcrystin was highest, and the lowest was isosilibin. And also we wanted to check what is the physicochemical properties, other physicochemical properties. And we evaluated the log P, log D, and pK of the molecule to just predict what could be the permeation and penetration of the drug. And log P, you can see the lowest was for taxifolin, and the highest was for isosilibin A and B. And log D, we can see as these 4 isodiastereomers, the log D at the pH 7.4 was something like similar in the same ballpark, and pK was for all the molecules was similar. And again, we just wanted to check what would be the solubility of all the components in the cyclodextrin, and we can see there was somewhat non-linear curve, but there was increase in the solubility of Silamarin constituents with in presence of cyclodextrins. And the fold increase, we can see it's something like for the molecules which had a higher log P had an higher solubility, as we know that the lipophilic cavity has some more affinity for the lipophilic drugs. There was 600 to somewhere around 400 to 700 fold increase in the solubility. And further, just before getting into the straight away to the formulation part, we just wanted to confirm whether the drug is undergoing any protein binding, or whether it is going a protein, whether it binds to the skin homogenate, which might decrease the availability of the drug for elucidating its therapeutic activity. And also the metabolism studies in human epidermal keratinocytes and human dermal fibroblast cell lines were being used. As we know that we say that something like when we use the drug through this, when we are delivering the drug through the skin, we say that the drug might bypass the first pass metabolism. It's something like it's been believed so, but there are some cases, something like testosterone, which has been highly metabolized by the skin enzymes, and also the procaine has been metabolized by the skin enzymes because of the esterase enzyme present in the skin. So we just wanted to evaluate whether these constituents are getting metabolized by skin or not. And we found that it's something like off-life were more, something like we can see that the off-life starting from for taxifolin, it was 30 minutes to, it was going up to 60 hours. We felt it was safer to deliver through the skin, and it is not extensively metabolized. And also the percentage of bone to the skin, just wanted to see whether it is binding and we will be losing any activities of the drug. We saw the percentage of a drug bound to the skin, and we found it was it's something like it's very minimally binding to the skin. With this information, we wanted to see what permeation, and initial studies show that silymarin was not permeating across the skin. So we just wanted to screen different permeation enhancers. And all these permeation enhancers were been something like we got from the different companies like BSF and GetFSC, who were funding us, who were funding the university for various projects. And here, the initial studies were performed with the porcine skin epidermis using a French diffusion cell. And we saw that initially only we didn't see any permeation of the drug with the blank. So that is controlled when it is solubilized in other solvents and using it as a vehicle. When we use cyclodextrin, we saw some extent of permeation of the drug into the tissues, that is penetration. And further, we saw that this three enhancers, PEG 400, propylene glycol, and transcutal, showed an enhanced penetration of the drug into the porcine epidermis. With this information, we started developing in formulation. And here we use an Silverson homogenizer to develop in formulation. And it's a conventional method what we used. And just the time programming for the Silverson homogenizer was been optimized with 0 to 30 minutes 70 degrees centigrade, and 30 to 90 minutes 70 to 25 degrees centigrade, and 90 to 120 25 degrees centigrade to get a stable formulation. And oil phase, we used these ingredients, that is light mineral oil, viscosifying agent, and emulsifier, and octododecanol. And aqueous phase was something like complex was 57%. With the enhancers, we used the propylene glycol as an enhancer, as we saw that it enhanced the penetration of the drug into the porcine epidermis. And the transcutol, the permeation enhancer, because of its stability issues, we just wanted to add it in later stages, and which was been added during the cooling stage when the temperature was below 50 degrees centigrade. And with this, something like different compositions were used, around 50 formulations were prepared, and to see the stability of the compounds of all the formulation what we prepared. A quick pre-stoss cycle tests were done, where for 7 days at the formulation were kept at 7 days at the room temperature, and then it was placed at minus 20 degrees centigrade for 24 hours, and again at 24 hours at room temperature, and again higher temperature at 24 at 25 degrees centigrade, and again 24 hours at room temperature, to just see whether the formulation what we prepared, will it be stable at different temperatures. And the main evaluation parameter we used was something like evaluating the global size distribution of the formulation, and visually observing whether the formulation, we are seeing any phase separation in the formulation. And based on this global size, we optimized, and from out of those formulation, we selected 6 formulation, which has an consistent global size across different temperatures. After that, the 6 formulations what we prepared, in vitro release profile were seen, and we can see that the in vitro release profile for the 2 formulations for I, that is F5 and F6, and even we can see the same with even in the release rate as well also it was increased. So these 2 formulations further were and we with the studies why it was been increased only in this formulation. We checked viscosity studies where we found that presence of the components, as we use different excipients like CSA50 in the formulation, which has increased the viscosity and thus decrease in the release profile. With this information, we took 2 formulations for evaluating into the human skin, where permeation and penetration of silymarin constituents across the human cadaver skin was evaluated for formulation F5 and F6 with the silymarin cream application. And we saw that with F5 and F6, the increase in the penetration and permeation was more for the F6 formulation. And the rationale behind that was something like F6 creams contains 4% of surfactants, that is MCSE and GMS, whereas F5 creams contains only 3% of surfactants. So the increase in the surfactants has increased the permeation and penetration of it. And further, we wanted to evaluate how many applications do we need to get the required penetration and permeation. We saw that was increased with the increase in the time for the both the constituents. Even the permeation and penetration was been increased with the both the cases. And the summary and conclusion is something like we saw that there was low specific binding of human skin to the human skin components, and in vitro clearance and study indicates that silymarin constituents have a longer elevation of life in skin except in silydianine. And cyclic enhance the permeation of penetration of the drug into the porcine skin epidermis for these 5 molecules, that is silcrestin, silydianine, silibin A and B, and isosilibin A. Propylene glycol and transcutol in the complex potential the skin penetration of silymarin constituents. And cyclodextrin cream developed increased the skin penetration and permeation of all silymarin constituents across the skin. And this formulation was evaluated in rat to check its potential to show its therapeutic effects. And we saw that significant it has a significant improvement in protecting the sunrays getting into the skin, and also in treatment of UV arrhythmia. I'm not presenting that data here. By this, I'm just concluding my presentation. I want to thank my research group, that is Dr. Murthy's research group back in university, and all other collaborations, something which I had during my university and my research career with federal agencies like NIH and FDA. I worked on the projects funded by NIH and FDA, and my proposal was funded. I got my NIH funding for one of my proposal in PhD, and all this ligand, as I mentioned, these 2 projects were funded by ligand, and FOSA and BSF and Rocket America, they supplied the excipients and also funded various projects. And these are the 3 organizations where I was working with Jubilant Biases before my PhD, and University of Mississippi, where I worked as a research scientist, and C4 Therapeutics. By this, I conclude my presentation. Thank you for giving me the opportunity to present here. I'm ready to take any questions.
Hello, Dr. Vijay Shankar. It was a very nice presentation, especially a brief comparison of the cyclodextrin and their derivatives was very nice. I have a question regarding Captisol. Is there any reason why Captisol enabled products have not been still approved for oral and topical use? It's only for the parenteral delivery, right?
Yeah, it's been parenteral delivery. That's the reason I started with working with the ligand pharmaceuticals. They just wanted to check the applicability of the oral and topical. So it's something like they are funding a different university to check the utility of captisols in oral and topical. Based on these results, they might get the approval for getting into the topical and oral.
Thank you. Other chairpersons have any comments or questions?
Very nice presentation and thank you, Mr. Vijay Shankar. No question from my side.
And thank you, Dr. Manoj.
Thank you, Dr. Vijay Shankar. It was very nice presentation and very informative for those who are working in a similar field. I hope the student can also learn with this, and
yes,
this industry academia interaction will help them to learn more better in their future. Thank you very much.
Thank you, Dr. Manoj.
Actually, it was a very good presentation, and I don't have any questions to you. Thank you.
Thank you, Professor Salmi.
So thank you, Dr. Vijay Shankar, for your nice presentation.
Thank you, Dr.
This discussion will help our young pharmacists who want to do research in this area a lot. And we thank you for your time. You can remain in the valedictory function, which will start from 3:30 p.m. Indian time. And meanwhile, you can take rest. So it is night time. So your award announcement will be at that. So we will invite you there. Okay.
Sure.
Okay. Thank you, Dr. Vijay. So without taking not much time, I will invite our next speaker. Let me see the schedule. Mr. Bisam Singh, who is the Director of PharmaWise Solution Private Limited, Dehradun, Uttarakhand, to deliver his expertise speech. Welcome, Mr. Bisam Singh. The floor is yours.
Good morning to all. And first of all, I would like to thank dear persons, Dr. Rajiv Dayaji, Founder President APP, Dr. Kashyap Nagaria, National Coordinator APP, IPDRA Division, Professor Ramesh K. Goyal, Chief Guest and Vice Chancellor, Delhi Pharmaceutical Science and Research. Apart from this, all respective chairperson who is attending these programs, I'm very impressed with the theme which is selected by this APP convention program, Academia Industry Interaction in Pharmaceutical Innovations. I'm a person from pharmaceutical industry, and all my career is spent in this area only, and quality systems and other functions of the department. So I would like to just present some of the key topic here. Yeah, can you see the presentation?
Yes, sir.
Okay, thank you very much. So my topic is knowledge sharing, bridging the gap between academia and industry through GMP and GLP function. I'm a director of Pharma Bio Solutions, which is the ISO 9001 2015 certified company and registered office in Dehradun, Uttarakhand. Uh, After completion of 30 years in pharmaceutical industry, I have just introduced my one of the unit at Najibabad, Kotwali Road, Uttar Pradesh, where I want to give training and auditing courses for all academia as well as pharmaceutical um, experienced professional. And this was my dream, which is completed recently, and this facility is going to be start soon. My education is I'm a MSc Chemistry, silver medalist of 1989 batch, and having 30-year experience in pharmaceutical industry, 10 year in API, and 20 year in pharmaceutical companies, various companies. I worked as a VP Quality and Compliance in different companies and handled various regulatory audits, US FDA, MHRA, WHO. And while working in Renversi, I have handled various launching of product in Europe and drug filing in US FDA, where one of the product was also commercialized. I've also attended Empower software training, Milford, USA, and Dubai. And apart from this, I'm a technically approved uh, and qualified auditor trainer. Various activities of our companies where we are providing auditing services, training, consultancy services, regulatory, validation services, GMP, GLP services, and other services wherever required, whether there is a academia or industry. So this knowledge transfer program was designed for academia as well as industry professional. While taking off from my full-time job, I thought that why we are finding difficulty in getting a suitable candidate, or you can say that competent person for our industry. And in last 2 years, I have gone through various the experiences with the academia as well as industry profession, and I have noticed that there is a huge gap between a job giver and job seeker. There are a number of students who are getting you know certification every year, and all academic faculty members are doing very working hard for giving the best knowledge to them. But uh, what is happening in the pharmaceutical industry, as soon as they are going in the company, so there are something missing, which is not expected from the academia whenever they are going in the industry. So, what is the main obstructions which industrial professionals are facing with them? First of all, the documentation part, which is not up to the standard of industrial level, because whenever we are working in a university or colleges, so the documentation is not is much important. You can override, cutting can be acceptable, but as soon as you are joining any of the industry, what is happening there? So the regulatory bodies are coming from different regulatory authorities, and as soon as they are just are doing inspection and finding some overwriting or cutting, so there is a suspicious thought process whether you did some data integrity or some other problem is there, or you are hiding some things. So that is a big problem. The second problem is that concentrations and discipline. The the fresher B farm or M farm my student is so enthusiastic, energetic, and he or she want to achieve everything within a short period. However, whenever joining the industry, you have to go through each and every step. You have to learn because first of all, you are in a learning phase, not in earning phase, and that is the biggest difference, which is also making everyone, in that situations where the always challenge is there, and every senior or your colleague is monitoring you, supervising you. That is the way of working in industry. So this is the second gap. And the third and major gap is that without understanding any of the organizations we start to join, and then within a short period times we want to just leave the job. So there is a impact on the CV, biodata, and confidence. And whenever any person is going for a second job, there is a lot of question why you left the job early. So these are certain issues which is industrial professional are facing. Otherwise, academian are very good. They are doing very well, and they are also putting a lot of effort. But if we can improve in certain area, we can be a very good and competent and qualified professional. So I have just highlighted certain points in our pharma bio solutions services, and this is knowledge transfer program which is giving lot of indications how we can control such type of things. So while understanding core issues, primarily we have noticed huge gaps between job seeker and job giver. We have planned knowledge transfer program for freshers, graduate, postgraduate who want to initiate their career in pharmaceutical industry, and we are training them and grooming as a fresher as per industrial standard. And we are also bridging the gap by shortlisting the candidates and giving basic training to them and presenting as a trained freshers to various companies. Where the company's HR professional are in our the touch, and they are regularly taking help from our company. We are also providing basic and advanced courses training to junior pharma professional, whether from industry or academia. And this knowledge transfer program is of 8 to 10 hours training and GMP or GLP aspect, as well as department topic. And we are also providing this training, medical training certificate to qualified professional, and 1 year validity for career counseling and regulatory update to registered WhatsApp number. So this is uh, this is our company way of working, and we are helping in our way to various professionals who are who want to start their career in a industrial level, whether there is R&D or QA, QC, or productions. Now I'm coming to the other topic of good manufacturing practices. So we have discussed about our pharma-based solution services and knowledge transfer program, which is bridging the gap between academia and industry. Now the second topic will be our good manufacturing practice, and the last topic and the third one is good laboratory practices. So again, these topic, the good manufacturing and practices and this good laboratory practices are also playing a very important part while the candidate is working in industry. So what is the expectation of any regulatory body? The good manufacturing practice as a system for ensuring the product should be delivered consistently with the same quality and standard. You can let me know if you are, the slides are not moving or something you noticed. Okay. Now the good manufacturing practices supported by the lot of regulatory guidelines also. So in last few years, a lot of guidelines published, whether there is a combination product FDA, documentation EU, drug product Health Canada, then herbal medicine products EU. So each regulatory body, whenever they are just publishing any guidelines, so we are just keeping into the consideration for knowledge purpose. And there are some other guidelines also. So these guidelines are the key document for pharmaceutical industry, how to run, how to design the policies and documentation. And if you are not meeting these guidelines, then you cannot, perform any your product marketing or distribution in any of the country. So unless otherwise you are not following these guidelines, no one will give you the approval of your product. So in this series, apart from the various guidelines, there is a 21 CFR, which is the Code of Federal Regulations USA, and there are various part under this 21 CFR, and we are just concentrating the whole part for good manufacturing practice, and this highlighted subpart I for laboratory control. So this is the pocket guideline, and this is the regulation which is already defined by US FDA. If anyone want to do the business in USA, then you have to go through each subpart and place these documentations in the respective industry or academia. Then only you can achieve the license of this regulated body. And once you have placed these guidelines, you have to follow. And whenever there will be a audit, you have to go through these SOPs only. What is GMP and law? So the regulatory authority already defined the regulations in different regulatory guidelines, whether they, if you are dealing in India, then Schedule M is there, WHO is there. If you are dealing in USA, then US FDA is there, MHRA, TGA, various regulatory bodies there. So regulation is already written, you have to follow. But if any company or any academia is not following those regulations, then regulatory body have a power to stop manufacturing from producing. So you cannot manufacture, they can stop manufacturing. They can also stop sale of the product or close down the plant and facility. They can suspend or revoke manufacturing license, prevent products from entering into the country, bring charges which could result in fine and jail sentences. So these are the regulations which is placed because until unless you will not have the authority, you cannot enforce certain rules and regulations. So the US FDA is the regulatory body which is taking a lot of aggressive action in these areas. Then who regulates the GMP rules? So the regulatory body, whether there is a US FDA or there is a ICS guideline is there, International Council for Harmonization of Technical Requirements, or there is a fixed guideline is there. These are the regulatory body which are regulate the GMP rules in any of the pharmaceutical company, and the government regulatory authority like Food and Drug Administrations. And then they also perform audit and sharing their information to each other. Until unless you are not sharing information with each regulatory body or wherever you have a agreement, you cannot conclude, or you cannot cover all the companies. That's why they are sharing their audit reports and then taking actions based on sharing of information. Then regulatory authority also provide framework also. Not only they are punishing or they are just doing audit, but they are also providing framework for regulations of medical goods to ensure quality, safety, and efficacy of medicines. So time to time, they are just publishing some guidelines or they are making awareness within the professional to follow and utilize the benefit. Now, this is very important part. Whenever we are designing any of the product, whether at academic level in R&D or at pharmaceutical industry, so we have to think first of all, this part is a quality by design, a systematic approach to development that begins the predefined objective, emphasize on product and process understanding, whether there is a selection of vendor or procurement of raw material or packaging material, or you are designing some experiment, you have to first of all write all the requirements and then make sure each and every step must be followed. And whenever is any discrepancy or incident observed, you have to note down or you have to record. Then only during investigation you will came to know what was wrong in my experiment. So product must be designed and developed with the GMP and GLP in mind. So this is begin with the R&D work or developmental work. Sometimes the scientists are thinking that this the documentation and GLP and GMP is not required for R&D work. Of course, not required. You have a liberty for doing so many experiments, but you should, if you are maintaining documentation, you are following good manufacturing practices and a good GLP, this will give you the return while you are completing your experiment. So the sufficient documentation will be available at the end of the experiment, and you can present to your management that I did such type of studies, and this is my final conclusion. This is one of the example how the pharmaceutical quality systems is running in industry. So there is a pharmaceutical developmental lab is there. Then after the developing a product, then final product technology transfer can be planned to production as a process transfer and QC to as a analytical transfer. So the second step is technology transfer. Third step will be the commercial manufacturing, maybe validation batches or optimization batches will be there. And once your product is manufactured and distributed into the market, then product discontinuation is the last step. Until unless you are not also doing discontinuation of your the product and you are keep adding the product, so lot of the documentation issues will arise. While you are manufacturing these products, there will be a investigational product also, like exhibit batches or ANDA batches will be there. So these batches can be followed under the technical transfer and this same process. There is a management responsibility to provide resources, man, machine, and material. And the PQS, performance, process performance, and product quality monitoring systems must be placed. And if anything happen wrong, then corrective and preventive action is required. Change management is required whenever you are changing any process or changing equipment or any breakdown. Then management review is very important. Whether you are working in academia or industry, you need to have a monthly management review meeting. And during the monthly management review meeting, all HOD must participate and give the presentation. And the chairperson or maybe a chairman or MD, they can note down if there is any resources related issue. They can also check whether my organization is doing well or we are doing some improvement on regular basis, or you have to be associated with the staff and the worker. Then only you will be able to take a right decision. There are some organizations where the management is coming once in a year. So ultimately the responsibility of management, because they are getting all license and all commitment by own. And until unless they are not involved in this process, you cannot make a progress on time or on innovations. Then there is a knowledge management is very important. Time to time, you have to give technology transfer to the process, as well as knowledge transfer to the your manpower or their uplifting. And then quality risk management is very important. Whenever you are doing any investigations or you are taking some major decisions, whether there is a deviations or change control, then you need to consider the risk associated with the process or manpower. Okay, if there is a low risk, you can take easily decision. Moderate risk is there. You can also take a decision with some conditions. But if the risk is high, then you have to think what will happen wrong, whether there is a patient-related issues or safety-related issues. You have to consider all these points. Now, another part in a pharmaceutical industry is the quality systems. And whenever we are just dividing all department as per US FDA 6 system. So these are the 6 systems they are considering in audit. Quality system is quality assurance, which is a centralized or leading department. You can say that. Apart from this, there is a production system is directly linked with the manufacturing part, where you are doing tablet or capsules manufacturing. Then the second part of production is packaging, labeling, and system, where you are doing bottling or blister packing and labeling work. The third system is facility equipment. This is regarding to the engineering functions, building facility and equipment. And then there is a laboratory control system, laboratory QA, QC, micro, and stability program. And the last one is sixth one is the material system, which is related to the purchase of material, whatever material, raw material, packaging material, water, or any miscellaneous item we are dealing so under the material system. So these 6 systems are very important. Whenever US FDA auditors are coming at site, all the time they are checking these quality systems. And apart from this, they are considering all 6 systems first time, but during the revisit, they are considering maybe quality system plus 3 more systems. They cannot check all the system every time. There is a personal training requirement, and this is very important part. Any organizations, whether academia or industry, success is dependent upon the personal and personal training. If you have a qualified manpower, then the senior person job is very easy, or even management job is very easy. All the time you will get the results on time without a lot of breakdown or losses. So the GMP guideline recognize the training requirement as per the regulatory expectations. You need to go through this twice year. It means you have to perform every 6 months CGMP training. And if you are doing this training and doing assessment, then there is a guarantee of the success in your organizations. So this GMP guidelines recognize training, and during an audit, an auditor may ask questions. That's why the competent person is required, because until unless you are not technically and administratively strong, you will not be able to understand the question properly and give the wrong answer. As soon as you will give the wrong answer, the directions of audit will be turned in a different area. So identify the skills and training required for each role. This is the job of the senior management. Then training record to be maintained, and these records must be kept for everyone. My recommendation, starting from director to the worker, you need to maintain the training record, because director is also involved in taking a decision for the organization. So he must be aware with the policies and SOPs of the academia or industry. Then apart from this, you have to consider contractor, cleaner, vendors for such type of training program. Because if they are not aware with the training requirement, then they may have some of the breakdown in the area or safety issue will be there. Then apart from this training program, there is a good hygiene is very important. Whenever any of the employees going for industry, they have to make sure that there should not be any open wound. Appropriate washing of hand is required. No eating, drinking, or chewing or smoking item is allowed to kept in the drawer because all these items are having a nature of decaying within a 3 to 5 days time, and then microorganisms will develop that will create a lot of issues either in change room or in other area of the facility. And all personnel entering into the manufacturing area should cover their head or other body parts so that there should not be any fall of hair. Unhygienic practice while manufacturing or other area product may affected badly. So you have to make sure all hygienic conditions while you are entering into the um, area. There is a quality management required as per ICS guideline ICH Q10, where senior management as well as each person is responsible for the participation and commitment. Because until unless the junior staff is not involved in this process, you cannot place the quality management in any of the organization, because the staff is also providing informations. The incident happened, deviation happened, and recorded and reported on time. Until unless you are not doing this type of activity, the real data will not be available for recording. So the quality management, of course, the responsibility of senior management of any organization, but the staff involvement is very important. There is a qualification cycle, equipment qualification cycle is defined here. Whenever you are procuring any of the equipment or machine in your any organization or academia, then you have to go through this process. Apart from user required specification URS, the DQ must be asked from vendor. IQ, OQ, PQ document will be provided again by vendor, but execution must be taken place in your presence. Then after completion of this installations, certification is required and GMP process under control review periodically. So once the instrument is placed, this is the function of department head and HR, this quality assurance head, to make sure all change control procedure, deviations, incident recording must be followed. And training is again the part of this process. So whether there is a software related training or hardware, you have to identify certain person and in your department who can participate in this process. Otherwise, all the time you will ask a person from the service engineer end for giving the training. Then in the GMP, there are 10 golden rule, which is concluded in the good manufacturing practices. The first golden rule, if any organization is following these golden 10 rule, then there is a assurance that you will have reasonable compliance in your facility. Golden rule number 1, get the facility design right from the start. So first of all, if you want to start any of the organization, then you have to design your facility and plan in a better way with a competent person. Second, process should be validated, whether there is a process or regarding the productions or testing. You have to validate all process, whether there is a cleaning validations or maybe qualification process. Then rule number 3, write good procedure and follow them. Whenever you are writing any SOP or procedure, must be in line with the regulatory guidelines. But at the same time, you have to give this procedure to the down the level so that they can easily familiar with the procedure. There should not be any language which is hindrance for understanding. Golden rule number 4, identify who does what. This is very important because you are a senior person of the organization. You have to identify what is the capacity of any of the person, and then you have to utilize the best capacity, best talent. And there are certain person who is having a talent, but put into another area, and ultimately we are draining the brain. Rule number 4, keep good record. Once your record developed, then you have to maintain also. Then train and develop staff. This is important part. If you have a very trained and qualified staff, then most of the person will be able to execute the job on time without repeat work. Practice good hygiene. This is also, giving you the best hand if you are maintaining the hygienic condition at your site. Maintain facility and equipment. So again, this is also a very important part. Whatever building or facility is available with you, you have to maintain on neat and clean on timely manner. Building quality into the whole product life cycle. So again, designing quality by design is starting from development to the distribution of your product. You have to go into the quality of the product. Then perform regular audit. This is one of the best part of this golden rule. You have placed everything. Manpower is there, machine is there, infrastructure is there, but regular audit is not there. Independent audit is not there. It means something, hiding, and that can create a lot of issue for your organization. Suddenly, the outside auditor is coming and they are finding some surprise. So that can give you the very bad impression to the organization. So regular audit and independent audit by the competent person or agency is very important. And you have to take action on audit finding also. Now, after completion of knowledge transfer program, this good manufacturing practices, now our third phase of good laboratory practices regarding the laboratory functions. So the foundation of quality control is based on adequate facility must be there and trained personnel is there, because these are the two function in quality control lab. In quality control lab, we are not doing any manufacturing or we are not doing any other activity, but only.
Excuse me,
Yeah,
Mr.
please.
Bishop Singh, our new
Yeah.
session will start at 12:30, so you have to bind up.
I'm
please.
just closing within 5 minutes.
Okay.
That is okay?
Okay. Because
Yeah,
other
thank you.
person are waiting to join.
Okay, sir. Thank you very
Okay.
much. So in the laboratory, there are various sampling activities going on, like raw material, packaging material, intermediate, finished product, process validation, cleaning validation, stability sampling, water sampling, and environmental sampling. So these are the different sampling happened by the QC person. Similarly, the testing of raw material, packaging material, intermediate, finished product as per specification and release of CA. And then other activity, process validation, cleaning validation, stability, water testing through the various protocol. In a QC lab, there are a few instrument or critical instrument. I would like to say that dissolution apparatus, where you have a basket and this pedal facility, and this instrument is playing a very important role finding of the drug release profile or pattern. The second instrument is high performance liquid chromatography. Again, HPLC is playing a very important role for related substance testing assay or any the scanning, whether there is a cleaning validation method or some stability testing procedure. Then stability chambers is very important for any of the industry or even developmental lab, where we are handling different type of conditions, whether accelerated condition 40-75, long terms, intermediate, and there is a zone 4 condition is also given there. So it depends upon the company business, what type of the stability chamber is required or study is required at your site. So thank you very much. And I have just gone through my sessions. You have a question, you can ask.
Thank you, Mr. Visham Singh, for explaining all the processes and stages of the GMP. I have a question about these things that current good manufacturing practices and good manufacturing practices. So the practice that we right now follow in India, is it CGMP or GMP?
It's very good questions you have asked. This technology, GMP, is used in various domestic plants. Like if you are dealing with the business in India only, so they are following Schedule M, and generally they are, telling that GMP is there or GMP certification is there. But when you are dealing with the US FDA, so they are talking about the CGMP, means current. Every time you have to follow the current good manufacturing practice. The reason behind that, various the organizations claiming that this is our SOP and we are following this SOP. But there is a questions by agency, why not current USP chapter, why not current guidelines? So that's why they have put all the time C. And whenever they will audit, they will check with the current guidelines only. They will not claim any the excuse by the agency.
Okay, okay. And about GLP, is the GLP an integral part of the GMP? That means if any firm is having a GMP compliance, does it mean that all the laboratories of that are GLP standard?
Whenever any agency is giving the certifications, so they are considering as a GMP certifications. Okay. Under the GMP certification, laboratory is one of the function they are just checking during audit. So you have to get the certification for GMP as well as GLP from local FDA, but for regulatory agency, there is no such discrimination at all.
Okay, okay. Thank you so much for this clarification. And again, many, many thanks for your time and sharing this presentation. Thank you.
Yeah, please.
Good afternoon, Dr. Bishman Singh. In order to become an auditor, qualified auditor, what would be the procedure? It may be a dream for some students to become auditor.
Yeah. Oh, very nice. That's a good question also. First of all, the fresher cannot be auditor within a short period, or maybe you can say that just joining, they will become an auditor. There is no such process. However, as soon as they join any of the organizations, they have to spend some time in a training process for at least 1 year, 2 years. Then they have to select the department, which is the quality assurance department. And if they will go join the quality assurance department, so within a 2 months, 2 years time, they will assist the senior and get the exposure of self-inspections or internal auditor programs. And once they will get this, that exposure of at least 3 to 5 years, they will get a certification of this lead auditor course also, which is the ISO certified course. And I'm just giving this the guidance to all junior professional. They first of all they have to assist their senior and then get the internal auditor course. which is a 2 days course. And then after getting sufficient experience, they should go for this lead auditor course of 5 days. And my organization is just doing this certification because I am a lead auditor as well. Our company is also certified agency.
Nice. Okay. Okay. So is there any global organization that recognizes your training and your certification?
Is there, IRCA is there.
Okay. Okay. Okay.
Yeah. And various accreditation body is there, which they are providing, like there is a URS or some other agency also. But this ISO certification lead auditor course is acceptable by all regulatory bodies.
Okay.
Yeah.
Thank you. Thank you so much.
Any other questions, sir?
Thank you very much, Mr. Vishan, for giving a very nice presentation on the PharmaWise solutions. I hope this will help our students as you are talking about the future, the gap of the industry need and what we are teaching in the class to them. So I feel maybe this, with the help of your PharmaWise solutions, our students will be make up their ways to go for the industry for excellence. So thank you very much.
I'm eager to help all academia who can take my experience and the way of business is something different. I'm not totally involved in a commercial activities. I'm also involving in some social activities also. So the 50% commercial, 50% is social is my theme of my organization. So I will be happy if these chairpersons or some other key person, and anytime you can take my help, I will be there.
As we are near to you, I'm staying in Muradabad and it's.
Oh, very nice, very nice.
We are running all the courses given by the Pharmacy Council of India. So we are running the D Farm, B Farm.
Okay, sir. Sure, sir. Whenever I will have the time, I will meet
Definitely
you.
will keep in touch with you. Yeah.
Yes, sir. Thank you, sir. Thank you very much. Anything else, sir?
Okay. Thank you.
Thank you. I'll be keeping in touch with you, sir. Thank you.
Oh, sure.
Okay, sir. Very good presentation. Thank you for your precise and presentation about the GMP, GLP and academia interaction. Thank you. No question from my side.
Thanks a lot, sir.
Sir, I request Dr. Saurabh Dahiya, sir. Sir, I request Dr. Saurabh Dahiya, sir, to please make Rajiv Dahiya, sir, as host. By mistake, I made Dr. Saurabh Dahiya as host. Sir, is Dr. Saurabh Dahiya, sir, available?
Can I discontinue?
Yeah, thank you, Mr. Bisam Singh, for your lecture on GLP and GMP, and thank you for the knowledgeable discussion about. I'm thankful to all the judges who have made the discussion very interactive, and we hope for the future support from Mr. Singh in our future conferences also, where GLP and GMP expertise is required. So, so thank you very much. I will interact with you for the other interactions. Okay. Thank
sir.
you for your time.
Thanks a lot. Bye-bye, sir. Thank you.
Bye-bye. Bye-bye. So, colleagues, now we will continue the session of oral presentation.
Sir,
on
excuse me, sir.
Yeah.
Sir, this is Harita from St. Paul, sir. Sir, as I made you the host, I'm no longer able to record the session. You can record the session, sir.
So guide me how to do.
It is in the last tab. You'll directly have the record button, sir.
I'm not seeing. So you have to guide me. I'm not seeing the record button.
You can see near to the share screen, there is a record button. You can just push it.
Okay.
Yes.
So I
yes,
have
sir.
to record on this computer. This I have to click?
Yes, sir. Yes, sir.
Okay.
Okay, sir. Thank you, sir.
So, I welcome the judges of the oral session. Group B. The judges are Dr. Saurabh Banerjee, who is the Dean and Associate Professor of School of Pharmaceutical Management, IIHMR University, Jaipur, Rajasthan. And another chairperson is Dr. Saurabh Dahiya, the Associate Professor from DIPSAR, DPSRU, New Delhi. So welcome Dr. Saurabh Banerjee and Dr. Saurabh Dahiya, sir.
Thank you, sir. Thank you.
Thank you, sir. Thank you very much.
So,
And all the
as
best.
you
to
are,
all.
so your all presentations you have seen. So I think 8 presentations are there in your group.
Yes, sir.
15-minute time is allotted and 5-minute discussion. So if we are late, then you can adjust the discussion time accordingly.
Okay.
So you can finish by 3-5. Before the you have to evaluate all presentation on different criteria of presentation, answering the question out of 10 marks. And when you will reach second last presentation, you have to send me your evaluation sheet, and then at last of the remaining candidate, so that we can do the compilation.
Okay, sir. Sure.
So one more modification is there of this. All the participants are here. Let me take the attendance. Archana.
Yes, sir. I'm here.
Then Sobhagya. No. Yasmin. Reena.
Yes, sir.
Okay. Vanisha. Vivek. Tarun. Jishan. Okay, Vivek Pawar is there. Okay, no problem. So you can switch. Judges have the right to switch the presentation if anybody is not there. So you can take other available slot first, and you can tick accordingly.
Sure, sir. Sure, sir.
Okay. So I have guided you regarding the rules. Another one more variation is there. These all Group A and Group B presentation are leading to first, according to the document given, were leading to 3 awards, but now we have increased 1 more award. So on second position, there is 1 more award, which is Professor MC Goel Memorial Award, which will also will be with cash of 5,000. So there are 2 first and 2 awards with cash of 5,000. Third award is with a cash of 2,500, and fourth award with a cash of 1,500. All are APP Memorial Award. So there is 1 more addition in APP Memorial Award at second position with 5,000. So now I will request the judges to take over the session, please. Thank you.
Yeah, thank you, Dr. Rajiv. So straight away we can move on to our schedule, and we also have now here Archana Mandava. So you can share the screen, Archana, and then start your presentation.
Yes, sir. Thank you, sir.
Thank you.
Sir, I need permission to share my screen, sir.
Okay, guide me on that. How to do?
Dr. Rajiv, I believe you have to make her the co-host, then only she will be able to share the screen.
So
go on the participants.
Okay.
And then right click and then make her co-host.
Okay.
Yes, she has become co-host now.
Okay.
Yes, sir. I can share, sir. Thank you so much.
Okay.
Sir, is my screen visible, sir?
Yes, it's visible.
Yes. Good afternoon, one and all present here. Myself, Archana Mandava. I'm a research scholar and assistant professor at School of Pharmaceutical Sciences, Vistas. I'm under the eminent guidance of Dr. M. Vijayanandi. I am doing my research work here. today I am going to present my topic, that is kinetic spectrophotometric determination of some selected antiretroviral drugs in bulk and pharmaceutical dosage forms. So today I am going to discuss about two drugs especially, that is amitriptyline and tenofovir disoproxil fumarate. So a simple, accurate, reliable, sensitive kinetic spectrophotometric method was developed to quantify these two drugs in bulk and pharmaceutical dosage forms. So here the method, two methods were adopted, that is initial rate method and fixed time method were adopted. So here the drugs are going to react with the alkaline potassium permanganate and NaOH, which results in the oxidation reaction and formation of a green colored product, and that is maximally absorbed at 610 nanometers. So apart from this, the validation of the method was also done using different validation parameters according to the ICH Q2 or 2 guidelines. And apart from that, molar absorptivity, Sandel sensitivity, and other parameters were validated for the method. So coming to the introduction, first of all, these two antiretroviral drugs are used for the treatment of the HIV infection in adults as well as in the children. And the chemical structure of these two drugs were given here. Apart from this, hepatitis B virus also, treatment also will be given with these two drugs. So what is the need of the study for these two drugs is, individually, these two drugs are analyzed using different analytical methods, either individually or in combination, by different methods such as spectrophotometry, HPLC, HPTLC, UPLC, LCMS. These methods were used. Till date, no attempts were made to determine or investigate these drugs in commercial dosage form using the kinetic spectrophotometric method. So that is the novelty of this study. And what are the experimental requirements for this study are a double beam UV visible spectrophotometer, weighing balance, and the drugs in pure form. Next, preparation of a standard solution for these two drugs and the standard solution of the reagents like KMNO4 and NaOH. And according to the temperature and parameters are adjusted, optimized for the method development. So coming to the methodology, first is initial rate method. As I have already discussed, two methods were adopted for this technique, for this method development, that is initial rate method and fixed time method. So in the initial rate method, first the concentrations of both the drugs according to Bios-Lambert's law, 4 microgram per ml to 36 microgram per ml was selected. And to that, reagents like 1.5 ml NaOH and 2 ml of KMNO4 were added. And this mixture was kept in a thermostatically controlled. This mixture was kept in a thermostatically controlled water bath for 15 minutes at 60 plus or to minus 2 degrees centigrade. So the initial reactions of different concentrations were evaluated by using the absorbance versus time curve by measuring the slope of the curve. Apart from that, calibration graphs were constructed for the absorbance versus concentration of the drug versus absorbance, and the amount was calculated for both the drugs using regression equation. Coming to the fixed time method, so here the pre-selected fixed time was used for the different concentrations of the drug, like time being like 5, 10, 15, 20, 25, and 30 minutes. So for these time limits, fixed time limits, the calibration graph was plotted, absorbance versus concentration. Again, using the regression coefficient was calculated for this, for the amount of drug to be calculated. So coming to the determination, how to determine for the pharmaceutical dosage forms is by taking the 10 of EM 300 mg and tenofovir disoproxil fumarate 200 mg, both were weighed and crushed and powdered. From the 20 mg equivalent weight was taken for the two drugs, and they were added with a mixture of reagents. And the proposed method, that is fixed time method, was applied on for the method development and quantification. Now coming to the results and discussion. So this method development, why this method has been developed is, first of all, there are different types of species of reactants are there which are having different rate of reactions. So majorly this will follow the pseudo first order reaction. So that's why using this kinetic spectrophotometric method, the method is developed for these two drugs. So here the aqueous potassium permanganate solution in alkaline medium reacts with this both two drugs, and there will be oxidation reaction is happening. And because of this oxidation reaction, a characteristic new band was developed at 610 nanometers. So here the reduction is happening. The reduction reaction is happening. That is permanganate is converted to manganate ion, and the color of the green color product is formed at the end, which is stable for at least 15 to 30 minutes. So the intensity of the color increases with the increase of the concentration of the potassium permanganate. So based on this, the method was developed and the different parameters were optimized for the method, and then it was validated. So what the effect of KMO4 concentration here? It as I have discussed already, it oxidizes both the drugs in the presence of alkaline medium. So the amount, like different for optimization purpose, different molarity concentration of KMO4 was tested. That is from starting the 0.001 molar to 0.5 molar, which is the least concentration, and at 0.005 molar concentration, the method was optimized and we got the maximum absorbance. And next, effect of NaOH. NaOH was prepared using 1 molar concentration, and different ml starting from 0.2 ml to 3 ml was tested for the optimization purpose, and it was found that at 1.5 ml of NaOH as a reagent, the absorbance we got maximum. And effect of time. So for the fixed proposed method, which is fixed time method, different range of time settings were used, that is starting from 5 to 30 minutes. So at 15 minutes, the color was stable, and up to 30 minutes, it was stable, and maximum absorbance was observed at, especially at 15 minutes of. And coming to the temperature, at higher temperatures, so there is a product is getting decomposed into manganese dioxide. So up to like 60 degrees centigrade, I have done with wide range of temperature ranges, like starting from 0, 20, 40, 60, and further 80, like that I have increased. But at 60 degrees centigrade plus or 2 minus 2 degrees centigrade, it was the product was stable, and after that it started to decompose. So I optimized the temperature to up to 60 plus or 2 minus 2 degrees centigrade. So evaluation of kinetic parameters in the initial rate method. So according to optimized experimental conditions, it follows the pseudo-first-order reaction, where the reactant is directly proportional to the concentration. Rate is directly proportional to the concentration of the reactants. So using that initial rate method formula, I have calculated the regression coefficient of both the drugs, and they are also within the limits. So we can see here. It's coming to the fixed time method. Calibration graph was plotted for absorbance versus concentration with the different time limits, that is 5, 10, 15, 20, and 25, 30 minutes. So at 15 minutes, we can see the maximum absorbance as well as a correlation coefficient also was within the limits for the two drugs. And here I have highlighted here, that is 15 minutes, we can see 0.9997 as well as for amitriptyline 0.996. So these are the analytical parameters which were applied to determine both the pharmaceutical dosage form over the 4 to 36 microgram per ml concentrations. And here, apart from that, molar absorptivity, Sandel sensitivity, both were calculated. And apart from this, other validation parameters were also calibrated. Like coming to the linearity validation of the method. So you can see the standard calibration curve was plotted for the absorbance versus concentration, where I have calculated the regression coefficient for both the drugs, which are within the limits. And this is as per the Beer's-Lambert's, the concentration ranges were taken. And as the curve is linear, it is obeying the Beer's-Lambert's law. And accuracy and precision, intraday and interday precision was done for both the drugs using the fixed time methods. And the recovery of these two drugs were within the limits of the guidelines for both the tenofovir as well as the amitriptyline. Next, limit of detection and limit of quantification were calculated from the calibration curve, and they are found to be 0.45 and 1.36. And robustness of the method for each operational parameter, especially I have taken two operational parameters here, that is volume of the reagents, KMO4 and NaOH. So as per the absorbance values, maximum absorbance, we have got that to 2 ml plus or minus 0.1 ml is the optimized KMO4 volume, and NaOH is 1.5 plus or minus 0.1 ml volume will be optimized for to get the robustness of the method. So here I have done for the 4 microgram per ml absorbance what is the absorbance and what is the percentage recovery. We can see at 2 ml, it is showing the 100% recovery. And as well as in 36 microgram per ml also, it is showing 99.91% recovery. Same applies for NaOH 1.5 ml. I got 99.91, here 99.95. Same for the amitriptyline. So I can suggest that the method is robust with the whatever the operational parameters, especially KMO4 and NaOH volume we have taken. So the proposed kinetic spectrophotometric method is very simple, accurate, precise, selective, and low cost as well as reproducible. And I haven't used any critical reagents as well as sophisticated equipment for this. We need a UV visible spectrophotometer and regularly available reagents in the lab to quantify these two drugs in the bulk and pharmaceutical dosage form. These are my references. Thank you.
Thank you, Archana. The work of analytical development and validation is really a nice one. So I really appreciate your PhD work. I believe this is a PhD work. But I have certain queries, which is that have you used only the API for this method development and then validation, or have you used any tablets, capsules, or any dosage form? Also, have you tested on any pharmaceutical dosage form also? And another part of the question is that is there any official HPLC method for the two drugs of which you have used, emtricitabine and tenofovir? Is there any HPLC method available in any of the pharmacopoeia, and have you compared with the official method, your method which you used in the UV spectrophotometry?
Yes, sir. Actually, there are in the literature review I have studied, there are HPLC, HPTLC, UPLC methods are also available for this method. That is like quantification of these two drugs in bulk and pharmaceutical dosage forms. And even UV visible spectrophotometry also is there, but kinetic way of determination is not there. So that's why I wanted to try this in that way where we can use the regular reagents which are available in the lab, and it is reproducible also. And coming to the first question, that is API, pure form I have taken, as well as I have done with the dosage forms also 10-OEFM, which is like 300 mg, and Disproxil 200 mg I have taken. And from that I have taken the minimum 20 mg quantity of both the drugs, and I have performed the procedure.
Okay, basically what I believe is that you are oxidizing, you are taking use of an oxidation reaction of your drug with the potassium permanganate in alkaline medium, and
Yes,
then you are using it based on the intensity of the color produced. Then you are using it at 610 nanometer on the UV.
Yes,
so for the audience, I am asking you that what is the basic advantage or the better advantage that your kinetic spectrophotometric method is giving over the other conventional methods?
Sir, it is first of all cost-effective method, very reproducible, and next, because the different species are present in the mixture, which are following different rate of reactions, and in that case, we can adopt this method to quantify them easily.
Okay, thank you.
Thank you, sir. Sir, next I have to give to host.
Yeah, who is the next available candidate? Please speak over your speaker, your mic. Who is the next available person for the presentation?
Good afternoon, sir.
Good afternoon.
Sir, myself Saurabh from Uttar University, Manipur.
Okay. So Bhagya, then please share your screen. Let's move on to the second presentation of the day. Archana, can you make him the host, co-host?
Yes, sir. But I cannot see that option here.
You have to go to the participants list and then you have to click, right click on his name. Saubhagya Linka.
Okay. Sir, as I am a host, co-host, I cannot make him that one, sir. Sir, only Rajiv Dahiya.
Okay. Dr. Rajiv, if you can hear us.
So what? So Bhagya is registered under which name? I'm not seeing Sobhagya. Sobhagya?
Sir?
Okay. So your name is Pabri.
Oh, sir, sorry, sir. I have forgotten to on the video.
No, please tell what name is being shown here with your account. We are unable to locate you. Locate your name.
Okay.
No, I did. He's Jena under Pabitra Moham Jena.
This is my
So
uncle's account.
I made him co-host.
Okay. Now I am sharing the screen. Sir, is it visible to all?
It's coming. Yeah. Yeah, we can see.
It's visible now.
Yes, it is visible. You can start. Yes.
Yeah. Okay. Yeah. Please start.
First of all, good afternoon to all. Myself, Sobhagya Lanka. I am completed my MPharm from Utkal University, UTPS Utkal University. Now, I would like to thank my guide and my co-guide for preparing this slide. I am very much thankful to him. Next, come to the topic. My today's topic is the antifungal evaluation of the plant Clenodendron inermi, which is the scientific name is gentian, and leaf extract. Next, come to the topic. This is the abstract, as you've seen. No need to explain the abstract. First of all, introduction. This plant is commonly known as the glory blower leaf. It is local to the Odisha region. I have collected the plant in various regions of Odisha. It's just like Jaspur and Khordha district. So from two places I collected this plant. This plant is traditionally used as the remnant or interremnant fever or any skin disease. This fever is in this fever the temperature fluctuation occurs. That is increasing 2 degree or 3 degree, but not come to the normal temperature. But in case of interremnant fever, the temperature rises to the simultaneously rises to the upper limit to 2 degree, 3 degree, but it comes to the minimum range or lower than that. Then it is also used as antioxidant, analgesic, or anti-inflammatory. This plant is native to the Southeast Asia, Australia, and Pacific Iceland. This compound, the phytoconstituent present in this plant are flavonoid, alkaloid, triterpenoid, steroid, and tannins. As you see, this is the plant picture. Let's come to the next slide. The objective. As you know, the pharmaceutical market. In the pharmaceutical market, there are various of antifungal agents. But when we take the antifungal agent, after some day, it will resist in our body because of the many protein change in the fungal strain, or it is the drawback of this antifungal agent that it resists in our body. As you know, the pharmaceutical phytochemicals are having the more therapeutic effect and less side effect. So it is the safest drug nowadays. And nowadays, more and more people are leaving the allopathic medicine and come to the pharmaceutical medicine or the Ayurvedic formulation. Next, the plants. There are many scientists are research on the plants which have the antifungal activity, and the plant product which have the antifungal activity. But I want to know if this plant having the antibiotic activity or not. So I have the present study deals with the antifungal activity of the virus extract. I have taken virus extract of these leaves of the plant of the leaves. Next, come to the material method. Before going to my research work, I have to do some literature survey, because from the literature I come to know about the what is the work is done on this plant, or what is the research work on what was done on this plant. The plant material is the plant material used is Clerodendron inermi leaves. Then next one is preparation of extract. I have followed the method the successive solvent extraction by succinylate apparatus. First of all, I started with petroleum method 68. Then same plant or same material was extracted in chloroform, and simultaneously after that we started in the methanol. These are the solvents which are used for the extraction process. After extraction, I have done the phytochemical study. In the phytochemical study, I have done some phytochemical screening. That is the phytochemical test to identify if the constituent are present in the extract or not. Then next one is antifungal activity, evaluation of antifungal activity. For the antifungal activity, I have used the nutrient agar medium. For the nutrient agar medium was sterilized by 121 degree for 15 minutes. Then the fungal this extract was tested against these are the fungal strains which are collected from the gene bank. The antifungal activity of the PET ether extract, methanol extract, or chloroform extract were determined by the agar well diffusion method. The agar well diffusion method and the yeast-like fungi. These are the fungi which cannot be determined by the agar well diffusion method. So we have to do the poison food method for the mycelial fungi. Next one for the result discussion. These are the characteristics of the extract. The PET ether extract was black color, greasy, and the yield is 2.5 feet. Then chloroform extract, that is color is black, consistency is sticky, and yield is 2.18. Methanol black, color is a black, consistency is greasy or non-sticky, non-greasy or non-sticky. We get the methanol extract having the highest yield. So the yield value is 12.45. And as the yield is maximum in methanol extract, so more and more constituents are soluble in methanol. Next one, these are the phytochemical tests. We have done for the general test for alkaloid, glycoside, anthraquinone glycoside, cardiac glycoside, steroid, saponin, tannin, and triterpenoid, protein, flavonoid, and amino acid. So these are the positive sign indicates the presence of the phytoconstituent, and the negative sign indicates the absence of the phytoconstituent. Next, this is the antifungal evaluation. In the agar well diffusion method, for the which I've done for the yeast-like fungi, we determine the zone of inhibition. As the zone of inhibition is maximum, so this extract shows the maximum antifungal activity against the specific microorganism. And for the mycelial fungi, we have done the poison food method. In the poison food method, we have to study the zone of inhibition and the zone of restriction. If the zone of restriction is minimum, so this extract has higher potency for the antifungal agent, and the zone of restriction is maximum, then it produces less therapeutic activity or less potency for the fungal strain. This is the organism, and we have taken the PET ether extract, chloroform extract, methanol extract, and the RA means reference antibiotic. A reference antibiotic we have taken for the comparison study between the extract and the antibiotic. The reference antibiotic, in the reference antibiotic, we have taken the fluconazole and clotrimazole. The amount of the antibiotic agent is 1.56 mg. And the P means PET ether extract, CH means chemical chloroform extract, MT means methanol extract. In agar well diffusion method, first of all, we have taken the agar medium and prepared the agar medium. And next one, the mycelial fungi, the yeast-like fungi, fungal strain are spread over the agar plate, then inoculate for the growth. Then we pour 2 to 3 bore or with the help of cork borer. Then we have taken our extract in this holes. So this method is called as agar well diffusion method. As the if the extract having some antifungal activity, then it inhibits the it diffuses to the agar media and inhibits the microbial growth in a circular manner. So we have to use this method is used to in the scale we have to use the radius or the diameter of the circle. In the mycelial fungi, same method, but it's opposite. That we have spread over the agar medium. We spread our extract and then pour in the well. We have taken the microorganism. So if the microorganism grows in circular manner, so the circular radius of the circle is maximum. Then we have said that this method, this extract was not. We have not this extract having some not having some antifungal activity. So basically, in the agar diffusion method for the yeast-like fungal strain, we have calculated the zone of inhibition. That is, the zone of inhibition is maximum. Then it has more potency to the antifungal agent, more potential antifungal agent, or zone of inhibition is minimum. That produces the less potency antifungal agent. In the mycelial fungi, in the poison food method, zone of restriction is maximum. Then it produces less potency and extract. And in the zone of restriction is minimum. Then it produces. Then we confirm that zone of that potency is maximum. Next one, we have conclude this session that we have got in this table. I have to just one thing I have to show that Candida albicans in the zone of restriction, zone of inhibition, yeast-like fungi, Candida albicans. In the Candida albicans fungi, the chloroform extract value is 16.74, which coincides the reference antibiotic 21.6 value, that is fluconazole. And in the zone of restriction, the methanol extract was coincides with the reference antibiotic, that is T ribosome, in the reference antibiotic 12.46, that is clotrimazole. So we can conclude that the methanol extract and the chloroform extract having some antifungal activity, which antifungal activity with the zone of inhibition or zone of restriction can be coincides with the reference standard. So various fungal strains were tested, and we have all the in the previous table we have studied that all the extract of the plant having some less or more inhibitory activity than the against the tested microorganism, from which the chloroform extract exhibits highest antifungal activity against yeast-like fungi. However, the methanol extract have more active agent against the mycelial fungi. Then the chloroform extract showed the highest activity, which is comparable with the standard drug is fluconazole. Therefore, we can conclude that the chlorindrol enemy shows promise for the development of potent antifungal agent evident from the present investigation. Then these are the references. Then thank you. I have concluded my seminar. That's all.
Thank you, Saubhagya. So basically, you have selected the plant drug, and can you brief once again as a query that how did you take out the active constituent from that plant drug for which is basically responsible for this antifungal activity?
Sir,
and what was the name?
This study is this much. Then further study we have to isolate the specific compound which are having some antifungal activity. That is the further study, future study.
Okay, so at this time you have not isolated the active constituent. At this time you are taking it as a whole extract of the plant.
Plant extract. Yes, sir. Specific compound for the further study in future. We will study the specific compound of the drug.
Okay, and another question is that how did you decide that which antifungal drug you should take as the standard against this test isolate or test medium?
Sir, we have taken that drug which have broad spectrum antibacterial in the market, broad spectrum antifungal activity. That is inhibit maximum species of fungal. That is the fluconazole and clotrimazole. These have broad spectrum antifungal activity. So we have taken this two drug.
Thank you, Saubhagya.
Thank you, sir. Sir, any more question?
So can we move on to the next candidate? Who is the next candidate? Please unmute and let us know. Yasmin Banu. Do we have Yasmin Banu? Okay, Reena Singh.
Yes, sir.
Okay, please share your screen and then you can go ahead with your presentation.
Okay, sir. Thank you. So please allow me share the screen.
Dr. Rajiv, can we make her the co-host? Dr. Rajiv. Dr. Rajiv, please make Reena as co-host.
Yes, made.
Okay, okay. Thank you, sir. Okay, Reena, now you can share.
Okay, sir. Good afternoon, sir. Myself, Reena, PhD scholar, GLU University. Now present the presentation of 2,4-disubstituted benzodiazepine. So microwave assisted synthesis of newer 2,4-disubstituted 1,5-benzodiazepine of biological evaluation. So now come to the introduction, synthesis and pharmacology of 1,5-benzodiazepine. Introduction, 1,5-benzodiazepine are bicyclic compound with 2 nitrogen atom at 1 and 5 position in a 7-membered ring fused to a benzene ring. And then the core structure of benzodiazepine are labels denote location of side chain which give different benzodiazepine their unique properties. A benzodiazepine, sometimes colloquially benzo, often abbreviated BZD, is a psychoactive drug whose core chemical structure is the fusion of a benzene ring and a diazepine ring. And the first benzodiazepine, chlordiazepoxide, Librium, was discovered accidentally by Leo Steinweg in 1955 and made available in 1960 by Hoffman La Roche, which has
also marketed diazepine Valium since 1963. So this is the 3D structure of 1,5-benzodiazepine and now. So 1,5 position attach a nitrogen. 2, 3, 4, 2 and 4 containing the nitrogen atom and substituted a benzodiazepine ring. And now next slide. So continue the introduction. Benzodiazepine enhance the effect of neurotransmitter GABA, resulting in the sedative, hypnotic, sleep-inducing, anxiolytic, anti-anxiety, anticonvulsant, and muscle relaxant properties, also seen in applied pharmacology of high doses of many shorter-acting benzodiazepine, are amnesic dissociative actions. The properties make benzodiazepine useful in treating anxiety, insomnia, agitation, seizure, muscle spasm, alcohol withdrawal, and as a pre-medication for medical or dental procedures. And now come to the next slide. Some of the literature review of 1,4,2,4 disubstituted benzodiazepine. So this is the 2,4 benzodiazepine. So these are the some searches from articles. These are the 1,5 benzodiazepine part. This is the references covered by various articles. And now come to the main part of the this paper scheme. So substituted benzaldehyde and ketone treated with ethanolic sodium hydroxide, we will get chalcones, and chalcone treated with orthophenylene diamine, various orthophenylene disubstituted benzodiazepine. So we will get 2,4 disubstituted benzodiazepine. So we have prepared the 19 derivative of 2,4 disubstituted benzodiazepine. And now come to the next slide. So we can conclude that data published on benzodiazepine synthesis, one of the most abundant benzodiazepine, suggests that 1,5-benzodiazepine has potential to be useful therapeutic agent. The synthesis of 1,5-benzodiazepine were accomplished as present in scheme. Substituted benzaldehyde on the reaction, the substituted ketone in the presence of ethanolic sodium hydroxide yielding corresponding chalcones 1A to 19A. The later was converted to desired 2,4 disubstituted 1,5-benzodiazepine 1B to 19B from spectral, physicochemical, and elemental analysis. So in the end, we can conclude that, however, further the studies are needed to validate future application. These are the references. Thank you, sir.
Thank you, Reena, for taking us to another side of the presentations towards microwave synthesis, microwave assisted synthesis. And I would like to know that what future do you see from the 1,5-benzodiazepine synthesis and its molecules, because it has been used since long in the form of Librium, as you told. So BZD has been in use since very, very long time in the allopathic system of medicine. So what future or what newness now you find in this topic?
Yes, sir. 1,5-benzodiazepine, like clobazam. Clobazam drug is 1,5-benzodiazepine used in anti-epileptic drugs. So future we can use other type of biological activities like enthelmintic activities. Some compounds give enthelmintic activities 1,5-benzodiazepine. But sedative hypnotic most powerful drug diazepam 2,4 benzodiazepine. But 1,5-benzodiazepine used in future prospective.
Thank you, Reena.
Okay, sir. Thank you. Thank you, judges.
Yeah, Rina, can I ask a question, please?
Yes, sir.
Hi, how are you?
Fine, sir.
So can you name any marketed preparation based upon this molecule? Is there any marketed formulation which is using benzodiazepine? Can you name any therapeutic category? Yeah.
Okay, sir. Compose, sir. Compose a diazepam drug. This is benzodiazepine. Compose, because I have done the sedative hypnotic activity by this benzodiazepine injection.
Okay. Okay. Thank you. Thank you, Rina. Over to Dr. Daya.
Okay, sir. Thank you.
Thank you. Thank you.
Thank you, sir.
Thank you, Dr. Banerjee.
Thank you,
So
sir.
can we move on to the next person, please, for the presentation? Is Yasmin available? Once again, I am asking, is Yasmin available? Okay, we have waited sufficiently for Yasmin. So can we move on to Vanessa? Vanessa, now it's your turn. Are you available? Vanessa.
Miss Vanessa Richardson James.
Okay, is Mr. Vivek Pawar available?
Yes, sir.
Okay, then Vivek, it's your turn. We are moving on to your candidature, and you can go on and share your scientific presentation.
Sure, sir. Thank you.
Dr. Rajiv, please make him the co-host. Okay, Vivek, now you are the co-host and you can share your screen. Vivek, your screen is shared. You can start your presentation.
Okay, sir. Good afternoon, all. My name is Vivek Pawar. I am PhD research scholar at School of Pharmaceutical Sciences, Sholini University, Solan. So the current study that I am going to present is based on design, synthesis, and evaluation of kinase inhibitors as anti-cancer agents. So as we all know, kinase is cancer is a multifactorial chronic disease, and that is, the cancer can either be invasive or malignant, and is one of the most plausible antecedent of human mortalities worldwide. It is a term to describe a huge cluster of disease that involves uncontrolled cell proliferation, and the progression of cancer may include genetic changes like gene mutations or epigenetic factors. So as we can see from the stats here, there are a number of cases that have been reported for cancer every year, as well as the mortality data. Among all the cancers, the new cases for breast cancer and lung cancer are maximum, whereas the mortality rate for lung cancer is maximum. So we have different kinases that are associated with different types of cancers that may include EGFR, mTOR, or HER2. If we talk about lung cancer, so EGFR is one of the most overexpressed cell surface receptor that is overexpressed in lung cancer, whereas mTOR is a cytosolic receptor and exists in two complexes, mTOR C1 and C2, with different modes of activity in terms of physiology, as well as deregulated or upregulated protein expressions may lead to cancer. So these are this is a generalized flow chart for different upstream and downstream regulators that are overexpressed in cancer. So if we talk about specifically tyrosine kinases, so these are basically the enzymes that catalyzes the phosphorylation of tyrosine residues in target proteins. So here we have this EGFR protein. So overexpression of these hormones, growth factors, or cytokines are involved in activation of this IRS1 PI3K regulated PI3K AKT pathway that in turn is activating the mTOR complex C1 and mTOR complex C2, which is again regulated with angiogenic events, lipid synthesis, adipose tissues, or we can say inhibiting autophagy and all these things. So these overexpressed kinases are therefore involved in different cancers. So what we have done, we have designed a library of compounds based on the already FDA approved drugs, that is quinazoline based like gefitinib, erlotinib, afatinib, lapatinib, pentitinib, and decomitinib. So these are EGFR inhibitors based on quinazoline moiety. So keeping this moiety or these important pharmacophoric features in mind, we have substituted this NH position with different linkers, and then these linkers are further substituted with different amines. So for the study, first we have designed the series of compounds. So for the designing process, Discovery Studio was used for sketching of compounds. Pharmacophore mapping was done using PalmMapper, and it is also a tool for the target identification of the design library. Then protein data bank was used to get the 3D crystal structure of the proteins, and the protein obtained was prepared using protein preparation wizard of Schrodinger software. Next, glide XV mode of the Schrodinger was used for docking analysis, that is a ligand and protein docking, and time was used for MMGBSA calculations, assessment for MD, and quicker from ADME calculations to predict the pharmacokinetics of the designed compounds. So this is the pharmacophoric data, and on the basis of this we have chosen these compounds, and then we did the molecular dynamics. So this is the final result of the compounds. Like these are the different docking scores of the best library. So we have docked all the 156 compounds, and among this the compounds with 1,5 linker were best potent. Aliphatic chain of 5 carbon was maximally active. So the docking studies were carried out, and this was the binding score. MMGBSA was also done, and the key interactions were also reported. So again, Lipinski rule of 5 was also taken into the consideration. Next, moving to the chemical synthesis and characterization of the sketched compounds. So for the synthesis purpose, commercially available reagents and solvents were used, and the reaction was optimized continuously using pre-coated TLC plates throughout the reaction. And after completion of the reaction, the final product obtained after workup, or we can say crystallization, were further purified using column chromatography and were observed under UV chamber. All the synthesized compounds have been characterized with their melting points, that is physicochemical properties, as well as spectrometric analysis, that is IR, mass, and NMR spectroscopy. So this is a generalized scheme for the synthesis of these quinazolinone derivatives. So what we have done, we have taken anthranilic acid and treated it with formamide for 4 hours at 140 degrees Celsius. That leads to the formation of quinazolinone. The product formed was passed through various purification processes. Like first the product was worked up using different solvents, and then the product obtained was further purified with the help of column chromatography. So in this we have used the solvent ratio of 9.8 is to 0.2 chloroform is to methanol for the isolation of this compound. Further, the compound was treated with 1,5-dibromo dibromopentane, that is dibromo linker, in the presence of acetone and K2CO3 as green catalyst, and the reaction was carried out for 12 to 16 hours. The product formed was again purified using column chromatography, and then different amino, aliphatic or aromatic amines were substituted at this position, leading to the synthesis of final 10 products. So this is a representative morpholino based compound that is used as a representative structure. So the final yield of the compound was 78%. Its molecular formula was C17H23 and 302 with a molecular weight of 301.39. The product was obtained in the as crystalline pale yellow solid, and the RF value was found to be 0.48 in methanol chloroform 0.5. The melting point was 180 degrees Celsius, whereas mass spectra done for the main peak was at 302. The IR for different functional group was done with different peaks, like the 6.1657 at 1657 we observed the peak for carbonyl carbon, and for C double bond N 1606, and likewise we obtained different peaks for different functional groups in the final structure. Next, for the further identification of the compound, proton NMR and carbon NMR were done. So different, as we can see in the structure, we have different aliphatic regions as well as aromatic regions. So aromatic region, for the aromatic regions we are having a peak around 6 to 8, whereas for aliphatic regions we are having peaks around 3 to 6 or 3 to 5. Likewise, for carbon NMR, we have different peaks for different carbon atoms. After synthesis and characterization of the compounds were. Hello, am I audible?
Yes, you are
Some
audible.
background noise is coming.
No, this is not from my side, sir.
Yeah, not from your side, but I think some participant, है ना?
Yes, sir. Next, after synthesis and characterization of the compounds, we further went for in vitro evaluation. So the in vitro evaluation of the compounds was done using HCT116 cell line, that is for colon cancer cell line. So the compounds were evaluated in vitro at different concentrations, and the results were compared to the standards. So the one of the compound was showing more potency than the standard, that was morpholino substituted derivative. So this compound in future we can use as a lead, or we can go for in vivo activities to further validate the study. So that is all. Thank you.
Thank you, Vivek. The study was nice, but the only thing from my side is that how do you relate your prepared compounds with the initial thing which you told about the tyrosine kinase inhibitors? Like, do you find that the structure is similar, or how do you relate both the things, like the initial concept with the what you have prepared now?
Yes, sir. Yes, sir. So in this study, what we have done is we have kept the pharmacophoric features in mind, basic pharmacophoric features like carbonyl carbon, or basic moiety with nitrogen in that cycle, that is quinazoline. Or we have substituted different alkyl chains, like we have done a kind of SAR study, SAR study, like what is the effect of adding 1,2-dibromo, 1,3-1,4-1,5, and then we have substituted it with different amines, that is aliphatic aromatic. One more thing what we have done in this is like FDA approved drugs, there are different substitutions at this position or this position, but this position was not substituted as such. So we have tried to explore the medicinal chemistry of this position in this study.
Okay.
Yes.
Thank you, Vivek.
Thank you, sir.
So after Vivek, can we move on to the next candidate? The next person for the scientific presentation is Tarun Chaudhary. Do we have Tarun here?
Tarun, are you there? Tarun?
Tarun, are you available?
Dr. Daya, I
So,
can't see.
yeah, it looks like Tarun is absent. Yeah.
Yes, yes,
I am
yes.
also marking it for him.
Yes.
Okay, so do we have MD Jishan?
Mr. Zishan, are you there?
Mr. Jishan, are you there? Looks like he is also
Looks
absent.
like he's absent, sir. He's not there, sir.
Yeah. Let's mark it in our
Yeah.
records. So,
Yes.
so once again, let's give a final call to the absentees. Other 1, 2, 3, 4 persons have already presented their work, and the persons who are absent now in our list, one is Miss Yasmin Banu. Miss Yasmin Banu. Another person is Vanessa, then Tarun, and then Jishan. So these 4 persons, yeah, Vanessa, sir, I am here. Okay, then you unmute yourself and come to the presentation side. Yeah, unmute yourself.
Yeah, Vanessa, you can share your presentation, please. Vanessa, can you hear us?
I think I'm not able to share screen, I think.
Dr. Rajiv, please make her the co-host.
Okay.
Yeah, Vanessa, now please try once more.
Okay.
Done.
Yes, 1 second. Is it visible?
Yes, it's coming. You can put it on full screen mode, Vanessa. Yes, please start.
Okay, so my name is Vanessa James. I am pursuing my PhD from Gujarat Technological University. My topic is estimation of metallic impurities and microbial evaluation in selected herbal formulations. So these are the contents of my presentation. So we all know herbal formulations are widely consumed throughout the world, and after COVID-19, it has been much popular. So the risk assessment of herbal formulation is much required. Due to the origin and nature of plants, contamination with toxic heavy metals can cause significant health risk. So it can be compromised by variety of conditions such as inappropriate manufacturing, packaging, and storage conditions. So trace elements such as iron, manganese, zinc, and copper are essential elements so that are required by the body. And heavy metals like cadmium, arsenic, lead, and mercury are very toxic, and it may cause cancer or like certain diabetes mellitus and certain terminal diseases. And heavy metals contamination could be caused due to various reasons such as fertilizers and pesticides, waste disposal, industrial process like mining, etc. So here we have selected herbal formulations which are having certain COVID-19 like against activity, which are reported and having good immunomodulator activity were selected, and that were single dosage forms. And among the main safety risk related to herbal formulation is also microbial contamination. So that can be due to like harvesting, drying, preserving, manufacturing, etc. So it is also essential to evaluate herbal medicines because this can cause certain gastroenteritis, diarrhea, poisoning, etc. This is the literature review. So here we have performed ICP-MS for determination of metallic impurities along with microwave digestion. Microbial analysis is performed as per Ayurvedic pharmacopoeia. We have collected certain literature and accordingly we have selected the technique, and pharmacopoeia is like our inherent, that is Ayurvedic pharmacopoeia we have considered. And aim and objective is to estimate heavy metals and evaluate microbial contamination in herbal formulations. Objectives are to estimate mainly heavy metals, which are like class 1 elements, which are like really toxic for human, and that should be controlled in the herbal formulations. So that is determined by ICP-MS with microwave assisted digestion. And evaluation of microbial contamination for TPC, total plate count, yeast and mold count, Escherichia coli, Pseudomonas aeruginosa, Salmonella, Staphylococcus aureus, that is performed as per Ayurvedic pharmacopoeia. And to compare heavy metal and microbial contamination results with the Ayurvedic pharmacopoeia standards. Materials and methods. So sample selection was performed, like such as tablets, churna, and bhasma were selected. For tablets, we have considered 4 materials, that is amlaki, ashwagandha, giloy, and tulsi. These tablets with their 3 branded formulations were selected. And we all know that these are all immunomodulatory along with certain other benefits. And for churna, we have selected Brahmi churna, Jethimat churna, and Shatavari churna. This were also selected with the popular brands were also selected. And this were also having the immunomodulator activity along with other benefits. So for bhasma, that is Loha bhasma, Tamra bhasma, and Yashat bhasma, these are all single unit bhasmas were selected for identification of metallic impurities and microbial contamination. So Loha bhasma is the preparation prepared from iron, Tamra is prepared from copper, and Yashat prepared from zinc. These are also having immunity booster activity. So sample is prepared on the basis of microwave assisted sample digestion. So acid digestion is done for sample, which is a 4:1 ratio, that is 4 ml nitric acid along with 1 ml hydrogen peroxide. So 0.5 gram of sample is taken. Along with this, acid composition is done, which will further digest in the microwave. And each digestion series will contain reagent blank. The final, when it is digested, final sample preparation when it is digested, it will again be diluted with 25 ml of distilled water or heavy metal free water. So these are the digestion parameters. It is performed in 2 stages. So first we have like performing for power, and power is mostly common for both of them, but RAM to temperature and hold time is it varies, but it will all be finished in 1 hour only. So all the microwave assisted digestion will take 1 hour. Apart from the hot water, like hot other digestion procedure, it is very simple, and it also reduces the amount of volatile impurities separation, because it gets evaporated. So these are the instrument details. ICP-MS, make is Agilent, model is 7700X. So mass spectrometer is quadrupole, and gas used is helium. Preparation of calibration solution, 0.5 ppm of solution is prepared by using certified reference material of single element standards are taken, and the calibration curve is prepared and linearity is taken. These are the standard curves for heavy metals: arsenic, cadmium, mercury, and lead. Result and discussion, these are the quantitation reports of all the samples. I directly skip to the results. These are all the quantitation reports which we have obtained. Okay, so this is the results. One second. So for heavy metals in ashwagandha, that is ashwagandha, in 1 sample we are getting arsenic as well as mercury. And in 2 samples we are getting mercury, as well as 1 sample we also contain arsenic. For cadmium, all of the samples we are getting are below LOQ limits, and lead is also below LOQ limits. Only the content which is higher is in mercury. Mercury is found in 5 samples of tablet formulations. For churna, lead is present in 2 of the samples, and arsenic is mercury in one sample. So here only SC2 sample is passes all the limits for the heavy metals apart from cadmium. For Basma, most of all the formulations doesn't pass the limit, and it is obtained in very high proportion. As we can see, lead is obtained in really very high proportion, which can be toxic. For if further we are taking in high amounts, or like it can build up to bioaccumulative potential, so that can be harmful. So here we can also see arsenic is also present, but apart from except from two metals, cadmium is still within the limits. Only it is present in one of the samples. Otherwise, all the samples are free of cadmium, but lead, arsenic, and mercury still poses some risk. And mostly lead is posing more risk compared to other metals. This is the microbial testing that is performed by Ayurvedic pharmacopoeia. The tests which are included for determination are TPC, YMC, E. coli, Staphylococcus aureus, Salmonella, and Pseudomonas aeruginosa. Media used are as per the Ayurvedic pharmacopoeia for each of the tests. Media is prepared we have procured from high media laboratories. So they are just sterilized in autoclave and used for the inoculation purpose. So samples were also taken, and they are pre-enrichment is done with the help of like a phosphate buffer or anything, and then they are further used for the determination of microbial content. An incubation period is done for mostly 1 to 5 days. These are the total plate count data for sample AM1, AM2, and AM3 tablets, 81, 82, and 83 tablets, GT1, GT2, and GT3 tablets, TT1, TT2, and TT3 tablets. This is for SC1, SC2, and SC3 churna. Again, BC1, BC2, and BC3 churna, and JC1, JC2, and JC3 churna. This is for Basmas. All the limits are within the limits. As you can see, it was all obtained in very less proportion. Afterwards, in further dilution, we can obtain that there were very less colonies, and so it passes the limit. Yeast and mold count also all were found to be absent, or very few colonies were obtained. These are all the for yeast and mold count. Pseudomonas aeruginosa. These organisms were also found to be absent for all the samples. Staphylococcus aureus were also absent for tablets, churna, and basma. E. coli also found to be absent. Salmonella is also absent. So here in results, we can see microbial contamination was, it passes the, it complies with the test, and few colonies for TPC and YMC are obtained, which are also within the limits for churna as well as basma. So major concern focus is on the herbal formulations is for heavy metal contamination, because they are still obtained in the final products. So methods can be taken for like to control all the heavy metals or like any elemental impurities, trace metals. So for that, further methods could be taken, such as like monitoring on quality based, like for each and every step, such monitoring measures can be taken. And we can see like from overall, we have taken 30 samples, but 15 samples, that is 50%, shows the presence of one or more heavy metal. Mainly basmas, which were used for prevention and treatment of certain diseases like terminal disease, like cancer and everything. So that is considered as threat to human health unless good scientific evidence proves their safety and efficacy. So like basmas, which are used so widely right now for even like small formulations along with like single or multi-dosage. So that could be evaluated, and we can already see that lead is already present in much more higher quantities then it should be present. So we can like take measures for our future, like for long-term studies, we can do that, and toxicological studies can prove that it should be controlled. These are the references. Thank you.
Thank you, Vanessa, for taking the presentations towards the Ayurveda side. And the study is really good, seeing the estimation of metallic impurities in various herbal preparations, as well as the load of microbial contamination also. So, but my point is related to do we still see, because in India also that these metals, they are contaminants, because also Ayurveda philosophy is that these metals also, even if we term them heavy metals, when present in minute quantities, Ayurveda science has been using these metals in various forms, in form of bhasmas and ras preparations, as because of their healing properties. So I still have like a concern that maybe that we have not understood the science well, and that is the reason like we are following what has been published in the Western journals, or what the other parts of the world they are saying about the Ayurveda, but maybe it is not the case. So
No, actually, they are saying that these are present in form of like not toxic. They are converted in the non-toxic forms. They are proving that. So my concern is that all companies are not using this kind of scenario. Like proper formulation is prepared or not, it's still a concern. So certain companies are there which are preparing in a good form, like how bhasmas should be prepared, because we all know bhasmas formulation are really complicated, but it is widely used for terminal disease and they are actually effective. So if we move towards that scenario and actually like concerned about the standardization, then it would be really good for our future aspects. I think in still in that way. So this is generally like awareness, because we need to still study on these aspects.
yes, Vanessa, I agree with your statements, but at the same time, like we understand that arsenic has been used in allopathy, homeopathy, Ayurveda in different concentrations, in different dilutions, in different kind of formulations for its healing properties. In the same way, mercury has been used. Like now, people are saying that mercury is using, or mercury is having like antimicrobial properties, and it has been used for ages in our Ayurveda. So maybe that. Yes, you are right that we have to make more awareness, and we have to use more standardized protocols for preparing our formulations of Ayurveda. But at the same time, like we also have to diffuse the this kind of situation, that they are not always contaminants, or they are not always toxic, but at the same time, sometimes they are also used in a balanced proportions, in a balanced protocol manufacturing method for the healing properties as well.
Yes, sure, because they are actually good. They have proved good scientific evidence that they are actually good for cancers, as well as like it's right now Ayurvedic practitioners are preferring over like this kind of allopathic are having certain side effects, but in Ayurveda they are not having certain side effects. So if we move towards that scenario, it's actually good, but we don't know about the bioaccumulative potential, like actually they are harmful or they are not causing like in long term they are not causing any side effects.
Thank you, Vanessa.
Thank you.
So once again, we are giving a call to Yasmin Banu, if she is available. Yasmin Banu. So this is the final call.
She's trying, but I'm not seeing her. I even called her. I don't know.
No problem, sir. If she is absent, like yeah.
She is not in the list, but she is messaging that link open nahi ho raha, like that.
Okay. And Tarun Chaudhary and Zishan.
One minute, I am calling again. Once again. Who three are Tarun,
Tarun, Jishan, Tarun and Jishan, they are also left out.
Hello. Hello.
Yes, sir.
So
Hanji,
all
sir.
presentations are over?
Yes, sir. All presentations are over.
So Jasmine, so one candidate I called, they are saying yet his turn is not there according to time given.
No, but it has already been told to all the presenters that they have to be present during the entire session.
Yeah, I answered the same.
Yeah.
At
Because
2:30, this Tarun is there. So it is yet 2:30 is there.
no, Tarun Chaudhary is absent, Jishan is absent, and Yasmin is absent. These three
No,
persons are absent.
Tarun Chaudhary presentation is at 2:30. Whether 2:30 is over?
Time? No, no, 2:30 is not over.
So at Jisan, who? Okay, Jisan.
But it is written that everybody has to be present
Yeah,
throughout the
I
session.
mentioned the same.
Yes.
You can evaluate the score and send to me via WhatsApp.
Okay, sir.
Okay, sir.
Okay.
Dr. Rajiv, I have sent you the scoring sheet.
Okay.
Dr. Daya, I WhatsApp you the marks. Dr. Rajiv. Dr. Daya, I'm just taking a 5 minutes break.
Dr. Banerjee.
Yes, sir.
Sir, I feel that like 12-13%, it is not justifiable to be waiting for another 1 or 2% who is already absent from the session.
Absolutely.
Because
Sir,
this walkover should be given to those candidates who are, sir, available from the last 2 hours with us, 1 hour, more than 1 hour.
I completely agree, sir. I completely agree.
Yeah.
Okay, no problem.
Yeah, because sir, in any games, in any participation, in any competition, because those who are available, they are given the priority, and these candidates, they are available with us during the entire session.
Absolutely, sir. Absolutely.
Yeah.
Actually, what happens now in one time, because these are registered candidates, and sometimes they claim that at our time we were there. We joined at that time, so that's why I was waiting. But we are at positive side. We have made the announcement that they should be present in all presentations. It was on the sheet.
Yes,
We
yes, sir.
can't claim like that.
Sir, because this is like a competition, this is arranged for them, so they have to be present.
Yeah, it is a learning
Okay.
platform also.
Yeah.
Okay, it is 2:08. So for 5 minutes more, and then we you can leave. Last
Sure, sir,
5.
sure.
2:13 you can leave. कहाँ हो आप? मुझे तो नहीं दिख रहे। Dr. Daya, one candidate is there, I think so.
Okay.
Good afternoon, sir.
Good afternoon.
You can start your presentation.
Okay, sir.
Your co-host.
Okay, sir.
And Dr. Daya, another presenter is also there.
Okay.
Okay. Yasmin.
Good afternoon, sir. My voice is audible, sir?
Yes, sir, your voice is audible. Please go ahead.
Okay, sir. My topic is today analgesic, anti-inflammatory activity and molecular docking study of 1,3,4-oxadiazole analog. First of all, I will introduce about oxadiazole moiety. Oxadiazole derivative offer a wide range of therapeutic possibilities because of their ability to bind to a variety of receptors and enzymes in the body. Due to the presence of this linkage and
please, Dr. Saurabh here. You can straight away, go to your objectives and your research methodology and direction because already we are running out of time, please.
Okay, sir. Okay, sir.
Yeah.
Yeah, Dr. Banerjee, that would be very good.
Yeah.
First of all, after the introduction of oxadiazole, I have done the docking. What is that docking and molecular docking, and what are the steps of the docking? What type of the material we have used in docking? That I am going to explain now. But first of all, molecular docking, it is a technique that places a small molecule ligand in the binding site of its macromolecule target or a receptor and estimate its binding affinity. Docking is an attempt to find the best matching between the two molecules.
Tarun, please, sorry to interrupt. Please come to your objectives, please. Please come to your objectives. Yes.
First of all, we have to select protein for the docking. Selection of protein. The literature study shows that the following proteins are in close association. These are the PDBs, which are used for the COX-1 and COX-2. I have selected the PDB for the COX-1 is 6Y3C and for COX-2 5IKR. How I have select this PDB? Selection for that PDB depend upon the organism. If you are working with a human or X-ray crystallography, lower resolution or inhibitory ligands or co-crystal ligands should be present in the selected PDB. We have to compare the IC50 and EC50. On the basis of that, I have selected the PDB. Now next is the preparation of protein. First of all, we have to prepare the protein for the docking. All the protein used for the docking were prepared using protein preparation. The preparation involved the assignment of the hydrogen bond order, addition of hydrogen, optimization, minimization of the protein, and deletion of water. Determination of highly potential binding site of the ligand on the protein was carried out by using a site map tool analysis. Docking of all the ligand were carried out using a glide ligand docking module. Excel precision were used for the visualization of the glide score. Now next, ligand preparation. After the preparation of protein, we have to prepare the ligand. The structure of the ligand was generated in the CDX format using the tool CamDraw Ultra version 8. These ligands were then converted to the mole format and subjected to the liquid module 2 of Maestro in the Schrodinger Shoot 2013. This software I have used for the molecular docking. They were converted 2D to 3D structure by including stereochemical ionization, tautomeric variation, as well as the energy minimization, and optimized for their geometry. The bond order of these ligands were fixed and the charge group were neutral. Next is receptor grade generation. What is that? In that we have to find out the active site of the receptor where the protein have to be bind or where the ligand has to be bind. Next step is validation of the docking program. The accuracy of the docking procedure was determined by finding how closely the lowest energy pose of the co-crystallized ligand predicted by the object scoring function, glide score, resemble an experimental binding mode as determined by X-ray crystallography. Acceleration gliding docking procedure was validated by removing co-crystal ligand from the binding site of the protein and redock the ligand with the binding site. Now, come to the result and discussion. Ligand code, I have used the standard drug indomethacin. In that drug, a docking score with the COX-1 is minus 1.84 and docking score with the COX-2 by this PDB, the score is minus 10.740. I have taken the drugs or a compound which have a high docking score. These are the 10 ligands which have high docking score. Now next is conclusion. Protein ligand interaction plays a significant role in the structure-based drug designing. In the present work, we have taken COX-2, 5IKR, that is the PDB, which is essential target for the novel anti-inflammatory drug design, and COX-1, 6Y3C. When the receptor were docked with a standard drug indomethacin, the energy value obtained is minus 8 to minus 11. The energy values are negatively greater than a standard. So it can be concluded that the design compound can be potent anti-inflammatory agent than the standard. It can be said that all the newly designed ligands may be safe anti-inflammatory agent than a standard. In future research work, ADME, adsorption, distribution, metabolism, excretion properties of these compounds can be calculated, and we have planned to synthesize these ligands and screen for their in vivo anti-inflammatory activity.
Thank you, Tarun. My question is that what newness do you see in your work and what is its future?
In future research work, sir, I have done first docking of 360, approximately 360 compounds. Out of that, 10 to 15 compounds having a better or high docking score. These 10 or 15 compound I am going to be synthesized in research in future. After synthesis, I have to do that ADME property, and further I will do in vivo anti-inflammatory activity.
Thank you.
Okay, sir.
Do we have the next candidate? Dr. Rajiv?
Yes, Jasmine had joined.
Mr. Zishan.
Zishan or Jasmine? I have.
So we can call their names. Mr. M.D. Jishan.
Not MD Zishan. The one more remaining candidate. I have seen her.
Okay. Miss Yasmin Banu.
Yes, Yasmin, yeah.
Yasmin Banu.
I think she again went out. Okay, you can wind up.
So thank you all the participants who came over and participated in this competition of the annual APP convention, and you shared your very nice scientific presentation. Some have shown their PhD research work, then some other people, they have taken other interesting topics, but all of them have done a very commendable job. Thank you all.
And
Thank
both
you,
the
sir.
judges, you can send the marks of last candidate.
Sure, sir. Thank you.
And all candidates, the valedictory will be at 3:30 p.m. with the same link.
Okay.
Okay.
So
now
we are signing
have,
off for the day for this session.
now we have increased one more award, which will carry a cash of 5,000. Late Professor M.C. Gohel Memorial Award. So there will be 4 awards for the 4, but those are combined award of this group and the second group. Out of 18 presentation minus absent, top 4 will be the APP Memorial Award. Okay.
Okay, sir.
So we'll begin at what time, sir?
3:30.
3:30.
Okay. So should we sign off, sir, this session now?
Okay. Yeah, it's okay.
Okay.
It's
Thank
okay.
you. Thank you.
Hey,
Okay.
thank you very much
Bye-bye.
for
Thank you.
the
Thank
support.
you, sir.
Yeah, Rajiv, sir. Dr. Rajiv. Dr. Rajiv, sir, can you hear me? Hello.
Yes, sir.
Sir,
Yes, sir.
this same link will be for the valedictory, na, sir?
Yeah, it will continue the validity.
Okay, I'm
All
here
will
only, sir. I'm not logging off, na?
Okay, no issue. Hello, Abhinandan.
Hello, sir. Ji.
So this session is now completed. So we have to start at 3:30 p.m.
Okay, sir. I will join at 3:30 sharp.
No, you can remain joined, but now no activity will happen until 3:30 p.m.
Okay, fine, sir. So, sir,
Okay.
can I know the schedule that what will be there at 3:30?
No, it's okay. Your name will be announced, and then you have to speak for 1 minute, keeping your video on about the objective, what you have thought about your work in the Maharashtra.
Okay, sir.
That's all.
Okay, sir. Fine. No worries, sir. I will join sharp at 3:30.
Okay.
Thank you, sir.
Hello, everybody.
Hello, Daya sir.
Hello.
Hello, Dr. Rajiv Daya sir. Are we waiting for somebody, sir?
Yes, sir. I'm compiling the results.
Haji sir.
Yet results are not ready.
Acha,
I'm
acha.
compiling.
Okay, sir. Okay. Okay.
Banerjee sir, good afternoon.
Good afternoon.
Sorry for the delay. I could able to compile up the results. So we can start the virtual valedictory function of APP 11th Annual International Convention. So just I will share the screen and I will change the slide. So Dr. Banerjee, you can confirm whether all things are okay.
Sure, sir. Please share, sir.
1 minute.
Still, it is not visible, sir. Yes, it's coming.
So now
Yes.
I will change the slide and you will confirm whether the slides are changing or not.
Sure.
So I'm putting on presentation mode.
Yes, sir. It is on full screen.
Things are appearing?
They are going, sir. Please move forward. Yes, sir. Yes, sir. It's moving.
Next slide changed?
Next slide. It's moving, sir. It's moving. Yes.
Okay. Then I think we are well off.
Yes, sir.
So before starting that, I want to just change the host again, because if somebody will enter in between, I will be unable to do. What I should do then? I'm the host of the meeting, and if I'm presenting and in between, if somebody enters, I will be unable to do. So
we cannot see the presentations now.
1 minute. Saurabh, Banerjee sir, I'm making you host.
Acha, okay.
I'm making you host, and you can let the people in parallel, whoever are admitting in between.
Okay, sir. Okay, no problem. No problem.
And make me co-host so that I can present.
Okay. Okay. I am making you co-host, sir. Na?
Okay.
Okay. Just a moment. Yes, sir. I have made you co-host, sir. Yes, sir. I have made you co-host.
So
yes, we can
are
see the
you able to-
presentation, sir. Yes, yes. Good to go, sir. Good to go.
Okay, so we can start the presentation here.
Yes.
So I welcome all of you in the virtual award ceremony of APP 11th Annual International Convention and the 5th Indo-Swiss Conference, which is hosted by St. Paul College of Pharmacy, Rangareddy, Telangana, which is a A+ NAC accredited institute. And the occasion is Vijay Divas India, which was yesterday. So we memorize the sacrifice of the soldiers in Indo-Pak War in 1971 on 16th of December. So before moving to the award section, I had made some announcements. So we have modified our Maharashtra state branch.
Okay.
So the vice president of the Maharashtra state branch is promoted to the president of Maharashtra state branch, who is Professor Mrunal K. Sirsar.
Okay, sir. Thank you.
Principal SBSPM's B Pharmacy College, Ambajogai, Bid, Maharashtra. So before I invite Professor Sirsar to say a few words, I want to
Okay, sir.
I want to just announce other branch members.
Okay.
Second is vice president. Vice president is Dr. Ramesh D. Ingol, who is principal at DJPS College of Pharmacy, Patri, Parbhani, Maharashtra. Secretary of the association will be Dr. Abhinandan R. Patil, Associate Professor, D.Y. Patil College of Pharmacy, D.Y. Patil Education Society Institution, Deemed University, Kolhapur, Maharashtra.
The slides are not moving, sir, now.
Thank you, sir.
No, I'm not moving the slides.
Achha,
These
achha,
announcements are verbal.
Okay.
Okay. Thank you for letting me know. Okay, then the joint secretary of the Maharashtra state branch will be Professor Sachin Kumar V. Patil, who is principal at Ashok Rao Mane College of Pharmacy, Vadgaon, Kolhapur, Maharashtra. And executive member of the branch will be Dr. Digambar Ambikar, Associate Professor, Indra College of Pharmacy, Pune, Maharashtra. Now I invite the president of the Maharashtra state branch to share his views, followed by other members if present.
Okay. Thank you, sir.
Please keep it short, half minute each.
Okay. Thank you, sir. Give me the charge for Maharashtra president. Especially thank for Daya sir and team of APP for India and all over world. Thank you for giving the chance for to improve the APP branches and your conferences and good giving the work for together for every executive members in Maharashtra state. For thank you, especially for the APP. Thank you, sir. Daya.
Okay. If any other member of the branch want to share their views, welcome.
Thank you, sir. I'm Dr. Sachin Patil from Ashok Rao Mane College of Pharmacy, Petwargaon, Kolhapur. And I really very much thankful for giving me chance to work in APP as a joint secretary in Maharashtra branch. And surely, as per the discussion with the Raju sir, in the Western Maharashtra, the awareness and the campaign the APP, I have already initiated. I'm discussion with the Raju sir also, and accordingly, definitely here too much scope is there for APP, and all team will improve members and we will take various activities there. Thank you for giving me chance in APP.
Thank you.
Good afternoon, sir. This is Dr. Abhinandan Patil. I am from D.Y. Patil College of Pharmacy, Maharashtra. First of all, I just want to thanks to the APP that they have given an opportunity to be the member of secretary for the Maharashtra as a representative for APP. So basically, we know that a number of like governing bodies are available for the pharmacy council, which are working related to academia, regulatory, hospital, and community pharmacy. But I believe that by using this platform of APP, we can have a good opportunity for the students and the faculties, basically related to the interdisciplinary research. The reason is that many of the pharmacy colleges, if you see, they have like different types of infrastructures available, along with that facilitations which are there, they are at the higher level. So if you try to connect by using this APP, then definitely we can have like upgradation in the studies and even in the research. The reason for that to share this thing is that currently I'm holding 37 patents to my that is my resume, and along with that I have my 2 startups. So in coming days, with the president of Maharashtra, that is Mrunal Sirsat sir, and along with the other members, I will believe that we can have like a good opportunity for all the teaching and students for their development. Thank you.
Okay, sir.
Okay, thank you. I hope Maharashtra branch will grow a lot under your leadership, Professor
Okay,
Mrunal.
sir. Yes, sir.
Coordinate
Yes.
with
Hello,
all
sir.
other. And any other is there?
Yeah, yes, sir. Yes, sir. Hello, sir. This is Dr. Digambar, Associate Professor Indira College of Pharmacy, Pune. And thanks for giving opportunity to work with Maharashtra team. And definitely, we are the team of pharmacy professional, and will connect the people and will work together to grow our field as well as individually, all of us. Thank you, sir. Thank you very much.
Okay. So we have more announcement. I will not able to give chance to everyone, so selective person will speak. Dr. Priyank Kumar, who is Associate Professor at Marshall B. Catcom University, California, USA, is been promoted from Executive Member to the Joint Secretary of APP American International Branch. Dr. Vijay Shankar, who was the speaker of today's session also, is a research scientist at C4 Therapeutic Institute, Watertown, MA, USA, will be our new international head of APP IP DRA division. Actually, we have separated the IP and DRA. DRA division head already we have, and Dr. Shankar will lead the industrial pharmacy division at international level. If Dr. Shankar is there, he can share his views. No. Next is Dr. Ronald Darwin, Professor and Head, Department of Pharmacology, Wells, deemed to be University, Tamil Nadu. He will be our new coordinator for APP Molecular Pharmacology Division. Dr. Ronald, you are here? Okay. Professor BVS Lakshmi.
Sir, I am here.
Yes, Darwin, you can share your views.
Good day to everyone who has joined the APP convention. I'm so happy and delighted to get this offer to be associated with APP as governing the molecular pharmacology division. Thank you so much, Dagia sir, for the opportunity. Thank you.
Okay. Next is Professor BVS Lakshmi, Head Pharmacology, School of Allied Health Sciences, Malla Reddy University, Hyderabad, Telangana, will be the new Joint Secretary of APP Telangana State Branch. Professor Lakshmi, you are here?
Yes, yes, sir. Good afternoon, everyone. Good afternoon, Dr. Rajiv Dahiya, sir. Thank you for giving me this opportunity to serve as Joint Secretary for APP Telangana State Branch. Definitely, I would strive myself that we can bridge the gap between this academia and industry. I would strive myself in Telangana region, because we have number of colleges in Telangana region where this academia is there. Industry collaborations have to go on. So we'll promote more number of conferences. So the gap between the academia and industry can be bridged up. And I'll strive myself to promote research culture in the students by organizing more number of conferences. And because the pharmacovigilance is coming up, we would strive ourselves to promote the students in this culture. Thank you for giving me this opportunity, sir.
Thank you, Professor Lakshmi. Next is Professor Vandana Sharma, Principal, Arya College of Pharmacy, Jaipur. She will be our new Joint Secretary of APP Rajasthan State Branch. Professor Vandana, you are here?
Yes, sir. Good afternoon, sir. Good afternoon, everybody.
You can share your views.
Thank you, sir. Thank you for this opportunity, sir.
Okay. Dr. Gaurav Gupta, Associate Professor, Department of Pharmacology, Suresh Gyan Vihar University, Jaipur, will also join Professor Vandana Sharma as new Joint Secretary of APP Rajasthan State Branch. Professor Kalpesh Gaur is promoted from Secretary to Vice President in Rajasthan State Branch. Professor Narendra Nayola is promoted from Joint Secretary to Secretary of the Rajasthan State Branch. If Professor Gaur or Professor Noela is there, they can share their views.
Hello.
Yes, Professor Gaur.
Thank you, sir, for giving me the opportunity again. Earlier, you have given me the chance to serve as a secretary in the year 2014. And now you promoted me to serve the profession in, or you can say the Rajasthan state. Thank you so much for giving me the opportunity to serve as a vice president, APTI Rajasthan state branch. Thank you, sir.
Thank you, Professor Gaur. Dr. Suresh Kumar, Principal, Bharat Institute of Pharmacy, Kurukshetra, Haryana, is promoted from Vice President to President of the Haryana State Branch. Dr. Suresh, you are with us?
Yes, sir. Thank you so much. Good afternoon to everybody. Thank you, sir, for giving me such a big responsibility. I will try to justify this responsibility for uplifting the academic as well as the research academia along with the Association of Pharmacy Professionals. Thank you so much, sir.
Okay, last announcement is Mr. Tarun Chaudhary, who is lecturer at Dehat Vikas College of Pharmacy, Tegaon, Faridabad, Haryana, will be the new executive member of Haryana State Branch. So these were some of the announcements. We were waiting; they were waiting since a long. So we concluded in our last CEC meeting. So we will move now to our award section. These all are officially minuted, and we will update the information soon on the APP website. So thank you all, and hope you all will serve with more enthusiasm so that we can strengthen APP a lot. So awards are divided into different categories. We will start from the international category awards. First award is Young Scientist Award. And this award goes to a person belonging to America, Dr. Vijay Kumar Shankar, who is DMPK research scientist at C4 Therapeutic Institute at MA, USA. So if Dr. Shankar is with us, he can share his views.
Hi, Dr. Rajiv. Thank you for recognizing me and awarding with Young Scientist Award, and also for appointing me as an international head of industrial pharmacy division. I will strive best to uphold this position and serve as required for the APP. Thank you.
Okay. If anybody want to share the achievements of Dr. Shankar, anybody want to endorse? Professor Banerjee or Dr. Terry? Dr. Terry is with us?
Yes, hi. So Dr. Vijay Kumar Shankar, he
First I want to introduce Dr. Terry. Dr. Terry Gavaskar Ramnanan Singh is the Joint Secretary of APP West Indies International Branch, and he belongs to Trinidad and Tobago. So
Thank you. So yes, I'd like to congratulate Dr. Vijay Kumar Shankar. And I just want to mention a few things about him. He has a PhD in pharmaceutical sciences from the University of Mississippi, whereby he worked on projects funded by the National Institute of Health, as well as the Food and Drug Administration of the United States of America. He is a research scientist at C4 Therapeutics Incorporated in Massachusetts, again in the USA, and he has spent approximately over 3.5 years in drug discovery projects. He has also published over 25 articles, and he also has one US patent. And he has a cumulative impact factor in research of over 100. So he definitely is deserving of the Young Scientist Award for 2022 at this conference.
Thank you, Dr. Terry. So congratulations, Dr. Shankar.
Thank you, Dr. Rajiv. And, thank you, Dr. Gavaskar, for giving a brief note on me. Thank you.
So our next award is Distinguished Teacher Award, and this award goes to again to America, and it is none other than our General Secretary, Dr. Sunita Dahiya, who is Assistant Professor at University of Puerto Rico, Sama, USA. So I will again invite Dr. Terry to brief about Dr. Dahiya.
Thank you. So again, I'd like to congratulate Dr. Sunita Dahiya for her award. She has a bachelor's and master's in pharmacy, as well as a doctor of philosophy in pharmaceutics. She currently actually serves as an assistant professor of pharmaceutics and drug delivery at the Department of Pharmaceutical Sciences in the University of Puerto Rico. She's actively engaged in mentoring and teaching graduate students, so she's quite occupied in enhancing learning. And her interest, of course, includes face-to-face teaching integrated with active learning approaches, such as PBL's problem-based learning, as well as team-based learning. She has published over 64 papers, inclusive of book chapters. And her research interests includes formulation development and drug delivery employing solid dispersion, inclusion of complexation and nanotechnology-enabled approaches. Currently, she is the holder of a research grant on nanocrystal project from Puerto Rico Science, Technology and Research Trust, and this grant approximates to $70,000 US. And she has another research grant of $30,000 US from the National Institute on Minority Health and Health Disparities from the National Institutes of Health. So she's well deserving of this distinguished teacher award for outstanding teaching globally. So I congratulate you, Dr. Sunita Dahiya.
Thank you, Terry. Dr. Dahiya, if you are with us, you want to share some of your experience?
Yes, first of all, I would like to thank APP, the Central Executive Committee, for selecting my application for this award. With my long academic experience, that is about more than 20 years, most of which was in India, serving the academic institutions who were running the programs and the bachelor's and master's. But now I'm engaged with only the graduate students and very less participation in the PharmD teaching, because we are mainly here, we have separate department of pharmacy practice, clinical pharmacy, and separate department for the pharmaceutical sciences. So I'm most of the time engaged for the for teaching and mentoring the PhD students. I am very happy to receive this award, and again I would like to thank CEC for recognizing my efforts and choosing my application for this award. Thank you so much.
Thank you, Dr. Dahiya. Our next award is Promising Pharmacist Award 2022. And this award goes to Miss Stella Giddings, who is pharmacist at Jolie Pharmacy Limited, Bega Branch, Rosseo, Dominica, West Indies. So I will again invite Dr. Terry to brief about Miss Stella.
So I'd like to congratulate, of course, Miss Stella Giddings for her Promising Pharmacy Award. and she's from the Commonwealth of Dominica. She has been a licensed pharmacist for over 28 years, and she has earned a Bachelor's of Science degree in pharmacy from the University of the West Indies. She has worked in different aspects of the pharmaceutical industry, including serving as a clinical pharmacist, and is presently a retail pharmacist. Ms. Giddings is a member of the Dominica Pharmaceutical Society and has served in the capacity of secretary for 8 years and is currently the vice president. She's also a member of the Caribbean Association of Pharmacists and was awarded the Pharmacy Practice Pioneer Award at the 39 Caribbean Association of Pharmacy Conference in Florida. And as a pharmacist, Ms. Giddings is mindful of her ever-evolving practice of pharmacy. So I'd like to congratulate you again for this Promising Pharmacy Award, whereby you have outstanding public service in the pharmacy practice. Congratulations.
Thank you, Teddy. Ms. Stella, if you are with us, you can share your experience.
Good morning, everyone. First of all, I feel honored to be awarded with this award, and I'd like to thank the members of the APP for awarding me and inviting me to be an executive member of this committee. It is indeed a pleasure to be have an active role in the lives of my patients, whereby I help with their day-to-day interests in their health. And I look forward to a successful collaboration with APP, this auspicious international body. Thank you.
Thank you, Ms. Giddings, and thank you for your contribution toward pharmacy practice in Dominica. We appreciate your efforts. Our next award is Promising Scholar Award, and this award goes to Trinidad and Tobago, and it is none other than Miss Whitney Arnaud. Dr. Terry, I'll again call you to brief about Miss Arnaud.
So Miss Whitney Arnaud is a research assistant at the University of the West Indies School of Nursing. She recently graduated with a Master of Science in Medical Biology. And she also has a degree from the University of Southern Caribbean, where she obtained a degree in biology. Ms. Arnaud is a founding member of a research group called the Alpha Research Expedition Team, formed at the University of the Southern Caribbean. And through this group, she has conducted qualitative research among indigenous people living in interior of Guyana, French Guyana, as well as Suriname. Her research focuses on population health with subsection on women's health and medicinal plants. She has also worked at the Caribbean Public Health Agency as a biosafety consultant, and she has presented at the World Health Organization meeting on guidance of biosafety and biosecurity regulatory framework in Geneva, Switzerland. So Ms. Whitney's MSc thesis was on the use of bacteriophage therapy to treat polybacterial infections in Trinidad and Tobago. In 2022, she presented her findings at the Caribbean Association for Clinical Microbiology, and she was awarded for innovative and impactful research. Ms. Anna is currently working on a number of national research projects here in Trinidad, with a focus primarily on maternity care, disaster-related psychological trauma, and mental health training for nurses in the Caribbean. So congratulations, Ms. Whitney, on your Promising Scholar Award for noteworthy research skills in the microbiology field.
Thank you, Terry. Ms. Whitney, if you are with us, you can share your views. We can move to our
Hi,
next
good day,
item.
everyone.
Oh,
I would like to thank. Okay, I would like to thank the members of the APP for giving me this award. I truly appreciate it and feel honored. And I also take this as encouragement to continue on the path of research. And I also like to congratulate APP as well as the Indo-Swiss Committee for the 11th annual convention and the 5th conference, and I hope that you continue to do the good work that you're doing. And thank you so much.
Thank you, Ms. Whitney, for this achievement. Our next award is Unique Student Award, and this award goes to St. Lucia, to the candidate Miss Kiana Gajadhar, who is pharmacist on internship at Owen King European Union Hospital, Castries, St. Lucia. Dr. Terry, can you brief about Miss Gajadhar?
So yes, congratulations, Miss Kiana Gajada. So Miss Kiana Gajada has a Bachelor's of Science in Pharmacy with actual first-class honors at from the University of the West Indies. She is both the valedictorian at the School of Pharmacy this year, as well as the entire of the Faculty of Medical Sciences. It's a difficult award to achieve, but she has become the valedictorian of the entire of the Faculty of Medical Sciences at the University of the West Indies. She is also a member of the Trinidad and Tobago Organization of Pharmacy Students, Caribbean Association of Pharmacists, Pharmaceutical Association of St. Lucia, as well as the Rotaract Club. So I'd like to congratulate Ms. Kiana Gajada for her Unique Student Award 2022 for outstanding academic merit and performance.
Thank you, Terry. If Miss Gajadhar, you are with us, you can share your views.
Good day to all. Thank you to the APP for your selection and the opportunity for this award. As a new pharmacist just entering the field, I hope to continue.
We cannot hear you.
Miss Gajadhar, can you speak loudly?
Hello.
We are unable to hear you.
She mentioned that she had some microphone issues, but from what I heard from her, she did mention that she says thanks for the award, and also in the chat she also gives the APP thanks for the award. So thank you, Ms. Gajada.
Okay, congratulations. So this ends our international award category. So I'm thankful to Dr. Terry Gavaskar for briefing all the awardees. Thank you, Dr. Terry. So now we will move to our next category, which is national category. The first award in this category is APP Lifetime Achievement Award, and which goes to none other than Professor B.N. Suhagia, sir, who is Research Director at Faculty of Pharmacy, Dharam Singh Desai University, Nadiad, Gujarat, and former Dean of same faculty at DD University, Nadiad. I will request President APP Tamil Nadu State Branch, if he is with us, to brief about Professor Suhagia.
Good evening, Dahya sir. It's happy to be a part of this valedictory event. And it's happy to share the biography of Dr. B.N. Suhagia, sir. Dr. B.N. Suhagia is a Dean and Professor, Faculty of Pharmacy, DD University, Nadiad. And he has previously rendered his duty as a professor in the Department of Pharmaceutical Chemistry, LM College of Pharmacy, Ahmedabad. He's an academician with 44 years of experience, and he has guided almost 35 PhD students. He has published nearly 190 research papers in journals and national and international reputed journals. And he has also done attended and presented more than 234 research papers in national and international conferences. And he has also published 2 books and has received nearly 11 awards for research presentations and APP Outstanding Achievement and Best Research Award by APP. He is dynamically involved in research, and he has received research grants from various agencies like BIRAC and GUJAST. And he's also a member of various professional bodies like IPA, ISTE, APTI, APP, FIC, etc. And his former educational activities involve serving as the president and secretary of GPTA, Senate member and academic council member of Faculty of Pharmacy GU, and he's also dean of faculty GTU and chairman of High Power Committee at GTU. And he was also the Senate member of NIPA Ahmedabad. Thank you, sir.
Thank you, Professor Shanmuga Sundaram. Professor Suhagia, if you are with us, you can share your views.
Yes. Thank you, Professor Palani, for a brief introduction. Very good afternoon to one and all. I am an alumni of LM College of Pharmacy, and I attest that the college has played a significant role in my achievement. My assets are my student, positive diffusion of my knowledge. According to me, the successful career of the students are the best achievement. Working as a dean, faculty of pharmacy, and professor at LM College of Pharmacy in past, I have been proactive and successful in resolving research problems in the field of pharmaceutical chemistry and analysis. Being a constant learner, I advise the student to make learning their habit for the qualifiable performance metrics on their resume. And last, I am thankful to Dahiya sir, thankful to APP for bestowing up on me the coveted APP Lifetime Achievement Award. And I also thanks to jury for selecting my name. Award has been increased my responsibility toward the upliftment of the pharmacy profession. Thank you.
Thank you, Professor Suhagia. You truly deserve this award for your lifetime service to the pharmacy profession. Congratulations.
Thank you, sir. Thank you all.
Our next award is Academic Excellence Award 2022, and this award goes to Professor J. Vijayaratna, Honorary Professor at Andhra University, Visakhapatnam, Andhra Pradesh. So, Professor Shanmuga Sundaram, if you can share the CV.
Yes, Professor Vijay Ratna is currently occupied as an honorary professor in AU College of Pharmaceutical Sciences, Andhra University, Vizag. And she is having a teaching experience of nearly 38 years, and she has published nearly 108 articles in peer-reviewed journals, and she has completed nearly 5 research projects, and she has guided 38 PhD students and 100 M.Pharm students, and she has got 3 patents in the field of pharmaceutical technology. She has also received the AP Best Teacher Award in 2018, and she is a life member of various professional bodies like IPA, IPHA, APTI, IGPA, etc. And she is also an ethics committee member in King George Hospital, and is also a member of ethics committee of Andhra University. And she is having various administrative positions, and she is the chairman of Faculty of Pharmaceutical Sciences, Andhra University, and chairman of Board of Studies, Andhra University, and coordinator of PharmD, and significantly involved in conducting medical camps in villages, programs, and competitions for women pharmacists. And she has won various national level prizes in academics. Thank you, sir.
Thank you, Professor Shanmuga Sundaram. Professor Vijayaratna, if you are with us, you can share your views.
Yes, sir. Thank you, sir. I hope you are able to hear me. I'm there. Somehow my picture is not coming, but I am there. My video is open.
Okay.
Thank you very much, sir, for giving me this award. I feel honored. So about myself, I spent 38 years of my career in teaching and doing research at Andhra University. In my research, I tried to develop formulations which are useful to the society. I tried to work with tuberculosis medicines. I developed nanoparticles of tuberculosis medicines. And I developed some other formulations also, for which I got the patent, sir. I am also actively involved in coordinating with PharmD activities in our college, in our university. And I used to take students to medical camps, and I used to very much connect with the society. So that is what I all did, sir. And thank you very much for recognizing my contribution to the field of pharmacy. I am glad to be associated with this association. Thank you, sir.
Thank you, Professor Vijayaratna. You truly deserve this award. This award was nominated by Professor Suresh V. Chandupati, who is our Anal Chem Division Head and presently working at Nalanda College of Pharmacy, Nalgonda, Telangana. Next award is Principal of the Year Award 2022. It is Administrative Award for Administrative Services, and this award goes to Professor Vandana Sharma, who is Principal at Arya College of Pharmacy, Kyukas, Jaipur, Rajasthan. Professor Shanmuga Sundaram, you can brief about Professor Sharma.
Yes, sir. This is the biodata of Dr. Vandana Sharma, and she is having a total professional experience of 16 years in both research and academics. And she is a professor and principal in Department of Pharmaceutical Chemistry and College of Pharmacy, Pratap University, Jaipur for 2 years. And she has also served in Kingdom of Saudi Arabia for 1 year in PharmD course program in Zazan University, and has got various in Jaipur National University, Maharishi Arvind College of Pharmacy, and so on. And she has completed her doctorate in November 2011 and the title Synthesis and Molecular Modeling of HIV-1 and 5-aryl-3 mercapto 1,2,4 triazole derivatives. And she has guided nearly 6 MPharm students and supervised various and co-supervised nearly 4 PhD scholars. And she has got a lot of various analytical skills and professional skills in handling various instruments and various things like ELISA reader, column chromatography, FTIR, SPTLC, and she is a member of various life board memberships in APTI. And she is also the Institutional Animal Ethics Committee member of Pratap University. She is a pharmacist from Rajasthan State Pharmacy Council. Thank you, sir.
Thank you, Professor Shanmuga Sundaram. Professor Sharma, if you are with us, you can share your views.
Hello, everybody. First of all, I would like to thank Dr. Rajiv Dahiya sir and APP members and Dr. Palani sir for such nice introduction. Thank you, APP, for giving me the award and the opportunity. Thank you so much.
I would like to mention Professor Vandana Sharma has hosted our 2 APP conferences at Arya College of Pharmacy successfully, and now she has been given the additional responsibility of Joint Secretary of APP Rajasthan State Branch. We hope the branch will grow well, and we really congratulate for this achievement, madam.
Thank you, sir. Thank you.
Our next award is Distinguished Teacher Award. And this award goes to Professor A. Sridevi, who is Professor of Pharmaceutical Chemistry at Sri Padmavati Mahila Viswavidyalaya, Tirupati, Andhra Pradesh. So, Professor Shanmuga Sundaram, you can share her CV.
Professor Sridevi is an alumni of Kakatiya University, Warangal, and she has nearly 25 years of teaching and research experience. She has served as head of the Department of Institute of Pharmaceutical Technology, Sri Padmavati Mahila Viswavidyalayam, Tirupati, Andhra Pradesh, and presently serving as in charge of head of Physical Science Department. Proving her scientific competence, Dr. Sridevi has completed 5 research projects with a research grant of around 70 lakhs. She has been guiding 8 PhD scholars and guided 6 PhD students. And she has published nearly 60 peer-reviewed publications and contributed 4 chapters in various books from reputed publishers. And Dr. Sridevi is a life member of professional bodies like IPA, APTI, APP, and she is also a BOS member. And chairperson played a various crucial role in PCA approved PG curriculum and also introduced MOOCs course in PG program. And she is a recipient of AICTE CAYT Award, UGC Research Award, and State Best NSS Program Officer Award. and she is a IAEC member secretary of various several institutions, and CPC nominee of Kupam University, and she is also in a NAC body, that is National Assessment Accreditation Council, and she has also visited various countries and presented papers like US, UK, and she is also an editorial member of various peer-reviewed journals. Thank you.
Thank you, Professor Shanmugasundram. Professor Sridevi, if you are with us, you can share your views. Okay, we will move to the next candidate. Next is Innovative Researcher Award. Innovative Researcher Award in female category goes to Professor BVS Lakshmi, who is Head of Pharmacology at Malla Reddy University, Hyderabad, Telangana. I would invite Dr. Saurabh Banerjee to brief about Professor Lakshmi.
Yeah. Good evening, everybody. And so I'm reading a short bio of Professor Lakshmi. Dr. BVS Lakshmi has a total of 20 years of teaching, research, and industrial experience. And she is working as a professor and administrative coordinator and HOD of Department of Pharmacology, School of Allied Sciences, Malla Reddy University, Hyderabad. And she has, to her credit, more than 70 research papers in journals of national and international repute. She has served as a guest faculty in NIPER Hyderabad and has a lot of publications to her credit. And we wish you all the very best, madam, and many congratulations for this award, Professor Lakshmi.
Thank you, Dr. Banerjee. Professor Lakshmi, you can share your views if you want.
Yeah, good afternoon, everyone. Thank you. I'm honored and delighted to have this award being presented from APP. I thank all the committee members for recognizing my CV for getting this Innovative Research Award. I have been doing my research. My research is my passion, and I've been performing several models. And my areas of interest are neuropharmacology, cancer, and pharmacy practice. So I have published various research work which I have done under reputed journals with highest impact factors. So thank you for again giving me opportunity to serve in the research and recognizing my talent. Thank you.
Congratulations, Professor Lakshmi. We appreciate your efforts in the research.
Yes, sir. Thank you, sir.
Congratulations, Professor.
Yeah, thank you, sir.
So next award is Innovative Researcher Award in male category, and this award goes to Professor Kuntal Das, who is none other than President of our APP Karnataka State Branch and Professor at Kripanidhi College of Pharmacy, Bangalore, Karnataka. Dr. Banerjee, can you brief about Professor Das?
Yes, and first of all, many congratulations to Dr. Das. Dr. Das has a very illustrious career of 19 years of his teaching and research experience, and he is specialized in the area of cultivation and standardization of medicinal plants. The list of awards with Professor Das is very, very long, but just to share you, he is the prestigious. He has.
Dr. Banerjee, you are not audible. So I think audio of Dr. Banerjee is not working. So here we have Professor Das to share his views. Professor Das?
Yeah, good afternoon, sir.
Yes, you can share your views.
Yeah, so good afternoon, one and all. So I am very thankful to the organizing secretary of St. Paul's College of Pharmacy, jury members, and especially Dr. Rajiv Daya sir, APP President, for the selection of the research award in male category. I have contributed about more than 115 research publications with the high impact journals and written 15 textbooks and my area of specialization mainly the standardization of the rare and endangered plant species. I'm having around more than 20 years of experience, and for my career, I am done around 27 MPharm research students and one PhD candidate. So thank you for the jury members for selection as a award for the male category in the research contribution. Thank you all.
Congratulations, Professor Das, for this achievement. You
Thank you,
truly deserve the Innovative Researcher Award of the association. Congratulations. Our next award is Young Achiever Award. This award is usually given to candidate who must be a postdoc. So the candidate is postdoc from Korea and have achieved lot of involved in the lot of professional activities, and he is none other than Professor T.S. Shanmugarajan from Wells University, Chennai. He is professor and HOD of Department of Pharmaceutical Biotechnology. Dr. Banerjee, can you brief about Dr. Shanmugarajan? I think we lost connection with Dr. Banerjee. If Dr. Shanmugarajan is with us, he can share his views.
Yes, sir.
Professor Shanmugarajan, you are there?
Am I audible, sir?
Yeah, you're audible.
Yeah. I am humbly thankful to the APP President, Dr. Rajiv Dahiya, and other team members who selected me for qualified for this award. Thank you for motivating me to continue having the highest level of contribution in APP Association. And my special thanks to our director, Dr. P. Shanmugasundaram, who introduced me and encouraged to be involved in the APP. Thank you very much.
Congratulations, Professor Shanmugarajan. So your university, we want to know which university you went to Korea to work with.
Dongkuk University, Medical Science Research Institute.
Okay, you remained how many years?
My area of specialization is stem cell differentiation and involved in the regenerative medicine.
You remained there for how many years?
Yeah, I was there for 1.5 years.
Okay.
I was there for 1.5 years, and I worked in the tissue engineering principles with the application of the biomaterials for accelerated regeneration process. That is genetically modified stem cells for the accelerated regeneration. So I worked and also published several papers, and my highest impact of 13.5, and for the more than 10 impact, we have published several articles. And,
okay, congratulations, Professor Shanmugarajan. You truly deserves the Young Achiever Award of the association, and we really appreciate your efforts, what you have made in the pharmaceutical biotechnology field.
Thank you very much.
Dr. Banerjee, you are with us. You can brief about Professor Shanmugarajan achievements if you are here. No? Okay. Congratulations.
Sorry, Dr. Yeah, I lost the internet connectivity. I'm sorry for that one.
Okay.
Suddenly,
so you can brief about Dr. Shanmugarajan achievements.
Yes, sir. I am there. I am audible, sir, properly?
Yeah,
audible?
You are audible.
Okay. So Professor T.S. Shanmugarajan, sir, is working as a professor in Department of Pharmaceutics in School of Pharmaceutical Sciences, Wales Institute of Science, Technology and Advanced Studies. He has a very illustrious career, and he has a postdoc research from South Korea, and then doctorate of pharmacy from University of Madras, and then he's also MPharm from JSS College of Pharmacy. He is skilled in a lot of, but his main area of research interest are stem cell technology, cell biology, molecular biology. his overall experience in teaching, research, and industry goes back to 22 years, and he has to his credit 53 publications in national and international journal with a cumulative impact of 60 and H index of 14. A very, very illustrious career. Many congratulations, Professor.
Thank you, Dr. Banerjee, for briefing the awardees. And congratulations, Dr. Shanmugarajan.
Thank you. Thank you very much, sir.
So we will move to our next category of awards, which is APP Research Scholar Awards. We have selected different research scholars from different university of different states. So, and one awardee is selected from each state for all the entries. So I will share the details of the awardees. So from Odisha state, the Best Research Scholar Award goes to Mr. Sobhagya Lenka, who is PhD scholar at Department of Pharmaceutical Sciences, Utkal University, Bhubaneswar. So congratulations, Mr. Lekha. If you are with us, you can share your views and experience.
Thank you so much, sir. I would like to very much thankful to my teachers of my department, and it is very honored to have received this award from APP. And I would like to thank the president of APP and all the members of the APP for selecting me as a Best Research Scholar Award. And I would like to thank all the organizing committee of the seminar, in which I have get a chance to deliver my seminar. Then thank you all.
Thank you, Mr. Sobhagya, for getting this award. Best Research Scholar from Uttar Pradesh state goes to Ms. Reena Singh, who is PhD Research Scholar, Institute of Pharmaceutical Research at GLA University, Mathura, Uttar Pradesh. So congratulations, Ms. Reena. If you are with us, you can share your experience. Okay, we will move next. Best Research Scholar Award from Karnataka state goes to Ms. Ratna Baburaj, who is PhD Research Scholar at Rajiv Gandhi University of Health Sciences, Bangalore. And her research center is Department of Pharmacology, Kripanidhi College of Pharmacy, Bangalore. Ms. Ratna, if you are with us, you can share your views.
Thank you, sir. It's a privilege and my pleasure to be part of this convention. And I would like to express my gratitude to the association for the award that has been given to me and for encouraging young researchers like this. Thank you so much.
Congratulations, Ms. Ratna. You deserve this award.
Thank you, sir.
Best Research Scholar Award from Telangana state goes to Mr. Ramu Govindan, who is PhD scholar at Jawaharlal Nehru Technical University, Hyderabad, and also working as lecturer at Department of Pharmacognosy, JSS College of Pharmacy, Ooty, Tamil Nadu. Congratulations, Mr. Ramu. If you are with us, you can share your views.
Thank you, sir. It's a great and memorable experience. I thank the President, Dr. Rajiv Daya, sir, Association of Pharmacy Profession, and Sunita, Madam, Secretary, University of Puerto Rico, USA. And I also thank the St. Paul's College of Pharmacy and the other members of organizing committee. Thank you very much for considering my application and considering me for the award. Thank you very much, sir. It is an opportunity for us to improve much in future. Thank you, sir. Thank you.
Congratulations, Mr. Ramu, for this achievement. We appreciate your efforts.
Thank you, sir.
Best Research Scholar Award for Tamil Nadu State goes to Mrs. Archana Mandwa, who is PhD scholar at Vistas, Pallavaram, Chennai, Tamil Nadu. Congratulations, Mrs. Archana. If you are with us, you can share your views.
Good evening, sir. Thank you so much. Thank you for bestowing me with such an honor and special award. I feel overwhelmed and emotional, and this is a very special moment in my life. I would like to thank my resource management as well as my director, Dr. P. Shanmugasundaram sir, and my guide, Dr. M. Buchanan for their continuous support and led me to win Best Oral Presentation Awards in the past 6 months. And I would like to thank APP for recognizing my efforts. I'm really overwhelmed to receive this award. Thank you so much, sir. Thank you.
Congratulations, Mrs. Archana. We look forward for other achievements like this in future.
Thank you, sir. Thank you.
Best Research Scholar Award in Madhya Pradesh State goes to Mr. Sunny Rathi, who is PhD scholar at Department of Farm Sciences, Dr. Hari Singh Gore Center, University, Sagar, Madhya Pradesh. Congratulations, Mr. Sunny. If you are with us, you can share your views.
For giving me such a wonderful opportunity, like Best Research Scholar Award, Madhya Pradesh State.
You are not audible, Mr. Sunny. I think he's hanged up. Oh. Yeah, you
Yes.
can speak.
Yeah. Good evening, sir. I am Sunny Rathi from Department of Pharmaceutical Sciences, Dr. Hari Singh Gore, Central University, Sagar, Madhya Pradesh. I want to thank Dr. Rajiv Dhaya, sir, and Dr. Sunita Dhaya, ma'am, for this Best Research Scholar Award 2022 at APP 11th Convention and from Madhya Pradesh State. Thank you so much, sir. Thanks a lot.
Congratulations, Mr. Rathi. We appreciate your efforts in research and in future for more achievement of this kind in your career.
Thank you, sir.
Best Research Scholar Award in Haryana State goes to Miss Uditi Handa, who is PhD scholar at MM deemed to be University Mulana, Ambala, Haryana. Congratulations, Miss Uditi. If you are with us, you can share your views. So we will move next. Best Research Scholar Award from Gujarat State goes to Miss Vanessa Richardson James, who is PhD scholar at Gujarat Technological University, Ahmedabad, Gujarat. Congratulations, Miss Vanessa. If you are with us, you can share your views.
Thank you, sir. Thank you so much, sir. First of all, I am very happy to be part of APP and especially Rajiv sir to be I'm part of the APP right now. So thank you so much for this opportunity. And I would like to thank my guide, that is Dr. Hiral Panchal, for her support during the project work. And my DPC members, Dr. Suhagya sir and Dr. Mandev Patel, for their continuous support and encouragement during my PhD work. And I'm immensely pleasured and I'm on an emotional roller coaster for this achieving this award. Thank you so much. Thank you so much.
Congratulations, Ms. Vanessa. You truly deserve the Best Research Scholar Award of the Association. Continue your research activities with more enthusiasm.
Thank you.
Best Research Scholar Award from Himachal Pradesh State goes to Mr. Vivek Pawar, who is PhD scholar at School of Pharmaceutical Sciences, Sulini University, Solan, Himachal Pradesh. Congratulations, Mr. Vivek. If you are with us, you can share your views.
Thank you, sir, for giving me the award. I'd like to thank Almighty God, my family, my supervisor, and the whole team of APP for considering my work and recognizing me as a Best Research Scholar Award. Thank you.
Congratulations, Mr. Vivek. Continue with the same pace.
Thank you, sir.
Best Research Scholar Award from Rajasthan State goes to Mr. Vishal G. Sakare, who is PhD scholar at Faculty of Farm Science, Madhav University, Sirohi, Rajasthan. He is also working as assistant professor at SBS PM College of Pharmacy, Ambajogai, Bid, Maharashtra. Congratulations, Mr. Vishal. If you are with us, you can share your views. Okay, we'll move next. Next category of award is APP Best Student Awards. And from different states, we have selected several students, mostly in the UG category and some in the PG and the PharmD category. So Best Student Award from Andhra Pradesh State goes to Mr. Vamsi Akula, who is B.Pharm student of Nova College of Pharmacy, West Godavari, Andhra Pradesh. Congratulations, Mr. Vamsi, for Best Student Award 2022 from Andhra Pradesh State. If you are with us, you can share your views. Okay, we will move next. Best Student Award from West Bengal State goes to Mr. Mizarul Sheikh, who is B.Pharm student of Department of Pharmaceutical Technology, Brainware University at Kolkata, West Bengal. Congratulations, Mr. Mizarul. If you are with us, you can share your views. We will move next. Best Student Award in Gujarat State goes to Miss Princy Patel, who is B.Pharm 5th sem student of Babria Institute of Pharmacy, Varnama, Vadodara, Gujarat. Congratulations, Miss Princy. If you are with us, you can share your views.
Good evening, everyone. Thank you, APP, for giving me this award. I truly feel honored. This further motivates me to perform my best. Thank you for having me. Thank you so much.
Okay, congratulations, Miss Princy.
Thank you.
Continue your academic activities with the same pace.
Thank you.
Best Student Award from Uttar Pradesh State goes to Mr. Sobhit Chaudhary, who is B.Pharm third year student of Institute of Pharmaceutical Research, GLA University, Mathura. Congratulations, Mr. Sobhit. If you are with us, you can share your views.
Yeah, good evening, sir. Sir, it's a huge honor for me to receive this award for being the best student. I'm deeply honored by the superiors of APP for recognizing my work. And this award will always be a motivation for me to work harder. And a special thanks to Dr. Yogesh Murthy, who guided me through the application process. And thanks again to all the members of the APP for giving me this award. Thank you, sir.
Congratulations, Sobhit, for the achievement, and continue the work like that. Best Student Award in Karnataka State goes to Mr. Rohit Agarwal, who is PharmD intern at Department of Clinical Pharmacy, JSS College of Pharmacy, JSS Aher Deemed University, Mysore, Karnataka. Congratulations, Mr. Rohit. If you are with us, you can share your views.
Yes, thank you so much, Dr. Rajiv sir, for recognizing the work and especially recognizing the young talent. Also, I would like to thank APP, also St. Paul's for giving the platform for young talent. Also, at the end, I would like to thank Dr. M. Ramesh for constant support and mentoring. Thank you so much, sir.
Congratulations, Rohit. Keep it up. Best Student Award from Punjab State goes to Miss Tanya Gupta, who is postgraduate scholar of pharmacology at School of Health Sciences, Central University of Punjab, Bhatinda, Punjab. Congratulations, Miss Tanya. If you are with us, you can share your views.
Very good afternoon to all the dignitaries. It's hard for me to put this feeling into words. Honestly speaking, I never thought of receiving this award for being the best PG student in the state of Punjab. It was my supervisor who pushed me for the nomination of this award, and it was him who believed in me more than I did. This moment is a significant milestone achievement in my life, and I'd like to take this opportunity to thank APP and the prestigious institution St. Paul's and all the concerned authorities who recognized me as the best fit for this award. Awards are tools that encourages us to work harder in life, and it generates immense eagerness to play my part even more dedicatedly. The journey so far till this prestigious award today would not have been possible without the constant support and blessing of my parents. So I'd like to dedicate this award to my parents and to all my teachers and well-wishers. I should not take much time of yours, so I like to stop here by sharing the thought that one should not dream of success, he or she should work for it. Thank you, APP, the organizers, and the entire team. Thank you so much.
Congratulations, Ms. Tanya. Keep it up.
Thank you, sir.
So, this hereby we come to winners of our oration competition, APP Memorial Award winners for outstanding research skills. So, as I mentioned during the competition, so we are giving 4 APP Memorial Awards. First 2 awards are with a cash of 5,000 each, and they are in the memory of 2 of our late persons, professionals. And third award is again a memorial award with a cash of 2,500, and fourth award is with a cash of 1,500. So three of them are sponsored from the APP members, and one is from APP. So we will move one by one. We have, I think, 17 applicants, which were divided into 2 groups. Out of them, I think, 1 or 2 were absent. So from that list, we have selected the 4 potential candidates, and out of them, almost all were those candidates which we have already selected on the basis of their CV and profile base as best research scholar awards from individual states. So this is further filtered to their capability of research skills. And the first memorial award, which is in the memory of late Professor M.C. Goel. So if Professor Suhagia is there, if he can brief about Professor M.C. Goel.
Professor Goel was the principal at LM College of Pharmacy. He served there approximately 40 years. He was a very good
You are muted, Professor. You are muted. Kindly unmute. Yeah.
Dr. Mukesh Goyal was a principal at LM College of Pharmacy, and he served there approximately 40 years. He was a very good researcher, scientist. He has published number of the paper, guided many students for MPharm, PhD, and on his credit there were 11 patents which was the registered and approved. So this award is given on the memory of Dr. Mukesh Goyal, and I saw that along with Mukesh Goyal, he was my colleague, and I can say he was a, for me, friend, philosopher and guide, and he was a principal when I was in LM College of Pharmacy. So thank you APP for giving this award on the memory of Mukesh Goyal.
Thank you, Professor Suhagia. I would like to share one another thing. Today, Professor Suhagia got the Lifetime Achievement Award, and Professor MC Gohel was also recipient of our Lifetime Achievement Award, I think in 2014.
Yes.
So a Lifetime Achievement Award holder is giving to a Lifetime Achievement holder. So I would like to congratulate Professor Suhagia, and it is the opportunity to honor the professionals like MC
Yes.
Gohel, who
So
has
this is a
contributed
good
a lot.
occasion.
So without waiting much, I would like to announce the award of winner, Professor MC Gohel. So this award is sponsored by Professor B.N. Suhagia, which include a cash of 5,000, medal and certificate. And this will be annual award from this annual convention. So this award goes to Mr. Parahars Kumar MR, who is PhD scholar at JSS Aher, deemed to be University Mysore, Karnataka, and is presently a research center. His research center is Department of Pharmaceutics at JSS College of Pharmacy, Ooty, Tamil Nadu. Mr. Prahars, if you are with us, you can share your views. I think he has left. As per my knowledge, his PhD guide is Dr. Natarajan Jawahar, who is Associate Professor of Pharmaceutics at JSS Ooty, and that's all. And he has got 9.25 marks out of 10. So congratulations, Mr. Prahars Kumar. Second award is late Srimati Fulvasa Devi Dubey Memorial Outstanding Oration Award. Srimati Fulvasa Devi is the granny of our APP Switzerland International Branch President, Dr. Sachin Dubey. If he is with us, he can brief about her. I don't know much about Srimati Fulvasa, but just I want to tell that he is sponsoring this award since last 4 or 5 years continuously, and sometimes it goes to for the academic merit, and now this time we have included this award in the memory of Srimati Fulvasa Devi for the research scholar, and we remember Srimati Fulvasa Devi on this occasion. This award goes to Mr. Shantilal P. Padihar, who is PhD scholar and assistant professor at Department of Pharmaceutical Chemistry, Faculty of Pharmacy, Dharam Singh Desai University, Nadiad, Gujarat. Congratulations, Mr. Shantilal. If you are with us, you can share your views. Otherwise, we will pass to Professor Suhagia.
I think he's not there. So Shantilal Padiyar was a student of mine. He did his MPharm from LM College of Pharmacy in 2012. After that, he joined the Pharmacy College at Cambay, and since last 3 years, he joined the Dharamsi Desai University Faculty of Pharmacy, and he is working or he is doing the PhD under my guidance, and he is a very enthusiastic, intelligent, and hardworking. Thank you, Dahiya ji, for giving the award to our faculty member, Mr. Shantilal Padia.
Yeah.
Thank you.
Mr. Padiyar got 9 marks out of 10 as an average of two.
Very good.
Two of
Very
the
good.
persons. So congratulations, Mr. Shantilal Padihar. You should take class about attendance. He has not attended his award ceremony. So, yet the guide are sincere.
So I have to take, sir.
Anyhow, I was joking. Congratulations, Mr. Shantilal, for this late Srimati Fulvasa Devi Memorial Outstanding Orison Award. Third award, which is late Professor Suresh Bhandari Memorial Outstanding Orison Award. Professor Suresh Bhandari, we met in 2014 or 15, and he is recipient of our APP Young Scientist Award. He was principal of St. Peter College of Pharmaceutical Sciences at Warangal, and then he left for Mississippi, USA for a postdoc position, and where he expired due to some reason. If anybody in the attendees want to share something about Professor Suresh Bhandari, that's all what I know about him. Professor Suresh Chandupati. Okay, so this memorial award goes to Mr. Vivek Pawar, who is PhD scholar and assistant professor at Department of Pharmaceutical Chemistry, School of Pharmaceutical Sciences, Sulini University, Solan, Himachal Pradesh. Congratulations, Mr. Vivek Pawar. And if you are with us, you can share your views.
Thank you, sir. Thank you, sir, for giving me the award. I'd like to thank my supervisor, Dr. Deepak Kumar, and the whole team of APP for the award. Thank you.
Okay, Dr. Deepak Kumar is with us?
Yes, sir. He is also in the meeting.
So, Dr. Deepak, you can share your views. Your research scholar got the award. Dr. Deepak? Okay, congratulations, Mr. Vivek, for
you,
the late Professor Suresh Bhandari Memorial Outstanding Orison Award. Congratulations to you, and we memorize Professor Suresh Bhandari on this occasion.
Thank you, sir.
Last award is late Sri Bholanath Nikra Memorial Award, which is sponsored by Dr. Madhu Gupta, who is Joint Secretary of the APP Delhi State Branch. If she is with us, she can share something about Sri Bholanath. She is not there. So we memorize Sri Bholanath Nikra on this occasion, and this award goes to Miss Vanessa Richardson James, who is PhD scholar at Gujarat Technological University, Ahmedabad, Gujarat. Congratulations, Miss Vanessa. If you are with us, you can share your experience.
Hello, sir. I'm audible.
Yeah.
Okay. Thank you so much again, sir, for the opportunity. And I would like to thank my supervisor, Dr. Hiral Panchal ma'am, and again my DPC members, that is Dr. Suhagya sir and Dr. Mandev sir, for the constant encouragement during my PhD work. And again, I would like to thank Dr. Rajiv Dahiya sir for being a part of my starting membership with APP. I am immensely grateful for this opportunity, and would like to be associated in more such occasions in the future. Thank you so much.
Thank you, Vanessa, for your nice words, and congratulations for receiving this award in the memory of Miss Sri Bholanath Nikra, and which is for your outstanding research skills.
Congratulations, Manisha.
Thank you so much, sir. Thank you so much,
So,
sir.
so Professor Suhagia, Gujarat in majority, what is happening? All research is going there, research
You see,
awards.
sir, nowadays India is governing by Gujarat.
And see, is it coming from this Prime Minister's
Yes.
influence? So whole thing is governed by, and you are involved as guide or some part here in
Yes.
Miss Manisha
Yes, yes.
also.
I am
Actually, sir
associated
is my
editor.
DPC
I was
member.
a DPC member, Doctoral Progress Committee member of Vaneja in
Okay, good.
Gujarat Technological Institute.
So your guidance is giving them awards.
Yes, yes, absolutely, sir.
Okay, thank you, Deya
Okay.
sir. Congratulations.
Yeah.
Manisha.
Congratulations, Manisha.
Thank you,
and,
thank you.
thank you, Professor Suhagia. Hosting college always have the award. So hosting college is the St. Paul College of Pharmacy, and these awards are given physically to the candidates in the who are the students and staff members of St. Paul's College of Pharmacy for their research and academic potential. So first award is Best Researcher Award, and that is given to Dr. Sneha Thakur, who is Associate Professor of Pharmacognosy at St. Paul College of Pharmacy. Congratulations, Dr. Sneha. If you are with us, you can share your views. Sorry, it is Best Researcher. So next award is Best Research Scholar Award, and that goes to Miss Mary Manisha Manam, who is a research assistant at Department of Pharmacology at the hosting college. So congratulations, Miss Mary, for the APP Best Research Scholar Award. If you are with us, you can share your views. Okay. Next award is Best Teacher Award, and that goes to Miss Bodhu Suhasini, who is Assistant Professor of Pharmacology in the St. Paul College of Pharmacy. Congratulations, Miss Suhasini. If you are with us, you can share your views. I think they are busy in their award ceremony physically. Best Student Award goes to the MPhom student of the Department of Pharmaceutical Hosting College, Miss Meghna Rothu, and it is also a tie with the another Department of Pharmaceutics postgraduate student. I have not included the details of that, so she might get physically in their valedictory function. Congratulations, Miss Meghna. So before announcing our upcoming APP conference, I would like to say thanks to all the speakers of today's and yesterday's session for their efforts and their time, all the guests, all state branch, international branch, division members who put their efforts in successful completion of this APP annual convention, and appreciation awards are extended to the chairman and principal and HOD of the hosting college. So here is the announcement of our upcoming APP conference. So there is not much gap. Next week, we are organizing Indo-US International Conference at College of Pharmaceutical Sciences, Puri, Odisha, December 23rd and 24th, 2022. And hope to see you who remained absent in this. They can attend that conference. So thank you. Before going away, I want to just invite if anybody among the awardees remained and they want to share their views and they could not join at their time of presentation and now are present. Anybody?
Nobody.
So, anybody is there? Or we can close?
Nobody is there, sir. We can close.
Okay, congratulations all the awardees and all those professionals who put their efforts in the successful completion of this conference, and hope the face-to-face session of this conference will go fine. They might be organizing in parallel. So thank you all. So I'm seeing some of the members here. Miss Whitney, you are with us? Miss Whitney.
Hello. Yes.
Yeah, you spoke when your award was given?
Yes, I did.
Okay, good.
Thank you.
Akula Vamsi. Mr. Akula Vamsi.
Yes, sir.
You spoke when your award was given?
Sir?
You spoke when your award was announced?
Oh, yes.
Okay. So I think all we have covered.
Yes.
So, and congratulations all the new representative of our state branches, especially Haryana and Maharashtra state branch and Rajasthan state branch. So they can do good in their states, and we can organize more and more conferences and honor more and more professionals, including seniors to students. So for their talent in pharma profession. So that's all from my side. I conclude here the virtual valedictory ceremony of APP 11th Annual International Convention and 5th Indo-Swiss Conference.
Okay,
Thank you
thank
very
you,
much
Daya,
for
sir.
giving your time.
Thank you, Daya, sir. Thank you to Sunita, Daya, and thank you, APP. Thank you very much, sir, for taking so much
Thank you
pain
very much.
for organizing the conferences. And I see the Sunita Daya. I saw in a Bhavnagar, in Bhopal, and USA. You so much they improved and so much progress. Your communication
Thank you.
skill is very good. You have improved very good.
Thank you,
Thank
sir.
you, Sunita. I am very happy to see that Sunita. Many times I am talking with the Rajiv, but I am not talking with you.
Yes,
And my
this time.
Suchita is, and I give the name of one you have suggested. Name I forgot, but you give one name to me for reference.
Yes, he is. Yes, well, because two earlier gave consent, but then they refused.
You? Okay.
Yeah, so one professor, he is my colleague, he is my senior colleague. So he told us, Sunita, I will do, don't worry. But other two, they first gave the consent, although they asked us for long time, but
Who has
we will
given the
still.
consent? The name which you have sent, that is right?
That was, no, the consent is our earlier assistant dean of graduate program, Dr. Joseph Bloom.
And recently you told your senior told
Yeah, he is the same person who
same
gave the
person.
consent.
So I can't write his name in this panel.
You can.
Okay.
I think you can, and you can directly communicate because he knows the name of the university. So he has already given the consent. He told me that he has time to review the
Thank you.
thesis. So please go
But
ahead
you,
for that.
Anything else?
You require for pharmaceutics or which?
Pharmaceutical chemistry. Your subjects are.
But I have given you two names, no?
No, but our university wants not of Indian origin.
No,
Every
I have given you other names.
No, you sent to me other names. You have given the one name of one Singh, I think.
I
Indo,
gave you
Trinidad.
Sting from Trinidad. I gave you
Janki.
one from chemistry, Nigel Jalsa.
And if that you are not given,
No, I have
you
given.
send me the name, address, and telephone number of that Jalsa. Okay, sir?
Yeah, I have emailed you the detail
But you
of
mail,
that.
mail me. Okay, no problem.
No, I have
Okay.
already emailed you. First, I
Okay.
gave you Nigel Jalsa, and then I have conversed regarding this Dr.
Okay,
Singh.
I will check it. But again, you email me.
Which one? Dr. Singh information you have?
Jalsa, yes. I will check
Jalsa
my
I don't have.
mail.
Nigel Jalsa I
Otherwise,
can give.
okay. Okay, we will
Okay.
meet again. Nice meeting. I am very
Yeah,
happy to see both Mr. and Mrs. Daya. Okay, we will meet in
Okay.
personally when you will come in the India, and especially I will call to the Nadiyad or Ahmedabad. Okay.
Okay. Thank you, sir.
We will have one conference at Nadiyad when you are in
Sir,
India.
just I forgot to mention, I talked to that Swami Narayan College.
No, I will talk Swami for
21st January we have mentioned one date, so already.
Hiral Panchal told me, but still I have not talked to his management. So I will talk today or tomorrow.
Okay, thank you.
And I will give you feedback. Okay, sir?
Okay.
Can I leave the meeting?
Okay, sir. Yes.
You will be
We
free.
close the meeting. Yes.
Thank you, everybody.
Thank you. Dr. Kiranmai?